Good morning, ma'am Shashi Rekha. Good morning, sir.
गुड मॉर्निंग, कैसे हैं?
ठीक है जी सर.
ऑल वेल.
हाँ जी सर. ठीक है.
ਕੱਲ ਸਵੇਰ ਦਾ ਸੈਸ਼ਨ ਨਹੀਂ ਹੈ YouTube ਤੇ ਯਾਰ. ਹੈ
ਸਵੇਰ ਵਾਲਾ ਸੈਸ਼ਨ YouTube ਤੇ.
YouTube ਤੇ ਨਹੀਂ ਹੈ ਸਵੇਰ
ਸਰ ਚੈੱਕ ਕਰਦੇ ਹਾਂ ਜੀ, ਹੁਣੇ ਚੈੱਕ ਕਰਦੇ ਹਾਂ ਜੀ.
ਹੈ ਨਹੀਂ.
ਓਕੇ ਸਰ.
ਈਵਨਿੰਗ ਵਾਲਾ ਹੈ.
ਅੱਛਾ ਜੀ. ਸਰ ਅਵੇਲੇਬਲ ਹੈ ਜੀ ਦੋਨੋਂ ਕੱਲ ਦੇ ਪ੍ਰੀ ਲੰਚ ਤੇ ਪੋਸਟ ਲੰਚ ਸੈਸ਼ਨ.
ਡਾਕਟਰ ਸਾਹਿਬ ਗੁੱਡ ਮਾਰਨਿੰਗ.
Sir, good morning. Good morning.
ਆ ਦੂਰ ਦੂਰ ਬੈਠ ਕੇ ਵੀ ਪਾਸ ਹੈ.
ਮੈਂ ਤਾਂ ਇਹ ਕੀ ਪੇਜ ਦੀ ਕੱਢੇ ਸੀਗੇ ਮੈਂ.
Hello.
Hello sir, good morning.
Hello Dr. Hibbert. Good morning. This is Avneet.
Good evening.
Oh, sorry. Good evening over there.
Yeah. Good evening.
Nice.
Good evening, sir. I'm
Nice
NSRT from Patiala.
Very nice to meet you.
Yes, sir. Same here.
It's an absolute honor to have you with us, Dr. Hibbert.
Oh, the honor is mine. You're very generous. Thank you so much.
Thank you so much. We have Professor R.S. Singh, who he's from Department of Biotechnology, and he will be chairing this session.
Great. So I'll just stand by. I have my slides ready.
That's good. So what we will do, we will have a brief introduction of the chair and co-chair brief. Then the chair will introduce you and other speakers, and we will start. So it will take about 10 minutes to start with this session formally.
Sure, fine.
Good morning, Dr. DJ Bhatt.
Good morning, Professor. Good morning.
Dr. Bhatt, we have with us Dr. David Hibbert from...
Yeah, I know it, yes.
Yes.
Warm greetings, Dr. Hibbert.
Thank you. It's a pleasure to meet you.
Yeah.
It's a strange way to meet, but it's a pleasure to meet you nonetheless.
I'm very delighted that you have agreed to give a lecture to us, and we know what a great work you have done. Wonderful it is, and nice meeting you.
It's kind of you. Thank you.
Yeah. Because your paper in 2007 really changed the face of fungal taxonomy.
Well, it was not just my paper. There were many co-authors.
I know. I'm aware of that. And, I worked for a long time with Dr. Kevin Hyde. So I know how much of contribution those 49 or 59 authors are there in that paper.
I forget how many. It was a lot.
That was a great paper. Yes.
That was as much politics as it was science.
Yes.
Maybe more politics.
Absolutely, Dr. Hibbert. Absolutely.
Yes. But in any case, that gave us a new impetus for the fungal taxonomy. That's fine.
No, thank you. Yeah, I think that's the one paper I'm most proud of.
Yes, it's absolutely amazing. Such a courage you had to write that. So congratulations.
Thank you. But now horribly out of date, superseded by other works.
Yes.
But still you keep contributing significantly to this cause of mycology, Dr. Hibbert. We regularly go through your references. It's amazing the way things have been presented. Analysis is wonderful. It's really guiding the mycology in the modern context well.
That paper was a turning point, sir. And everybody working in mycology has no option except to follow that paper.
That's true. That's right.
Monumental. Yes.
We welcome Dr. M. Krishna Mohan
Good
from
morning,
Birla Institute of Science, Jaipur. We welcome Dr. Sunil Kumar Deshmukh from Mumbai, who is very actively engaged in mycological research. And so as soon as other members join in, we can start with this session.
Dr. Deshmukh, good morning.
Morning, sir.
Good morning, Sunil.
Good morning, sir. You're not late today.
Yesterday, I kept on trying on mobile.
Hey, actually, what happened that time only there was a problem of the current.
Yes.
Atri Sahab, namaskar.
Namaskar, sir. Namaskar, ma'am. Please.
It's very nice meeting you, all of you here. And it's an amazing platform.
Amazing platform.
I thoroughly enjoyed this, and I must congratulate Avneet in a big way because he did such a wonderful exercise in this. I have no words to explain.
Yes,
really.
sir. Yes, sir. Definitely.
And he and his team, and Madam Kaur also.
Earlier, we were thinking that we'll have a dual mode, online, offline, both, and then ultimately we decided finally, but let's go in for
Fair enough, because it would have been really tough for you to do that exercise.
Yeah.
Because this is not the time where we can really meet and do many things.
Yes.
I wrote a letter to you some time ago. Let's do this online.
Yes. We never wanted to postpone it again because once it was postponed, then it will not happen. There will be a gap.
Correct.
We made it that we should do what may come.
This works very well because Dr. Deshmukh has put such a amazing series of lectures through his Mumbai unit.
Basically, sir, that gave confidence to everybody.
Absolutely.
They led from front and they dragged everybody into it. That is the force. Basically.
That was amazing, and still it is continuous at the same pace.
Yes.
Really wonderful. Really.
And we should do that. Yes.
Yes, sir.
It's a great learning.
Yes.
So many new papers, and everyone contributes in a big way.
And you cannot expect. We are having Dr. Hibbert. We had you yesterday. And
yes.
Visited Langer.
Excellent papers.
It is this platform which has provided us opportunity.
Pradeep, good morning. Yeah, it's very nice seeing all.
Dr. Pradeep, unmute yourself.
Good morning to everybody, Professor Bhatt sir, Professor Atri.
Good morning, sir.
Good morning.
Sir Atri.
Good morning, sir. Good morning.
Dr. Avneet Paul Singh.
Good morning, sir. Welcome to the session. Dr. Hibbert, we have with us Dr. N. Raman. He is the Honorable Secretary of Mycological Society of India. He is Professor Emeritus in Center for Advanced Studies in Botany, University of Madras, Chennai. So he is the Secretary of MSI.
Good morning,
nice
everyone.
to meet you.
Good morning, madam. Good morning.
Morning.
Very nice seeing you today morning. Yes.
Dr. Hibbert, we have with us Professor Manruchi Kaur. She is the Head Department of Botany, Punjabi University, Patiala, and convener of this conference, and she's the team leader under whose able guidance we have organized this conference.
Okay, how do you do?
Hello.
She is muted. She is muted.
Okay, okay.
Hello, sir.
How do you do? He's asking you.
Although being heading the Department of Botany, sir, I am also hardcore classical taxonomist, working in the
Excellent.
laminate mushrooms in the northwest Himalayas especially. And we are eagerly, all my lab is waiting to listen to Dr. Hibbett. Because, you
I'll
know,
try
they
not
have
to
been left
disappoint
their
you.
paper Hibbett at all a number of times. So they're all very eager.
Well, it would be nice to be able to meet them in
Welcome
person
you, sir,
someday.
and for this bonanza of a lecture that you have agreed to deliver in this conference. Hearty thanks to you from the core of my heart, sir.
Thank you. Thank you.
Professor Herbert, welcome to the meeting, as well as on behalf of MSI Mycological Society of India. I welcome you for this conference. And
let's formally start with the session of the day. And on behalf of the organizing committee, I welcome all the honorable members of Mycological Society of India, as well as the leading mycologists. Special welcome to Dr. Hibbett, Dr. Bhagyaraj, Dr. Gurpal Singh Dhingra, Dr. L.M. Suresh, who will be the key speakers in this session. And this is the session number 9 of the 3-day conference, and we will have 4 lead lectures in this conference. Now, in today's session, we have with us Professor R.S. Singh, who is currently Professor and Head Department of Biotechnology, Punjabi University, Patiala. He will be chairing this session. Welcome to the session, sir. And we have with us Dr. M. Krishna Mohan, who is from Biotechnology Division of Birla Institute of Scientific Research, Jaipur, currently associated with the Bioinformatics Center of this great institute. We welcome you, Dr. M. Krishna Mohan, to this session.
Thank you very much.
Before starting with the proceedings and handing over the session to the chairperson, I would like to have the privilege of introducing the honorable chairperson and co-chairpersons. Professor R.S. Singh, he is currently Professor and Head Department of Biotechnology, and he is also having the charge of, and he's also the Dean Faculty of Life Sciences, Punjabi University, Patiala. His research interests, they include protein and carbohydrate biotechnology, and he is contributing immensely in the research on industrial enzymes from microbial sources, microbial exopolysaccharides, and microbial lectins. He has contributed significantly to the industrial microbiology. He is instrumental in the development of processes for the production and purification of industrial enzymes, microbial exopolysaccharides, and their applications in food and fermentation industry. He is the product of Punjabi University, Patiala. He did his MSc, MPhil, and PhD from this very university, and joined the Department of Biotechnology in 1998, and he was elevated as professor in 2009, and since then he is there in the Department of Biotechnology as the senior professor. He has to his credit number of fellowships, the award of visiting scientists, and recognitions. He is visiting professor to University of Clermont, France. He was visiting professor to Swiss Federal Institute of Technology, Switzerland. He was an INSA visiting scientist in 2008. The list goes on. It will be even half an hour will not be sufficient to list his accomplishments. He is on the professional. He's having the professional membership of so many societies: Fellow of Biotech Research Society of India, Life Member of Indian Science Congress, Association of Microbiologists, President of Biotech Research Society of India, Unit Mumbai, Unit Patiala, Punjabi University Patiala. He also is having honorary consultancy with the industry, Mount Shivalik Breweries in Patiala, Rangar Breweries, Mahatpur, Una in Himachal, Pepsi Food Limiteds. So he is equally involved in translating his research to the industry. He has been on the editorial panel of so many journals, to name a few: International Journal of Food and Fermentation Technology, Biologia, Journal of Scientific and Industrial Research, Indian Journal of Experimental Biology, Applied Biochemistry and Biotechnology, and Journal of Punjab Academy of Sciences. To his credit, he has 2 patents, 5 books, as many as 32 chapters, many book reviews, a number of technical reports, and nearing 100 publications. They are to Professor R.S. Singh. And it is a matter of honor to share with you that he has been in that list of top 2% scientists of the world, which has been recently released. And he is also the Fellow of Mycological Society of India. With this brief introduction, I welcome Professor R.S. Singh to this very session. Welcome, sir. We also have Dr. M. Krishna Mohan, who is from Bioinformatics Center, Birla Institute of Scientific Research, Jaipur. Dr. Mohan did his master's and doctoral degrees in microbiology from GB Pant University of Agriculture and Technology, Pantnagar. He joined Tata Energy Research Institute and has been associated with the Microbial Biotechnology Group in various capacities over a decade. He has been a member of an international group that worked on innovative bioprocesses for energy generation from solid waste. He was also involved in UASB reactor for treating liquid waste of a food processing unit. He was an active member of much acclaimed TERI project on Green India 2047. Since 1997, he has been with Biotechnology Group at Birla Institute of Scientific Research, and he's continuing his work on the environmental mitigation. He handled a number of projects, particularly dealing with decolorization of textile dye effluents. He is actively involved in microbial exopolysaccharide production technology, its demonstration and transfer of technical know-how to private sector companies. Presently, he is involved in assessing microbial biodiversity of salt lakes and deserts following the principle of polyphasic taxonomy. He has co-edited a book, has to his credit number of publications, and we know Dr. M. Krishna Mohan for his management skills. We have been witness to a very well-organized Mycological Society of India's conference that took place in Jaipur. So with this brief introduction, we welcome Dr. M. Krishna Mohan to this session. Welcome, sir. So now I would like to hand over the stage to Professor R.S. Singh for further proceedings. Professor R.S. Singh.
Good morning, all of you. I welcome all of you in 9th session of the 3 days National Conference of Mycological Society of India. In this session today, we have 4 speakers, and the first speaker is Professor David Scott Hibbett. He is Professor of Biology from Clark University, Worcester, and in his credit, he has so many awards. It is a long list of honors and awards like Mycological Society of America, Mycological Society of America, Lexoconus Prize, Clark University Liver and Dorothy Hayden Junior Faculty Fellowship, Clark University Gordon King's Junior Faculty Prize, Mycological Society of America Honorary Fellowship, Warren Litsky Edodes Chair in Biology, Clark University. So for Dr. Hibbett is concerned, he is on the editorial board, so many of so many international journals. He's a book review editor of Mycotaxon, associate editor Systematic Biology, associate editor Mycologia, Mycoscience, Mycological Research, Mycological Progress, editorial board member of so many other journals like Fungal Science, Molecular Phylogenetics and Evolution, associate editor of Molecular Phylogenetics and Evolution, Applied and Environmental Microbiology. So Dr. Hibbert, he's also the steering committee member of Fungal Environment Sampling and Informatics Network, Research Coordinator Network in 2008. So in his credit, he has a long list of publications, and he has the very high H index, 66, and I-10 index, it is 129. So total number of publications he has published, 142, which including research papers, review articles, short communications. He is the authority in the mycological taxonomy. I welcome you, sir, for this national conference. Kindly, you can now start your lecture.
Okay, thank you very much. Let me see if I can get this straight. So it's really a great honor to be here. So thank you very much for the kind introduction. Thank you for welcoming me to this very interesting conference. I, of course, wish that I could be with you in person, but that's just not possible today. I don't know if you can see this background behind me on the video, but this is a recent photograph of Clark University, my workplace. So this is a photo just taken a few days ago. We are in the depths of winter here, so we have lots of snow on the ground, and it's the middle of the night, so rather different situation than what you were dealing with. But it's a great pleasure to be here. So I'm going to start my slides, and I trust you will tell me if you can see them.
Yeah. It's perfect, sir. It's perfectly visible.
Great. Okay. So, so when I was invited to come and give this talk today, I had a challenge to decide what to speak on because I've been doing a lot of different things over the years. Most recently, a lot of my work has been in the area of sort of comparative genomics. But at heart, I'm really a fungal systematist. That's really what I think of myself as. And this is one of my favorite groups of fungi. This is Lentinula, the shiitake genus, which I've been working on for a very long time. And so I thought I'd give you a little bit of an update on my work on Lentinula, as well as a little bit of background in history. So let's see. So I'm going to break the talk into 4 parts. I'm going to talk a little bit about background and taxonomy, including some of the early molecular work I did in this group. In the second part of the talk, I'll give you an update on some studies on phylogenetic diversity using a large data set of ITS and TEF1 sequences. The third part will be the really new material on phylogenomics. So we have a new project on phylogenomics. None of that work is published yet. And then I'll conclude with some outstanding mysteries that remain in the genus. And then I hope that there'll be time for me to take questions. So I think you all know that Lentinula is the shiitake genus, contains the species Lentinula edodes, known in Japan as shiitake, pyogo in Korean, or shangu in Chinese. And this is certainly one of the world's most famous mushrooms. So you could say that Lentinula may be one of the world's most familiar genera of mushrooms because it contains this very important edible cultivated species. Lentinula is now the #1 most highly produced mushroom in the world, according to this 2017 publication by Dan Royce. And most of the recent production has been driven by activity in China. So over 1/5 of all mushroom production is shiitake. Of course, the fungus goes back, it's been known a lot longer than that. So this is where shiitake was published in 1875 by Berkeley in England using material that was collected in Japan on the voyage of the Challenger. Berkeley put this in agaricus, which all gilled mushrooms were at the time, although into Armillaria in parentheses. In the 20th century, shiitake has had a lot of taxonomic attention paid to it. Rolf Singer, the great Rolf Singer, put shiitake into Lentinus. He had an odd concept of what Lentinus is, but he was convinced that shiitake was in Lentinus, and so you still find shiitake referred to as a species of Lentinus. But the person who wrote the monograph on Lentinus, David Pegler, transferred shiitake into the genus Lentinula in the Tricholometacea. So two conflicting placements. For my PhD work, I studied the genus Lentinus a very long time ago, and it was fairly clear from anatomical data that shiitake was not in Lentinus in the strict sense, but to do due diligence, I included it in some of my early molecular data sets. And this is a tree from a paper that I published a very long time ago, 1993, and we showed that Lentinula edodes was closely related to what we were then calling Colibia early E, now Gymnopus early E. So in the Calibii of the Tricholometacea, just where Pegler said that it should be, far away from Lentinus, which is in the Polyporales. So I would have been happy just to leave Lentinula alone at this point and go on to focus on Lentinus. But for my first postdoc, right out of graduate school, I found myself at the Tottori Mycological Institute in Japan. And this is me with some of my colleagues from Tottori in 1991. And at Tottori, they're very interested in Lentinula because they're largely funded by the Japanese shiitake industry. So I found myself being drawn back into studying taxonomy of Lentinula with the encouragement of my friends from Tottori. So let me go back quickly to the monograph by Pegler. This is the only monograph that we have of Lentinula from 1983, so it's a little out of date. And he recognized 5 species in the genus with a very strong geographic component. So 3 of the species were in Asia and Australasia. So edodes, which I've mentioned and shown you a picture of. Also Lentinula lataridia from what he called Southeast Asia, and Lentinula novizelandii from New Zealand. So I'll show you just a couple quick pictures of those taxa. These are the sketches from Pegler's monograph. So edodes, novizelandii, and lataridia. Here's a photograph of Lentinula lataridia from Australia, but you can also find this in Tasmania, Papua New Guinea, and elsewhere. So it's got this classic sort of cinnamon brown, thin-fleshed pileus, but it smells and tastes like shiitake. And here's Lentinula novizelandii, which has this very fuzzy stipe and a sort of fuzzy pileus as well. And this is restricted to New Zealand. So one of the first projects I did on Lentinula was a study using ITS sequences focused on the Asian and Australasian taxa. And this is now a rather old paper, but we recognize 5 lineages or groups in Asia, Australasia that might correspond to species. So one that we called group 1, and you're going to hear these group numbers again and again today. Group 1 from Northeast and continental Asia, so Japan, China, Korea, also Thailand, and also Borneo, so getting down into the tropics. Group 2 corresponds to Lentinula lataridia in part, and we had samples from Papua New Guinea and Australia. Group 3 is Lentinula novizelandii from New Zealand. An interesting group that we're going to talk about more later that we call Group 5, Lentinula affine edodes, something like edodes, at this time represented only by sequences from Nepal and also from China. And then the last group, Group 4, another group from New Guinea. So 2 groups sympatric in Papua New Guinea. Now, I thought that these might warrant recognition as species because they look like independent lineages. But my colleagues in Tottori were very resistant to that idea because all of these taxa or isolates from all of these areas have been shown to be mating compatible, with the exception of group 4 from New Guinea. So this is one of the 2 groups from New Guinea. This is a crossing diagram, and you can see that group 4 is reproductively isolated from group 2, which is the other group in New Guinea, and it's partly compatible with the other groups that occur outside of New Guinea. And everything else is compatible. So there's starting to be some reproductive isolation here based on laboratory studies. So my colleagues in Totori conceded that the shiitake populations in Asia, Australasia are in the process of speciation. This is from a paper by Shimomura and colleagues published in Mycoscience. So that's the Asian and Australasian material according to Pegler, and in the light of some of my older work. Pegler recognized only 2 species of Lentinula in the Americas, Lentinula boreana and Lentinula huarapiensis. And I'll show you pictures of those. Here's the sketches from Pegler's monograph. So here's boreana, and here's huarapiensis. And this species is only known from the type collection from the 19th century by Spegazzini from Paraguay. So I'll show you a picture of a specimen later on. It's still rather controversial whether or not this is actually in Lentinula. So really the most widely distributed species of Lentinula in the Americas, according to Pegler, is this thing Lentinula boreana, which had a very broad distribution across South America, Central America, the Caribbean, and even up into the Gulf Coast of North America. So Juan Mata and Ron Peterson worked quite a bit on the Lentinula of the Americas. And based on ITS phylogeny and also mating compatibility, they carved out 2 new species, Lentinula raphanica from the Gulf Coast of North America through Central America and the Caribbean, all the way down to Brazil and Venezuela. And then also something called Lentinula aciculospora, which at that time was known only from Costa Rica. So 3 species in the Americas besides huarapiensis. So here's raphanica, boreana, and aciculospora. So just to sum up, this is the background. For the last 20 years or so, there have been about 8 lineages recognized in Lentinula that might correspond to species, but there has not been any formal taxonomic activity in Lentinula to carve out new species since Mata and Peterson's work. So now what I want to do in the second part of the talk here is show you some more recent work using ITS and also TF1 alpha sequences with a much larger sample of data, including many sequences taken from GenBank. And this work is done by many people. This is Nelson Manoli, who's a Brazilian mycologist who's really been leading the charge on this work. This is another Brazilian mycologist, Noemia Ishikawa, who's been doing really important work in ethnomycology in Brazil, showing how the Yanomami people, the indigenous people in northern Brazil, use mushrooms, including Lentinula. Chao Chung Wang is from Guangdong, China. Marisol Sanchez Garcia is now in Uppsala, Sweden. These folks are members of my lab, and then these are other collaborators, and I'll show you pictures of some of them later on. So working with all these people, we've performed new analyses, which we're getting ready to submit for publication shortly, using 340 ITS and 116 TF1 alpha sequences, including about 70 new ITS and 53 new TF1 alpha sequences, and then a bunch of data from GenBank. We also extracted ITS and TF1 alpha sequences from some unassembled genomes, from some unassembled genomes from China by Yang Xiao, and I'll be talking more about that data set in a little bit, a little later on. So with this expanded data set, we're now seeing what had been 8 groups, we're now up to about 13 groups, the same 5 in Asia and Australasia, and 8 in the Americas. So I'll just give you a little sense of where those taxa occur. So this is these are the Asian, Australasian lineages here. So here's group 1. So the vast majority of our samples are group 1 edodes mainly from China, also Japan, Korea, Thailand, the Russian Far East, Vietnam, the Bornean material I mentioned. Also samples from India. So there are group 1 edodes in India, as well as the Philippines. I'll just mention group 5 now, since it's right here next to it. In this tree, group 5 is the sister group of group 1, and it includes mainly samples from China, but also India and Nepal and Vietnam. And then the others are more or less as we found them before. So group 2, Lateridia, New Guinea and Australia, group 4, Lateridia, also in New Guinea, and then group 3 still in New Zealand. So here's a cartoon of the whole Lentinula phylogeny based on this expanded ITS TF1 data set. And I'll focus on the Asian Australasian things for now. So this is the combined ITS TF1 data set. Group 1 and group 5 are sister taxa, sister lineages in this combined analysis. But when you look at the genes independently, they don't tell the same story. So ITS alone does not support the monophyly of group 1 plus group 5, and TEF1 alone does not resolve the species of group 1 and 5 at all. They have virtually identical TEF1 alpha sequences. So this is a hint that something is going on, some conflict there, which we'll come back to when we talk about the genome data. And this is the geographic distribution. So it's very similar distribution to what you saw before, only now it's more densely sampled, particularly in the group 1 and group 5 lineages. Okay, turning to the Americas, we now have 8 groups. And you could consider them sort of Lentinula raphanica sensulato with 2 groups. The main one, group 6, in very widely distributed from Brazil to Colombia, Costa Rica, the Gulf Coast of North America, and the Caribbean. The Lentinula boreana species complex here with now perhaps 4 independent lineages in Central and South America. And then Lentinula aciculospora in the broad sense, mostly from Costa Rica, but 1 sequence we have from Ecuador. So it looks like aciculospora now is spilling into South America. And Lentinula aciculospora is a species we'll be focusing on particularly in just a few minutes. Okay, and so this is the geographic distribution of Lentinula species in the Americas, so a very complicated biogeographic picture. Nelson and colleagues did some molecular clock analyses, which suggested that Lentinula as a whole is about 53 million years old, roughly, with broad confidence intervals, as one often has in beast analyses. The American lineages, which here in this analysis includes Aciculospora, Boreana sensulato, and Raphanica sensulato, would be about 43 million years old. And the Asian Australasian lineages, much younger, only about 18 million years old. So the Asian Australasian things look like a much more recent radiation compared to the things in the Americas. I'll focus for just a moment now on host range. Shiitake in Japanese means oak mushroom. So I think many of you may be aware that shiitake mushrooms tend to occur on the Phagales. So Phagaceae, Nothophaгаceae, Quercus castanopsis, Lithocarpus, things like that. With the exception of the Lentinula raphanica group, which has a very wide species host range. So it's on 7 different orders of angiosperms, but not on the Phagales. So that's unusual. So this looks like a recent expansion in terms of its host range. And then group 13 of Lentinula aciculospora, for which we have only 1 sample, is on oxalidales in a species of Cunoniaceae. So summing up that part of the talk, based on ITS plus TEF1, we now think that there are up to 13 lineages in Lentinula that might warrant recognition as species, 5 in Asia and Australasia, and 8 in the Americas. And so now we'll take a look at what we get when we look at genome data. Okay, so the next part will be some recent, ongoing work on phylogenomics of Lentinula. And this is work that, again, involves a big team of people. Many of those you saw on the previous slide for the ITS work are involved in the phylogenomics. Our genome work is done in collaboration with a US Department of Energy research facility called the Joint Genome Institute in California that has been doing an awful lot of fungal genomics. And then these are collaborators. Kazuhisa Terashima is from Totori Mycological Institute. Karen Hughes and Ron Peterson at the University of Tennessee. Juan Mata is at the University of South Alabama. John Gibbons at UMass Amherst. And Chris Smith is in New Zealand. And then there are others as well, and 17 collaborators involved in this comparative phylogenomic study. So at this point, we've generated 25 new genomes from Lentinula, and we've imported 4 published genomes. So three genomes of Lentinula edodes we've imported, 1 of Lentinula novi-zelandii that Chris Smith kindly provided, and then the rest that are not highlighted are all new things that we've generated. And we've really tried to span the diversity of Lentinula with our genome sampling. So we've used that ITS and TEF1 phylogeny to guide our sampling of genomes. We have 11 of the 13 ITS groups represented in our genome sampling and really across the entire geographic range of Lentinula. And if you're interested in working with the data, you can go to the JGI Mycocosm webpage. The URL is here, but you can just Google Mycocosm and Lentinula, and it'll take you to the page where you can get to the data. The genomes are fairly typical genomes for agaricomycetes. They're around 33 to 49 megabases, and they have 12 to 16,000 genes. So they're very, very typical basidiomycete genomes. Not too big, not too small. Here's the first phylogeny based on genomes that I'll show you. These are all species of agaricales with some bolatales and amylocorticeales and etheliales outgroups. Here's Lentinula, and what this result just proved to us is that Lentinula is in fact a monophyletic group with colibioid taxa closely related as outgroups. We'll zoom in a little bit closer now with just the colibioid things as the outgroups. And the one thing I want to point out here is Lentinula aciculospora, but it's not the sister group to the other taxa in the Americas. So Lentinula boreana sensulato and Lentinula raphanica sensulato. Instead, Lentinula aciculospora from Costa Rica comes out as the sister group of the Asian Australasian lineages. So this contradicts the ITS phylogeny and was very surprising. So Miguel Naranjo Ortiz, who was photographed in the previous slide, postdoc in my lab, did a series of topology tests using the genome data, trying to test that placement. And the only placement that could not be rejected with 2 different data sets and different kinds of tests was the placement of aciculospora as a sister group to the Asian Australasian lineage. So a position as sister group to the taxa in the Americas was rejected, and a position as the sister group to all of Lentinula was also rejected. So the topology tests reject monophyly of Lentinula in the Americas, and they support the placement of Lentinula aciculospora as a sister group of Lentinula in Asia and Australasia, even though this is from Costa Rica. So that's kind of a big picture look at Lentinula based on genomes. I'm going to zoom in now on group 1 and group 5, these things from continental Asia, mostly, so China, Korea, Japan, but also Nepal and India. And for that purpose, we're going to take advantage of some data that were published by a Chinese mycologist named Yang Xiao in Wuhan. And he published a paper in 2016 where he had 60 genomes of Lentinula edodes from China. Cultivated things with the red dots and wild things with the green squares. And this is a SNP-based tree rooted with Coprinopsis, which is kind of an odd outgroup choice for that purpose. We got a hold of these data with Yang Xiao's support and we assembled the genomes because these were unassembled and called genes and put them into a phylogenetic analysis. So now this is a tree with 77 genomes, 56 from that data set of Yang Xiao, 19 of the JGI genomes, and 2 published things. So this is a 415 gene tree. The isolates with the red lettering are cultivated. The isolates with the blue lettering are wild. The G1s are the G1 type ITSs. The G5s are the G5 type ITSs. And what this analysis shows is that all of the cultivated strains have this G1 type ITS. G1 plus G5 form a strongly supported clade, but neither G1 or G5 form a monophyletic group. So this showed us that there's some conflict between the ITS groupings and the phylogeny based on whole genomes, based on SNPs. So this is another tree showing you many of the same taxa. This is now a tree made with a program called Astral that uses gene trees as input and gives you a phylogeny. The bars here are outputs from admixture analyses. This is done by my PhD student, Sean Pativ, shown here. And what you can see is that there's a group here that contains G1 and G5 mixture, which is fairly homogeneous. And then another group that has 2 major populations, one in purple, one in green, with some admixture between them, suggesting some kind of pattern of hybridization or introgression somewhere. The asterisks are the cultivated isolates, and the lineages without isolates are, without asterisks are the wild isolates. This is a phylogeny splits tree analysis based on the SNPs that shows you the same data basically, and it resolves within the group 1, group 5 clade 4 different clusters, one containing most of the cultivars, which is this group here, another containing one single cultivar, which looks like a recent domestication event, and then this cluster of isolates with both the G1 and G5 type ITSs. So again, showing us that there's something going on with those ITS lineages. They're not consistent with the genome-based phylogeny. Now, there's a caveat here, which is that when we do PCR, we pull out a single kind of ITS per isolate. And when we use bioinformatic methods that create consensus sequences of genes, we also pull out a single ITS type from each isolate. But when we look at the individual reads from a single genome sequencing run, we see heterogeneity. So this is one of the isolates that was identified as having a G5 type ITS, but each of these colored bars shows you a heterogeneous site where there's variation among the individual reads. So there's clearly some intragenomic heterogeneity here. We don't know if that reflects recent introgression or hybridization or some kind of ancient polymorphism in ITS. So we're just getting around to this now. We don't know what the real story is, but there is certainly some conflict. So the conclusions for the phylogenomics part are that the genome data largely support the ITS groupings, except that we place Lentinula aciculospora as the sister group of the Asian Australasian lineages, and we reject the monophyly of these ITS-based groupings fairly conclusively. Okay, I want to wind up now with some outstanding mysteries. And I think the take-home message of this part of the talk is just that even in a very well-studied famous genus like Lentinula, containing the #1 most widely cultivated mushroom in the world, there are still a lot of interesting taxonomic problems remaining to be solved. And so here's one that I mentioned earlier. This is Lentinula huarapiensis, known only from Paraguay, collected by Spegazzini. This is a photograph of one of the collections from one of the exicati that he made. This does not look very much like a lentinula to me, but who am I to argue with Pegler? So maybe this is a lentinula, but we need more collections, more basic field work from South America. Recently, a species of lentinula was described, well, 2011, from South Vietnam. This is the tree that was produced based on large subunit ribosomal RNA sequences. It's really not very well resolved, but this certainly is something that requires more analysis, but we don't have any material of this lineage, whatever it is. Also, if you go to GBIF and you search on lentinula, as I have done, and you look all over the world, you find that lentinula is reported from Africa. So there are some collections of Lentinula edodes or a Lentinula edodes-like taxon from the Democratic Republic of the Congo. I have not seen this material. It's in a herbarium in Belgium, but this is very interesting because this would be the only known material of lentinula from the African continent. Just offshore of the African continent is Madagascar. This is Bart Bike, who's been doing work. He's in Paris. He's been doing work on the fungi of Madagascar for some time, working on edible mushrooms. And in 2008, he published a paper where he reported finding Lentinula edodes in Madagascar. And this is the collection. This was in the title slide for this talk. You might have thought this was shiitake. It certainly looks like shiitake. It's a lentinula for sure. It was found on Eucalyptus robusta. It's also possibly on Uapaca, which is an indigenous tree that was the dominant tree in the forest where this material was collected. There are no Fagaceae native to Madagascar. So we got that material recently. This is Brian Looney, a postdoc in my lab. This is a large subunit tree that places the Madagascar material firmly within lentinula in the Omphalotaceae. And this is now a tree based on ITS and TEF1, which places this Madagascar material as the sister group now to Lentinula aciculospora from Ecuador and Costa Rica with moderate bootstrap support. But we don't trust this because the placement of aciculospora is not well resolved with ITS and TEF1. So we feel like we really don't know yet where this Madagascar material goes, but we are going to describe it and hopefully get a genome sequence shortly. So in any case, this is a 4,000-mile range extension for Lentinula. The closest relative of this Madagascar material is in Costa Rica, over 8,000 miles away. The closest possible material is in the Democratic Republic of the Congo. So it's likely that these things will prove to be closely related. But even if they are, this material probably got to Madagascar by long distance dispersal because Madagascar has been separated from Africa for about 150 million years. Okay, with that, there are many people to thank. As I think should be evident, this is a large international collaboration. I'm really grateful to everybody who's contributed to this project. There's still quite a lot to be done in Lentinula. There's lots of Lentinula in India, Nepal, Bhutan, elsewhere. So I hope that you will be looking for Lentinula in your fieldwork. And I will stop sharing now, and hopefully there is time to take questions.
So any queries from delegates?
Can I ask a question?
Yeah.
I'm Krishna Mohan from the Line for Scientific Research. Excellent talk, Professor Heber. So I just want to have an opinion from you. With the availability of the whole genome sequences, would you recommend a kind of MLSS for fungal taxonomy, where we can do targeted deep sequencing instead of only ITS?
I'm sorry, was the question about the impact of genome sequencing for taxonomy?
Yeah.
Yeah. I think, well, I think it's really clear that fungal genomes are, there are so many fungal genomes coming out these days. And fungal systematics has been transformed by genomics, but that does not mean that fungal systematics is genomics. I think the challenge for us is to integrate the genomic information with all the other information that we have to use in fungal taxonomy. So this huge database of ITS sequences that exists and the multi-gene phylogenetic analyses, you know, if you put that together with the genome data, then you can end up with very large trees with very robust backbones. So I'm hesitant to advise everybody to go charging into genomics, which seems to be happening, because there's just so much basic biodiversity sampling we still need to do, and there's so much work we need to do going through many, many isolates, getting molecular data, ITS, and others. So the genomes give us a very good skeleton, you know, they give us a framework, but to flesh that out, you need to have all the other data as well. Excellent question, thank you.
Excuse me, sir. I am N.S. Atri. We have been going to Northwest Himalayas for the last so many years, but we could not, we could not encounter any material of Lentinula, especially what to talk of Lentinula edodes. I don't know, we may have escaped it. We'll try to look into it. I wish to have one clarification that there are, we have been following during our times Singer's agaricales in modern taxonomy, and I have been collaborating with Rolf Singer for quite a long time, even with Dr. Pegler. So where do these taxa stands now vis-a-vis multi-gene taxonomy approach?
You know, Rolf Singer and David Pegler did their work at a time when molecular data simply were not available, right? And I look back at their work, and they had tremendous insight. You know, they were able to look at anatomical characters and make predictions based on really very thin evidence, and frequently they were right. So Pegler's concept of Lentinus, even though the genus turned out to be polyphyletic, you know, his idea that they were derived from polypores turns out to be absolutely correct. Similarly with Singer, you know, if you take the agaricales in modern taxonomy, at a high level, there are many groupings that are not supported today, but he got an awful lot right. You know, it was really quite remarkable. One of my mycological heroes is M.A. Donk, who did work on Aphelophorales, and that man had incredible insight. He really got a lot of the groups right based on anatomical evidence. And to make those kinds of inferences, you just really have to know the taxa deeply. You have to be familiar with the anatomical characters of many, many things. Very few people have that kind of knowledge now. I certainly don't, for example. Yeah, so I'm very impressed by the work that was done by people like Singer and Pegler in the absence of molecular data, in the absence of phylogenetic methods.
In addition to that, what suggestion you will give to that new entrants into the field of taxonomy, which what they should follow, basically, besides traditional techniques along with molecular techniques, if they reach a right conclusion based upon morphological, anatomical, and biochemical characteristics, do they still need to go in for phylogenetic analysis?
I think it's really hard to be a systematist now. I think being a fungal systematist these days is an incredibly challenging thing because you really need to be on top of all kinds of data. I mean, a young person starting out now needs to understand phylogenetic methods, needs to be aware of phylogenomics. That doesn't mean they have to be doing it, but they need to be on top of what the data, what it means. Also, to be a systematist, you have to know the groups, right? So you have to, and that's really hard. So it's just a difficult thing. I think what I see is a fair amount of technophilia in systematics right now. There are a lot of people who feel like they have to be doing genomics. I don't think that's necessarily true. I think the most important thing is to know the organisms and have a good question, and then the appropriate methods will suggest themselves. It's hard to be a systematist these days because the field is so broad. I'm glad I'm not just starting out right now. It would be very challenging. One thing that I think is certainly true is that we're not going to see another agaricales in modern taxonomy anytime soon. I think the days where one researcher could write a giant book that goes from the species level to the most comprehensive level, I don't see that happening again. There's just too much data. There are too many different approaches that need to be brought to bear.
Sir, can I have one question, sir.
Yeah.
How do you foresee environmental sequences with novel species getting integrated into the mainstream species descriptions through the morphological descriptions and the cultural ones from the uncultured, cultivatable? How do you see their integration?
I think that's a very, very hard problem. So are you talking about environmental sequences where you have sequence data but no physical material?
Yes, yes.
Yeah. Well, okay, so that's something I've thought about a lot. I think it's a very hard problem. There are sort of two separate issues there. One is nomenclatural. One is, the question, can you describe species based on sequence data in the absence of a physical specimen? And that's very controversial. I'm in favor of it because I feel like if you make a discovery, you should be able to formalize it with a name. And putting a name on something draws attention to it. A name is a hypothesis. And so you might be proven wrong, and that's good. That's science. So that's one issue, the nomenclatural part, sequence-based typification, which I favor, but I think that's most mycologists, I think, do not. The other question that's more slightly different is, how do you integrate sequence data with taxonomic data, with or without having names for sequence-based taxa? And I think a good example of how that can work is this Unite platform, this user-friendly Nordic ITS-based sequence grouping platform. And that's, I think, an excellent tool that lets you integrate sequence data with or without physical specimens into a large database of sequences that are based on specimens. So I think that shows promise. The problem there is that it's just one platform, and so if you don't like that one, it's that's what you're stuck with. But yeah, I think that problem of integrating sequences, environmental sequences, with the mainstream taxonomic data is a great challenge of systematics right now.
And many feel that the structure-based taxonomy will get sidelined.
I don't think that will happen. I don't think that will happen. I think we're always going to need taxonomists who really know the organisms, know what they look like, know their natural history. So I don't think that's going to happen. That is a worry. I mean, one of the things I've heard people say is that if you have sequence-based taxonomy, we won't need herbaria anymore. And I don't think that's true. I think if we have sequence-based taxonomy, it will just make taxonomy more interesting, draw more people into the field, including ecologists, including bioinformaticians. So that's my vision for the future of fungal systematics, something very integrative that draws together lots of folks, many of whom who would not consider themselves taxonomists.
Dr. Hibbert, there is a sort of personal question. Can can you guide us what will be the right stage for isolation of DNA in a polypore?
In a polypore,
Yeah.
for getting DNA,
Yeah.
polypores are really tough to work with. I've worked on polypores some, although I'm not an expert in their taxonomy. I've always been fortunate to work with cultures from polypores. That's one of the great things about polypores is they grow so nicely in culture, most of them anyway. For genomics, single spore isolates are what you really want. You want monokaryons. And frequently when you collect polypores, they're not making spores, so that's a challenge. But trying to grind up a trimitic polypore is not easy.
Yeah. Thank you, sir. Thank you very much.
Sure.
So the lecture delivered by Professor Hibbett, it was very informative. He has given a very impressive and very informative talk on the molecular-based techniques of Lentinula. I think his talk would be very informative and useful for the new researchers who are working in the area of mycological taxonomy. Thank you, Dr. Hibbett, for sparing your valuable time and for delivering talk in this national project. Thank you, so nice of you.
Well, thank you very much. And thank you all for your questions. And it was really a great pleasure to be part of this. So thank you.
We are extremely thankful, Dr. Hibbett, and that too in the odd hours you could be with us, and it's really an honor for this.
Yeah, it's a wonderful talk. Thank you.
Thank you very much. Thank you.
Now we have second speaker, Dr. Joseph Vageras. He is National Academy of Sciences of India senior scientist. Presently, he is having a position of chairman of CNR VDCD Bangalore. Apart from this position, he has so many other positions like NASI senior scientist in the year 2011, INSA honorary scientist in 2009 to 10, INSA senior scientist, University of Agriculture Sciences Bangalore, ICAR Emeritus Scientist, Bangalore, Head Department of Agriculture Microbiology, UASF, Bangalore, Professor Department of Agriculture Microbiology, UAS, Bangalore, Visiting Scientist, Rockura Research Station, Hamilton, New Zealand, Assistant Professor, Department of Agriculture Microbiology, USA, Bangalore. So his area of research is agriculture microbiology, mycorrhizal symbiosis, biofertilizers, biological control. Dr. Joseph has so many awards and fellowships in his grades like Mycological Society of India Shom Memorial Award, Association of Microbiological Society of India SR Vyas Memorial Award, University of Agriculture Sciences Bangalore Distinguished Alumni Award, Society of Soil Biology and Ecology Distinguished Science Award, University of Agriculture Sciences Dharwad Lifetime Achievement Award. He is fellow of so many societies like Indian Phytopathological Society, fellow of National Academy of Sciences India, fellow of National Academy of Agriculture Sciences, fellow of Indian National Science Academy, Association of Microbiologists of India, fellow of Mycological Society of India, fellow of National Academy of Biological Sciences, fellow of Society of Agricultural Biotechnologists. So I welcome you, Professor Joseph. Please present your talk. Dr. Joseph.
Yeah. Good morning. Thank you very much for the kind words of introduction. And so the topic I am going to cover today is, can agricultural practices influence fungal population and diversity? Okay, we heard about a couple of lectures already about the mycorrhizal fungi and how they improve plant growth. It's very well known that mycorrhizal fungi help plant growth and nutrition and productivity. So I just included a couple of slides here just to show how they improve plant growth, inoculated versus uninoculated in different crop plants and tree species. So this is just to summing up, these mycorrhizal fungi help in the nutrient uptake, especially the ones which are less mobile in soil solution like phosphorus, and having synergistic interaction with other beneficial soil organisms, protecting the plants against soil-borne pathogens, helping in water uptake under drought situation, and producing growth-promoting substances. All these influence the growth of plants in a better way by mycorrhizal fungi. But today, what I'm going to talk is how the mycorrhizal fungi.
Avneet, you please mute.
How mycorrhizal fungi are influenced by the various agricultural practices. Okay, so like crop protection, cropping, crop rotation, tillage, fertilizer application, organic amendment application, and so on. It just go one by one and see how the various agricultural practices followed by the farmers affect the mycorrhizal population as well as the diversity. And mixed cropping is a very common practice in compared to the monocropping also. So farmers take monocropping, some mixed cropping. So in this particular experiment, what we did, we had a monocrop of maize, monocrop of soybean, then a mixed crop of maize and soybean. And look into the infective propagule numbers, which gives the total number of propagules. But when we take the infective propagule numbers, in the mixed cropping of maize and soybean, we have the maximum number of infective propagules compared to monocropping of maize or soybean. So that shows that the root intensity in mixed cropping is much closer, which gives more room for the mycorrhizal fungi to colonize, and also takes away more nutrients from soil. And we know that fungi work better in a less fertile soil. So that is the reason why mixed cropping or intercropping is much better compared to a monocrop. And then we did an experiment on crop rotation. Here we have taken in the first season, we know there are 3 seasons. In the first season, we have taken finger millet, that is ragi. And in the the third season, we took cowpea in all the treatments. We changed the crop in the second season alone. In the second season, we took cowpea in the first treatment, which is a mycorrhizal host. In the second treatment, we took mustard, which is a non-mycorrhizal host. And the third treatment, we left the land fallow without any crop. And you can see the infective propagule numbers here. It was maximum when a mycorrhizal host is grown in all the 3 seasons. The infective propagule number comes down when we grow a non-mycorrhizal plant in the second season. And when the land is left fallow in the second season, again, the number of infective propagules come down. And this is the influence of alley cropping on fungi. Alley cropping is nothing but growing a tree crop in between the food crop. So this is a very common practice followed in Africa. It is coming into picture in India in many places. So in this experiment, what we did, we took 2 tree species, Lucina leucocephala, Gliricidia cipium, both are green manure plants. And we took 3 different field crops grown in between the tree species, finger millet, peanut, and pigeon pea. And we also looked into the propagule numbers. And here we can see here when we compare the 3 crops, finger millet supported the maximum number of infective propagules compared to pigeon pea or peanut. Coming to the 2 tree species which we used, and Lucina supported much compared to Gliricidia. And the tree species were planted at different spacing. That means we had 4 rows of the crop plant and then the tree species, 8 rows of crop plant and the tree species. Again, we could see maximum infective propagules in the crop plant was seen when the trees were taken at a distance of 12 meters. And another thing, a project we had funded by the United Nations Environmental Program, where we studied the mycorrhizal propagules in coffee ecosystem. So coffee is always grown under shade trees. And there are 2 types of shade trees, evergreen shade trees and deciduous shade trees. Some farmers take only 1 type of shade tree. Some farmers take 2 or 3 shade trees. Some farmers take many shade trees. And so we have studied the infective propagule numbers under the coffee root system. What are the results? And there are 2 types of coffee commonly grown. One is Arabica coffee, other one is Robusta coffee. The results are seen here. The coffee under the evergreen ecosystem had higher infective propagules. Of the 2 coffee types, Arabica supported higher infective propagules of AMP fungi. And of the 3 shade typologies, coffee grown under 2 shade trees had the maximum number of infective propagules. Then we compared the various tillage practices. We know the conventional tillage where the bullock are drawn and then we plow the land, and now versus now tractor is used. What we found was the deep tillage always disturbs the mycelium of the mycorrhizal fungus, whereas the conventional tillage has less effect on the mycorrhizal fungus. And the same thing is reported here also. When we take a rice crop, the conventional tillage has a positive effect on AMP fungi, and the deep tillage has a negative effect. And again, farmers put a mulching in between the rows of crops. When we take, this is a wheat crop, and when mulching was done, it had a positive effect on AMP fungi. And sometimes the residue is burnt. That is very commonly done. And the residues, when it is burnt, then it has a negative effect on AMP fungi. Then we tried the various organic amendments, and we took 3 different organic amendments: paddy straw, maize straw, and Pangamia leaf. And you can see the infective propagule numbers. The Pangamia leaf supported the maximum number of propagules, probably because it is a legume compared to the 2 others, which are cereals. And again, we tried 3 different green manure plants. And this was done in a coffee plantation where the green manure plant is incorporated amidst the coffee plants. So on the 3 green manure plants which we tried, Dolichos, Canavilla, and Crotalaria, the result shows that Crotalaria is taking Crotalaria as a green manure increases the number of infective propagules. And again, we tried the role of vermicompost. When we added vermicompost along with the mycorrhizal fungi, you can see the colonization increases and also the spore number significantly increases, and in turn the plant growth. So adding vermicompost supports the mycorrhizal propagules. And then we tried the chemical fertilizers versus the organic manures like farmyard manure or compost. Here we can see the control, the infective propagule number is 0.65 into 10 to the power of 3. And with the compost or the farmyard manure, you can see it is tremendously increased. And with the fertilizer, chemical fertilizer, it comes down. And again, we know, going through literature, the phosphorus fertilizers, all the fertilizers have a deleterious effect when added in a large quantity on mycorrhizal fungi. And of the phosphatic fertilizer, when we add heavy doses, it decreased AMF. But the slow release phosphatic fertilizers like rock phosphate, bone meal enhanced the mycorrhizal population. And again, heavy doses of nitrogen fertilizer has a deleterious effect. Again, we compared nitrate versus ammoniacal form of nitrogen. Nitrate was found to be more inhibitory compared to the ammoniacal form. Regarding potassium, we could get only one report that is done in Colombia with a level up to 200 kg had increased the AMP from propagules. This is again a simple experiment which we could do, show that deleterious effect of chemical fertilizer. With no phosphatic fertilizer, the population, the infective, the percent colonization was 95%. With 50%, it did not affect very much, whereas with 100%, it came down to 74%. With double the recommended level, it drastically affected the mycorrhizal colonization and spore numbers. And in the university, we had a long-term fertilizer application. That means the same plot, the same fertilizer is applied year after year, year after year. And this we took after 16th year. So here we can see again very clearly, the recommended level of phosphatic fertilizer, the infective propagule number was 540. But when it was reduced to 50%, it significantly increased the infective propagule number. When it was increased to 1.5 times, again it drastically reduced the infective propagule numbers. And soil sterilized before raising the nursery by soil solarization. That is, we spread a polyethylene sheet and sterilize the soil. Again, we tried in this experiment 2 different thickness of polyethylene sheets. And solarization was done for 20 days, 40 days, and 60 days. The result very clearly showed solarization was with 0.1 thickness reduced significantly fungal population compared to 0.05 And again, solarization for 60 days drastically reduced the fungal population compared to 20 or 40 days. Slash and burn agriculture is a common practice followed in Northeast India, where they burn the forest and then raise the crop, and which is also called as zoom cultivation. And so we tried this particular thing in one of the experiments, and the total population, we could isolate from the natural forest 44 species of fungi belonging to 6 genera, and which I have listed here. Whereas the slash and burn cultivation or the zoom cultivation reduce the fungal population and diversity. We could see 10 species were totally eliminated because of this slash and burn cultivation. Again, because repeated burning is there, and naturally, and the certain host species are also lost, these fungi are also lost. And what is the effect of pesticides? And we can say that when we talk of pesticides, it includes fungicides, insecticides, and herbicides. And so we can see most of the pesticides are inhibitory to fungi. And some of them do not have any effect. And it is interesting to see some of them stimulate the proliferation of fungi. I have given some of the examples here. Taking fungicides into consideration, binomil and thiobindazole are inhibitory, whereas captan and rylon have no effect on fungi. Taking insecticides, metacistox and aldrin are inhibitory to fungi, whereas chlorpyrifos and sumicillin have no effect on fungi, whereas DBCP stimulates fungi. Taking into fumigants into consideration, so far all of them are inhibitory to fungi. Coming to herbicides, again we have 3 categories. Glyphosate, ethyl are inhibitory, whereas 2,4-D, alachlor, pendimethalin have no effect, whereas the herbicide anilofloss stimulates fungal population in the soil. And in acidic soils, for remediation, farmers add lime to raise the pH. So there are 3 reports which came from 3 groups of scientists from 3 different countries. It was very interesting to see that one group reported that it reduced fungal population. Another group said it has no effect. The third group said it increased population. So I think probably we may have to do more studies to draw the right conclusion. And we had another project funded by the Global Environmental Facility to study the diversity of fungi in Western Ghats, where different types of land use is made. So different land use types, how do they affect the mycorrhizal population? So the land use types are, we have the natural forest, natural grasslands, acacia plantations, cardamom plantations, coffee plantations, and paddy fields. So we studied the mycorrhizal population and the diversity in these different land use types. We could isolate 282 isolates of fungi, and species diversity and richness was also studied by the Shannon-Wiener index, which is used for comparing the diversity between various habitats. And we also studied the Japanese index, which gives the number of species in a community. The results are given here. And with both the indices, Shannon-Wiener as well as Japanese index, in the natural forest, you can see it was maximum, where it is not disturbed by humankind. It was also very high in grasslands, where it is not disturbed by man. And Where it is slightly disturbed, as in cardamom acacia plantation, it came down. The maximum intervention by the human being is in paddy fields. And you can see both the indices were the least in paddy fields. And again, there are some bacteria which help mycorrhizal fungi. The colonization as well as the function of mycorrhizal fungi, which are referred to as mycorrhiza helper bacteria. And we tried to see where we screened various bacteria and see which are the ones which act as mycorrhiza helper bacteria. And we have tried here various of bacteria, and you can see Pseudomonas fluorescens is the best mycorrhizal helper bacterium. So we can also see that Acetobacter aceti, Excicobacterium acetylicum, Pantioanglomerans, they are also mycorrhiza helper bacteria. And this is one experiment which we did recently. We have one Indo-Swiss collaborative project which is ongoing. And here it is trying what is the effect of intercrop. And under dry land agriculture conditions, I would like to emphasize this, under dry land agriculture conditions. And in this particular experiment, we took a deep-rooted crop like red gram, pigeon pea, and we took a shallow-rooted crop, finger millet, which is ragi. Okay, and here we can see the dry land, the top yellow portion, it is dry. And see the blue portion at the deeper layer, there is moisture. Okay, what we were interested in studying is whether, and these mycorrhizal fungi can connect the root system of crop plants which are growing nearby. So what we were interested in studying was whether from the deep rooted plant, the red gram, whether the mycorrhizal fungus can take water and supply water to the shallow rooted crop. And that is what we studied, which is referred to as bioirrigation, which is a hot topic of the present days. So we could see that it helped the finger millet growth under dry land conditions. We have taken it under the glasshouse conditions, and we have taken it under the field conditions. And we could very clearly show that the deep-rooted plant supports and gives water to the shallow-rooted crop. And this we wanted to prove under lab conditions. This was an experiment that was done in Switzerland group, and where they took a deep-rooted container, the red one, big container, and a small container which is inside here. So in the deep container, they planted pigeon pea, that is red gram. And in the shallow-rooted container, which is a perforated bag, and this perforation of this bag was 10 micrometer. That means it will not allow the roots to pass through, but it will allow only the hyphae of mycorrhizal fungus to pass through. So, and of course, we had the different layers of sand, gravel, etc. And here at the deeper layer, we had deuterium labeled water, where thereby we can look into the hydrogen isotope, monitor the hydrogen isotope. So what we could see, this is the deep-rooted crop. The water is here. If it is taking the isotope, hydrogen isotope, then finger millet, the ragi, the root zone must be labeled with the isotope. That is what was checked. And we found that the root zone of ragi had the isotope, which was monitored, measured by the NMR spectroscopy, which very, very clearly proved that the deep-rooted plant, through the common mycorrhizal network hyphae, transfers water to the shallow-rooted crop. We still have these field experiments at present. So in conclusion, what I would like to say, it is very well established, the AMP fungi improve plant growth. And one of the impediments coming in the way of field applications of AMP fungi is very less information is available on the ecology of these fungi, especially in tropical countries. I would like to underline this. A lot of work has been done on temperate countries. In tropical countries, very little work has been done on ecology. And more information is required on the different agricultural practices and human impact influencing AMP fungi. And There are two strategies for managing mycorrhizal fungi. First one is inoculation with an efficient AM fungus, which most of the mycorrhizal workers do. We try to take the best mycorrhizal fungus, inoculate the crop plant, and see the improved plant growth. But the second approach is how to manage the indigenous mycorrhizal fungi by judicious agricultural practices. So such studies, that is what we have done in this, what I told so far, so that we will help the farming community by doing which agricultural practice helps them best to improve the crop growth and the productivity, which nowadays is given a very nice term, ecological engineering. So that by adjusting the native mycorrhizal fungi and the various agricultural practices which could improve the colonization and productivity of the crop plant in turn. That is the approach which is done, which is looked upon by ecological engineering nowadays. I think more studies on this type of work will be done in future in our country so that we improve crop growth and productivity in future. Thank you very much.
Yes, topic is open for discussion. Any queries from the delegates?
I have no query, but I would just like to add, good morning, all of you.
Good morning. Good morning.
Raj, it is a pleasure to hear you. Every time we hear, our knowledge get enriched on mycorrhiza. It's very easy to get an opportunity to hear you. Thank you so much.
Thank you, madam. Yeah.
Dr. Singh, I have one query from sir.
Yeah.
Sir, your lecture was very much inspiring and very much informative, but I have one query. I have seen one slide where you have mentioned that some of the pesticides, they are inhibitory to AMF, some have no effect and some are stimulating. So is there any possible reason for that? Or we can say a specific strain of AMF is specific for a particular pesticide, please?
Maybe that depends on the chemical composition of the pesticide which we use. And we are not, people are
because
not.
I am also working on this line, but using different systems. That is, I have a curious about this, what is happening in this system.
Yeah, it must be mainly it must be based on the chemical composition of the pesticide.
Yeah, please.
Thank you for the nice lecture, sir.
Oh, thank you, Dr. Raman.
Yes.
Is there any other questions?
Yeah. Good morning, sir. Suresh.
Nice to see you, Dr. Suresh.
How are you, sir?
Old student of mine.
Yeah, I'm very pleased to hear you after so many years, but it is a great occasion for me as I learned many things from you on those days, but as I keep seeing you and then mycorrhiza and maize where I'm working with.
Okay.
Just to know the mycorrhizas and you said it's more good with ragi. Is there any evidence or suggestion for maize? Because maize is one of the major food crops in Africa. So where we can enrich or support farmers for mycorrhiza, Pseudomonas fluorescens to enhance the crop productivity.
Yeah, it's possible. When we have done it with ragi, I think it should be possible with maize also. Any fibrous, shallow root crop, it should be possible. Yeah, it should be possible. Okay. Yeah.
And there are reports on this, sir. There are reports on this maize.
Yeah.
Good morning, sir.
Good morning.
Yeah, Professor Rodriguez. So a lot of new information on this on mycorrhiza. Thank you very much.
Thank you. It's a pleasure. Yeah.
Sir, good morning. NS Satri here.
Oh, good morning, Professor. So nice.
Nice to see you and nice to listen to your.
Thank you.
Good morning, sir.
Very good morning, Professor Avneet.
Thank you very much, sir.
Lovely lecture, Bhagyaran.
Oh, thank you, Dr.
Thank you. If there are no more questions, I would like to invite the third lead lecture, Professor Gurpal Singh Dhingra. it is my pleasure and honor to introduce Dr. Gurupal Singh Dhingra. He retired as a professor from the Department of Botany, Punjab University, Patiala. Professor Dhingra did his MSc as well as PhD, both from Punjab University, Chandigarh, in 1977 and 1984 respectively. In 1978 to 79, he did his postdoctoral work with Professor Petmas II in Estonia, and he joined the Department of Botany, Patiala, Punjab University, Patiala in 1987. He became a professor in 2006 and superannuated in 2014. He had 95 publications to his credit, and he contributed 7 new genera and 19 new species and 30 new varieties of corticoid and poroid fungi. He guided about 17 PhD students, and he had 2 sponsored projects to his credit. And he widely traveled to Sweden and New Zealand, Germany, and he is a life member of Mycological Society of India, Indian Botanical Society, and Indian Science Congress. And he has been awarded by our own society's award, MSI's award, Dr. K. Natarajan Memorial Award. And then he had a few international collaborations. Today he would be speaking on non-poroid and poroid Agricomycetes taxa, new to science. Professor Dhingra.
Thank you very much for the nice introduction and saying lovely words for me. I'll share my screen now. Can you see?
Yes, yes.
Sir, it's visible.
Good morning, everybody, and it's my pleasure to participate in this conference with the blessings of the Almighty, my parents, and Professor K.S. Thind, my PhD supervisor, and with the collaboration and guidance of Dr. Nils, Dr. Biden, late Professor Parmasto, Dr. Langer, Professor J. Erickson, Peter Buchanan, Sean Pennycook. My research team, Dr. Avneet Pal Singh, Nishi Singla, Sanjeev, Gurpreet, Suricha, Shruti, Priyanka, Jaspreet. My family, Meena, my wife, my daughter Dipinder, my son, and my grandchildren, their patience and cooperation, I have been able to compile this preparation on the poroid and non-poroid new species, our contribution. We started this work in 1978 in the Eastern Himalayas and adjoining hills from 78 to 1984. And then after joining Punjab University Patiala, I shifted it to the Northwestern Himalayas and Punjab. We have basically followed the classifications proposed by Blackwell, James, Hibbett, and accepted by the Dictionary of Fungi. And also we have modified our species as per the online repositories, Mycobank and Index Fungorum. The journey started with the first trip to Darjeeling in 1978 in West Bengal. And it was followed by visits to Sikkim, Tripura, Arunachal Pradesh, Assam, Mizoram, Manipur, Nagaland, and the Royal Kingdom of Bhutan in the eastern side, and Himachal Pradesh, Uttarakhand, Jammu and Kashmir, Punjab, in the northwestern Himalaya. These are some of the photographs with my research scholars, and we were lucky to have Niels along with us while we were visiting the northwestern Himalayas. The forest from where we have collected all this material is basically the pure deodar forests, mixed forest in Mussoorie, mixed forest in Donalty, pinus in Chail, then Barot, Jitingri, pure forest, Cupressus torulosa in Ramban, Abies spindro in Kalatop, young oak forest in Solan, Icea smithiana in Hatupik, and mixed forest in Kufri, Workus forest in near Solan, and then in Narkanda it's a mixed forest, and Pinus valiciana in the upper reaches of Kufri, and mixed forest in Kandiali. The basic systematic study is based on two basic things: macro characters having the details about the basidiocarp, and micro characters, which I'll show you, and the reactions of the various structures with cotton blue, Melzer reagent, and sulfovanillin. These are the basic techniques which we have followed, the basic stains which we have used, and the reactions, and finally documentation is done. And the characters which we generally see is the macromorphology, the resupinate, reflexed, pileate, subpileate, and the branched pileate basidiocarps. The various types of surfaces, hypoconite, smooth, watted. Then we have got poroid, we have got spiny, and different types of pores. You can see the different colors of the hymeneal surfaces. Then we have got different bright colors of all these surfaces. The micro characters include the different types of hyphae, which constitute generative hyphae with or without clamps, skeletal hyphae, and binding hyphae, constituting monometic, dimetic, and trimetic hyphal systems. Various types of basidia, spores, cystidial elements, sterile structures of different types, and then the constitution of all these microstructures in the basidiocarp, whether it is loosely constituted, it is compact, or it is very compact and it is having multi-layers of the hymenium. Review of literature, comparison of all the characters with published literature, comparison with the types we have done at the herbarium at Punjab University, Kew, then Gothenburg, Oslo, and Landcare, and also taken expert opinions, and recently also started the molecular work. We have so far published 7 genera new to science, which include Confortisium, Cordochete, Dendroflavia, Hallenbergia, Redulomycetopsis, Repetobasidiopsis, Primatiala, and majority of these are from the temperate areas and one from the tropical areas. We have published 90 species. You will find the ones in blue color, 2 species each. In yellow color, we have got 1 species each in genus. These are belonging to 56 genera. These are all 1 species each in yellow. Then maximum number of species which we have published is of Hypoderma, which is in green, and Phlebia, 7 species we have published which are in green, and Phlabiopsis we have published 4 species, Phlinus we have published 4 species in pink, and Tomentella and Veraria 3 species each, which are highlighted in red. And these are the 13 varieties, and maximum varieties again we have published of Hypoderma, followed by Botryoidobasidium. Coming to the genera new to science, the genus Cordochete, which we have published, it is very peculiar and differs from Phlabiopsis and Rhizocete. From Phlabiopsis, it differs in having these hyphal cordons, which are extending into the hymenium and with thick-walled encrusted cystidia, and from Rhizocete in lacking the reaction of the hymenium with KOH. Dendrophlebia is, we collected this from the cryptomeria in Darjeeling area. It's very peculiar. It resembles phlebia in its texture, but it differs in having these dendroid hyphae. Hallenbergia, very brightly colored. It is a unique fungus with having some pseudoparenchymatous type of tissue and very thick and gelatinized basal zone. It had some resemblance with Clavulium and with Hypokinesium in having the similar type of spores, but the constitution of the hymenium is very different. The next genus is Radulomycetopsis, which basically resembles with the Radulomyces, but it was different from Radulomyces in lacking clamp connections on the hyphae and presence of the cystidial elements. The fifth genus, that is the Repetobasidiopsis, basically you find it is close to Repetobasidialum in having the repetitive basidia. But basic thing is, in this case, the hyphae are without clamps and presence of these cystidial elements. Genus Trimetiala is unique to corticoid group of fungi because this is one of the rare ones which has trimetic hyphal system. You can see all the three types of hyphae. It was quite similar to Illyrodiscus in the bigger size of basidiospores and basidia, Lorelia in having trimetic hyphal system, Laticoticium in having the similar type of basidiocarps, but it was unique and it was named Indica from its locality in India. If we look at the new species, Illyrodiscus, this was very unique because there was thickening hymenium in this, and the color of this was brownish, which is quite rare for this genus, because normally the color of the basidiocarps in Illyrodiscus is pinkish, and the size of the basidiospores and basidia is very large. This we collected from Kufri. And if we see this Pisocoticium, this species has been collected from Uttarakhand, and it is very peculiar and it differs from Lutescens in having the clamped generative hyphae. If we look at Candela brocade, it resembles Africana, but it differs from Africana in the reaction of the hymeneal surface, as well as with QH, as well as larger basidia and wider basidiospores. Clavulium hallenbergii is quite close to Clavulium mercurii, but it differs from it in having smaller basidiospores and the lack of Gloiocystidia. Serratiomyces bizonatus, you can see there are two zones. One zone is encrusted and one is of the smoother hyphae. So it was named bizonatus on the basis of these two zones. And Conohypha grandispora is basically differentiated from the other species in having very large sized basidiospores. If we look at this Corticium masuriensis, you can see that this is different from the other closely related species. One species is that Albidocremium in having very broadly ellipsoid spores and larger basidia. Cristinia tubulocystidia, as the name indicates, has got tubular, very large cystidial elements. Cylindrobasidium, when we collected this fungus, we thought that maybe it is the jelly fungi, but when we worked it out, it turned out to be a new species of Cylindrobasidium with very, very long basidia and large-sized basidiospores and hyphae without clamp connections. Datronia, a poroid genus. This species we collected from Narcanda area, and it differs from Scutellata in having thicker basidiocarps and larger sized of the pores and long, narrowly clavate basidia. Phyllomyces cystoides, it has been collected from the Eastern Himalayas, and the basic difference is that there are present encrusted hyphae in this. Flavoflavia spherocospora, as the name indicates, has spherical spores, which is a different character from rest all the species of the genus. Tremates indicus, it differs from Tremates gibosa, the common species in the Himalayas, in having larger basidiospores and watted hymeneal surface. Hymenocete longis sterigmata, as the name indicates, has very, very long sterigmata on the basis of which this species has been named. And in addition, it has got all these moniliform cystidial elements. Hyphoderma, as I told you, we have got maximum taxa for this, 11, 8 species and 3 varieties. And you will find these are different species by cystidium having 2 types of enclosed cystidial elements. Then these all species, you can see the very, very long cystidial elements and the cracked hymeneal surface. Then densest textus, very dense texture. Earlier we thought maybe it is a species of Libya based on the dense texture, but then the basidia and basidiospores were typical of Hyphoderma. Then this Hyphoderma, we got very little material, but it is very unique in having almost subglobose to globose basidiospores. Hallenbergii, it is very peculiar in having patches of the encrustation on the cystidial elements. Then this species, the hymeneal surface is basically having spines, and this is the only species we could find without any cystidial elements. Then this one having 2 types of cystidial elements, that thick-walled encrusted throughout its length cystidial elements and the thin-walled cystidial elements. And in this case, the spores and the basidia, they were with lot of oily contents. Hyphoderma satigerum, variety Y cystidium, basically having 2 types of cystidia. And if we look at the next species, which I collected from West Bengal, it's a very thin layer of the basidiocarp, which we can see on the surface of the quercus branches. Then this Hyphoderma was very unique in having the 2 types of cystidial elements. One are the subglobose type of, which are enclosed, and other ones are the projecting cystidia, which are almost clavate. Then Hyphodontia coli cystidiata, basically it has been named because of the very long stalk of the cystidial elements. And this species has got very, very large sized of the odontoi, which is almost heteronoid, and it has been named after that. Then this species of Hypocnesium is quite unique in having very, very long cystidial elements along with the punctate basidiospores. Laticoticium usuriensis, basically it was differentiated from Laticoticium tenellum on the basis of broadly ellipsoid to ovate basidiospores. Leucogarafana is basically very close to most of these coniferae, but this was unique in having thick-walled, very small-sized cyanophilous basidiospores. Then this Leptosporomyces, the color was very unique, and there were no cystidial elements in this new species. Polycoticium indicum, very loosely interwoven basidiocarp with cystidial elements having very fine type of oily contents. Then Piniophora Now, this species was selected as the back page for Mycotaxon, and it differs from Boydinia in having basically broadly ellipsoid basidiospores. Earlier, otherwise, in Piniophora, we have got narrower spore than this. In Flabia, we have got 7 species, the next genus, and then Crassus ubiculata. The subiculum area you can see is very, very prominent and very thick, and this has made us to name this species as Crassus ubiculata. And this species of Flabia has been named based on the totally gelatinized basal zone at the base of the basidial elements and these highly convoluted generative hyphae. This species of Flabia, you can see the sterile elements are very long and they are encrusted, and we can see the small spines and broadly ellipsoid basidiospores. Flabia carmangii, the collection is done from Bombdilla and Carmang. The hymenium is highly meruloid in this case, and it has got enclosed cystidial elements and broadly ellipsoid basidiospores with oily contents, which is quite rare in Flabia. Then Flabia singularis, very large-sized cystidial elements in this species. Flabiopsis, we have 4 species which we have described, 2 from Eastern Himalayas, 1 from Missouri, and 1 from Punjab. Now, this species differs from the rest of the species in having narrower cystidial elements, and Himalayensis has got broader and smaller basidiospores. This Missouriensis again has got this broader, and the shape of the basidiospores is different, whereas in Punjabensis also the shape of the basidiospores as well as size is quite different from the rest of the species of the genus. Redilodon, we have described 2 species. One, this was collected from a park in Chandigarh, very prominent spines in this case, whereas the other one has got less prominent spines. And you can see the variation in the cystidial elements. These are just hyphoid and these are irregularly shaped. Cystotrema angustispora basically is named after the shape of the spores, and this macrosporus is again on the base of the bigger size of the basidiospores. Cystotomestrum roseucremium has been named on the basis of these rosaceous patches on the basidiocarp. Then Scytinostroma, these 2 species, they have been published in a paper by Boyden, in which these collections, they were included, and they are different on the basis of the shape of the spores. And this species, if we look at the tubulicarinus, the cystidial elements and the spores, they are quite unique for this species, on the basis of which it has been named. Sterium peculiar was the first publication which I did with Boyden, Parmasto, and this was collected from Manipur, and it has got some peculiar markings in the sectional view. These appeared like this, and it was named peculiar. For Tomentella, we have almost 4 species which we have named on the basis of the color of the basidiocarps and the shape and size of the basidiospores and the nature of the basidia, whether they are thin-walled or thick-walled. This is named on the basis of the color of the basidiocarp. This is on the basis of the thickness of the basidial wall and the locality, and unicusa is, it's unique in the combination of all the characters. Then as far as this tubulicium species is concerned, it is different on the basis of the type of the spores. Vararia Himalayana, we have got different species, and all these species have been differentiated on the basis of the type of spores, the branching pattern of the dicohyphidia and the cystidial elements, you will see, and there is also variation in the color of the basidiocarps. Vararia indica, spores are different, cystidial elements different, longicystidiata, very long cystidial elements, and the color is also different. Then Himalayana, then Xylodon, we have two species which we have collected from the eastern Himalayas, that is subglobosa, based on the shape of the basidiospores, and Mussuriensis. You can see the spores, they are quite different in this species in comparison to that. In poroid species, this was again collected from Chandigarh and Rhodiala. It was quite different in having small-sized cystidial elements in addition to the different shape of the basidiospores. For mastomyces, you see this was again looking like a gelatinous fungus, but when we worked out, it turned out to be a poroid genus and different species. Another species, this is Postia chopaliana, and there is another one that is, these are basically differentiated on the basis of different types of cystidial elements, branching pattern of the hyphae. Now we have got few species of Phylinus, and you will see the Phylinus species are differentiated on the basis of type of CT, cetal hyphae, and basidiospores. This one, you can see thick-walled basidiospores. It differs from, again, thick-walled, but the shape is different. This is thin-walled basidiospores, smaller sized. This is, again, very thin-walled basidiospores. And Oriporia, the cystidial elements were very unique in having these tips which are thinning and somewhat sinuous. Then Fomitiporia, it has been taken out from Phylinus, and it differentiates basically from Rosemarini in having smaller size of the pores. The Fulmiformis pileatus are also taken out from Phylinus. It differs from Johnsonianus in having pileate basidiocarps. Then Periniporia, having it is different from other species of the genus in having the subfusiform type of the basidiospores. So in all of the 7 genera, 6 are from the temperate areas and 1 from the tropical. So concluding that out of all our 103 new taxa, 69 species and 13 varieties are from temperate zone, 6 species from the subtropical zone, and 15 species from the tropical zone. So distribution of the 27 taxa, 25 species and 2 varieties, if we look at the eastern side, 19 are from the Indian continent and 8 are from the Royal Kingdom of Bhutan. So out of these 19, maximum are from the West Bengal, followed by Arunachal Pradesh, and 1 each from Sikkim and Manipur. And state-wise distribution of the species from the northwestern Himalayas, maximum we have collected 45 from Himachal Pradesh, followed by 17 from Uttarakhand, 7 from Jammu and Kashmir, and 6 from Punjab. These 2 diagrams were selected by the editor as back page of Mycotaxon, and because of our lovely diagrams. In the end, I would like to thank the organizing committee, Department of Botany, Punjab University Patiala, UGC, DST, international scientists, my research team, family, friends, and the online audience for patiently listening to my talk. Thank you very much.
Thank you very much, Professor Dhingra. We can take maybe 1 or 2 questions quickly if anyone has. If there are no questions, let us move to the next lead lecture. The next lead lecture is by Professor L.M. Suresh. He is a maize pathologist for sub-Saharan Africa, and also the head of maize leaf necrosis screening facility at CIMMYT. He works at Nairobi, Kenya. He leads the CIMMYT's maize pathology efforts in sub-Saharan Africa. and he regularly contributes to global maize program projects that have strategic significance in maize pathology. Suresh also works on maize lethal necrosis phenotyping with public and private partnership at CIMMIT and at Kenya Agricultural and Livestock Research Organization's Joint Research Station at Naivasha, Kenya. His team has phenotyped around 2 lakh maize germplasm from various partners, and 19 MLN resistant tolerant hybrids have been released in East Africa so far. He has supported the training of more than 500 researchers, and he has about 50 publications and one book. So may I now request Professor L.M. Suresh to start his talk on maize lethal necrosis. Professor Suresh.
Yeah, thank you, Dr. Krishnamohan. Thank you for organizing committee for giving me opportunity to share the experience. I'll shift the gear from fungus bit to virus. As you all know that the food crops in Africa is one of the key element to focus on the research. I have been working in CIMMIT last 6 years. Before I was working in private sector in India for almost 20 years. So today I'm going to share the learnings from maize lethal necrosis in a similar way that they say corona of maize, what we are experiencing now. More or less all the arrangements or in terms of disease management is same. But today I'll be sharing key aspects of maize lethal necrosis disease, and next the global occurrence and distribution of maize lethal necrosis or MCMV and disease resistance breeding program, and mainly focusing on the next is a safe seed movement with effective quarantine measures, MLN disease diagnosis, and how we focus on the MLN pathogen-free commercial seed productions and capacity building. And we have some information on the MLN web portal and community of practice. Africa is mainly focusing on maize, and it is life for them. In maize, there are many fungal diseases. These are all very key diseases of maize, without which farmers cannot grow maize in any part of Africa. So we are focusing mainly 5 diseases of fungal diseases, and Touchsicum leaf blight, grave leaf spot, Fusarium ear rot, common rust, downy mildew, which I have not listed here, and 2 major viral diseases, that is maize lethal necrosis and maize streak virus. So all the maize breeding community, both public and private, has to focus on these key diseases, without which they cannot bring the varieties to the public domain. And of course, this is the same one. And coming to maize lethal necrosis is a viral disease, which is a combination of maize chlorotic mottle virus and potty virus, that is in case of this sugarcane mosaic virus. Sugarcane mosaic virus is globally occurring, and which is there in Africa too. But whereas in maize, the chlorotic mottle virus has reported lately. So that is causing a huge havoc in a kind of Eastern Africa, especially Kenya, Uganda, Tanzania, Rwanda, DR Congo, and Ethiopia. A study has been conducted by CIMMIT to understand in how the disease loss, crop loss, and in 2016 the study was conducted. In Kenya alone, the crop loss was almost tuned to the $180 million. And a recent one more article has been published in Plant Disease. Again, the disease has been declined so fast and so contained in the Eastern Africa, and within the Eastern Africa also, we could able to contain the disease with several multi-pronged approach. As I mentioned, maize chlorotic mottle virus and the potty virus is in this case is sugarcane mosaic virus. The deadly combination is the maize lethal necrosis. Both the virus can cause diseases by itself, but the individual virus infection is not lethal to the maize. And the symptoms are, you can see here, especially the disease will start with the chlorotic specks, streaks, mosaic, and stripes, and then necrosis, and very few set of ears, and that is how the farmers recognize. And it is not the old, it is not new diseases. The disease has started somewhere in 1973, and further it is reported in USA. All the way it has gone to like Thailand, Brazil, Mexico, Hawaii, and China. And of late, it started in Kenya 2011, and it has spread almost all the Eastern African countries. And later it has been observed in Ecuador and Spain. And further, we could not notice any parts of the world. This is how the distribution is. Why this disease is lethal in Eastern Africa? Maize chlorotic mottle virus is new to the region. Otherwise, it was not a problem. Even in India, this virus is not reported. Otherwise, there will be a huge havoc in India as well. Potentially new strains of SCMV or MDMV is another party viruses. And Africa is more conducive for not only for pathogens, but also for growing crops that continuously help in building the inoculum. And this particular virus, MCMV, is seed contaminations. That's why it is more dangerous. And a lot of susceptible germplasm is grown in the region. So as a CIMMYT, when we started this study, we started with a multi-pronged approach, engaging private and public partners, various universities across the globe. And in case of diagnostic epidemiology, we had a separate project, and where we could simplify the protocol of our diagnostics using immunostrips. We could deploy on 8 countries for understanding the diseases. We have a detailed surveillance mechanism, and we focus on MLN free seed productions. We train more than 8,000 team members in Africa, including NPPOs, seed companies, and several other international organizations. We had a rigorous monitoring and surveillance mechanisms through NPPOs, and we have agronomic management by which we could train the people how to manage the diseases, and we have been able to improve the capacity building and communication and outreach. More so, among all this one, the key element is breeding for disease resistance. That's where our emphasis as a CIMMYT strength. So you can see when we started, there was all the farmers suffering like this, that there was absolutely no crop. And with all our efforts, we could able to identify and develop and deploy the disease-resistant hybrids and make farmers smiling on their face. And you can see the just numbers. We could every year screen more than total together 2 lakh germplasms. It is huge in terms of maize, and around 3 lakh rows. So mainly the focus is coming from the CIMMYT, NARS, and private seed companies. You can name any companies that are associated with CIMMYT in Africa. They are working on these diseases. It is not just screening. The quality of data is very high. Heritability is up to 0.99. We turn over almost every 5 months, we give the data to the team. And you can see the quarantine facility. This is the very significance. There are no other maize growing areas around these quarantine facilities. There is behind, there is a lake. But you can see the kind of screening which we have been doing. It is not just that screening. We have been able to convert almost 50 elite lines into resistance from the susceptible background. So this could happen only with a very keen focus on phytosanitations, and then on the management practices and various other safety procedures. So we could have the clear-cut 1 to 9 scale for understanding the maize germplasms, 1 being the resistant, 9 being susceptible. And most of the germplasms are hybrids, which is grown in Africa, falling under 7, 8, 9 categories. You can imagine there is no hybrids which can be 1, 2, 3 categories. And another interesting that the CML 442, CML 443 are these lines, which has been widely used last 5 decades in Africa. But they are used in various agroecological conditions, but they are very much susceptible to maize lethal necrosis. We'll hear an interesting story on these lines very soon. And also when we working on this area, we continuously improved our efficiency and accuracy of phenotyping from various years. So now it is 0.99 heritabilities with a very clear-cut distinction of resistance and susceptible in the phenotyping. And not only this, when we screen our germplasms, we also take these germplasm into various parts of Africa in the hotspot areas. And when we grow in Tanzania, it is completely susceptible. You can see in Naivasha, it's a very good resistance, although it is also subject to drought conditions, but in Naivasha, it is fully irrigated conditions. And we also take into Ethiopia, the yield is quite good, and under optimal conditions, the yield is quite good. You can see here. And you can see the loss of yield due to MLN disease, and in the commercial hybrid is almost like the 97% loss. But whereas in case of the next generation hybrids, you can see here, we got almost 77%. Yeah, and then we released 19 hybrids so far to all the partners to give these hybrids to farmers, which is completely given free from CIMMYT angle. And you can see we have identified the resistant hybrids. This is the second generation. This is the first generation. These are all the commercial hybrid checks, what is grown currently in Africa. This particular, on the left-hand side, you can see named Bazooka, which is grown widely in Eastern Africa, especially in Uganda. More than 3,000 metric tons seed has been produced and distributed from the private companies. And another success story is coming from Kenya, and also is one of the key success. So not just how we could able to bring this resistance. Originally, it is coming from KS23, KZ Start University from Thailand. We could incorporate resistance to our elite germplasms. And you can see, as I told you earlier, whatever it was susceptible, due to with the help of marker-assisted backcross program using markers, we completely converted this line into a resistant one. And you can see here, this is completely dead before. Now with this technology, we could able to convert it into completely resistant. This new version, except this susceptibility, now it is resistant. It is now available to the larger group of people. So this is not just that. Most importantly, when we move the seeds from Eastern Africa to Southern Africa, we have a very 4 levels of quarantines. And once we check our seeds in Kenya and also in other parts of like our regulatory labs, we take these entries to the Mazoe in Zimbabwe. There we again quarantine and multiply and distribute the seeds if it is found free to entire world. You can see now the comparison between the Harare Zimbabwe facility and Naivasha. And there we have stringent phytosanitary measures and to multiply the seeds and give it to the farmers. So we every year check, and last 2020, it is more rigorous work than before. We checked around 14,000 entries. And not just this, we have focused on several other projects, especially immunostrip. I could optimize this diagnostic protocol, very simple, cost-effective, fast. And this we have deployed in 8 countries for 4 years. We could bring more than 15,000 data points. Now this entire information is on the web portal dynamic. So the immunostrips could able to diagnose whether it is having MCM and SCM. Even in India, they can follow if any suspicion can come. So this is just surveillance data points over 4 years in different 8 countries. And now you can see here, what I can say, the success of this, the flagship of this entire study, we kept the football of Africa, entirely Zambia, Malawi, Zimbabwe, completely clean of these diseases. Whereas in case of Eastern Africa, still the disease is persisting. You can see here how every year we are making progress in identifying the diseases. And you can see here we moving on every part of Africa and focusing now this situation we could able to understand. Due to the fund situation, we are not able to do it 2020, but the partners themselves are doing. But we are struggling. We are getting the fund from different donors. We are going to continue. We don't leave this studies in the year. And this is a very simple mobile kit. So all the 8 countries have deployed very effectively. And this is one. Next is most importantly, since it is a maize is this MLN is seed contamination disease. So we focus on producing MLN-free seed production in the different areas. We trained all the stakeholders and producing the checklist so they could follow and adapt. All the private seed companies in the entire 8 countries, especially in Eastern Africa, they focused. Now they are able to bring the infection level from where it was almost 99% to almost 23%, which is an immense effort done by CIMIT. And not just that, we are able to train several private companies for engaging them in understanding the diseases and also scouting, surveillance, and disease management practices. This is another key area we could help the farmers and partners. And this is the key element we could do the intensive efforts on being made to prevent the spread, and especially in MCMV through contaminated seed from the endemic to non-endemic areas. Several seed companies now implementing voluntary MCMV control programs and SOPs to minimize the risk of seed transmissions. Both public and private key staff are trained to tackle this menace. So I was involved in training so many partners in the entire Eastern and Southern Africa, which could give immense support for them. I can say confidently that now Africa, I mean, this disease they can easily tackle, or they are prepared to tackle many other such deadly diseases of maize. They have a very established, very effective network, and this network can give very much confidence to the researchers to make sure that the new varieties can easily deployed and distributed to the farmers. So this kind of mechanism is very much required even in India to handle the diseases hand in hand, both resistance mechanisms and disease surveillance mechanism, and multiple approaches with public and private partnership is the key to manage any such endemic or pandemic in the agricultures. So I can say it is not just limited to seed companies. I could say I was involved in training more than 10 to 12,000 farmers, which they are in various parts of Eastern Africa. So this is the flagship of these entire projects. And we also engaged all the partners through community of practices, through which we increased the MLN knowledge and sharing the knowledge among the shareholders. We could also help them to harmonize the surveillance diagnostic protocol and adoption of rapid diagnostic using immunostrips by seed companies and NARS. And also we sensitize policy holders, which is quite important to strengthen the borders. They're also been available. All this information up to date has been uploaded in the MLN web portal. If anybody is interested, they can use mln.cimit.org. And not just that, like we are also working on the collaboration with the gene editing project with Carteva seed companies, which is globally giant in the agricultures. So we could very clearly distinguish the products, and now the FAK has been done. You can see the gene edited conversion product has been completely identified, developed, and now we had to take a last trial in Naivasha. And this is the first instance of maize in the entire globe for this type of diseases. So in summary, I can say MLN, maize lethal necrosis disease in Eastern Africa is under control with constant surveillance and better quarantine measures, host-free period, producing MLN disease-free seeds. Southern Africa is maintained free of maize lethal necrosis with safe exchange of MLN seed-free seeds, and the emergency preparedness is ready in case if there are any situation comes in. Focus on MLN is breeding various is been in progress. Strong private and public partnership producing MLN-free seeds is in good deployment. And all our efforts has been published in this manuscript with multiple stakeholders. Anybody who is interested, they can get this link. It is a wonderful paper based on my presentation in South Korea during last year. On the, with this, I sincerely thank CIMIT team for getting this entire work being done, and donors, USAID, Bill Melinda Gates Foundation, Syngenta, CRP Maize, and also CASP. Calro is an immense partner to give the land free to conduct the research. So we immensely thankful to those partners who has been supporting in and out. And finally, I thank all my colleagues and then friends, and then I sincerely thanks for organizing committee for giving me this opportunity, especially Dr. Raman and Avinit. I sincerely thank you, sir, and I'm open to take questions, and I'm also thankful to take this feedback.
So now the talk is open for discussion. Couple of questions from the participants.
It is not question. Dr. Suresh,
pardon me?
You have given a very good talk.
Yeah.
And I'm happy that you have joined the Mycological Society of India, and your membership number will come to you very soon.
Yeah, sorry, although I am late to join, but I'm humbled to be part of this society as I work on major key fungal diseases, especially in Africa. And whatever the knowledge and experience what we gain, we are more than happy to share with you all. And also, likewise, I want to learn from you all so that we can enrich our knowledge and help the farmers.
Okay, thank you. Welcome.
Yeah.
Any questions? Professor Suresh, I have one question.
Yeah.
Is there any possibility of this virus jumping the host from maize to some other agricultural crop?
Yes, it is possible. You are absolutely correct. Especially maize chlorotic mottle virus has several wide weed hosts which can go, and also it can go to sorghum and several other some of the cereal crops. But maize being one of the primary hosts, so that's where we could find this one. And more importantly, seed contamination is the key. So we need to keep a constant vigilance on the seed movement in the especially transboundary areas.
So in the surveillance methods, so isn't it right to include those possible hosts also?
Yeah, definitely yes. But the emphasis was on maize, and also we collected other hosts. Yeah.
Thank you, sir. Thank you. So if there are no more questions, it's my pleasure to conclude this session. In this session, we have had 4 excellent lead lectures. First, we heard Professor Hibbert, who dealt with the integrating classical taxonomy with modern phylogenomics, taking Lentinus genus as a model organism. Next, we heard Professor Bhagiraja, who spoke about AMF biodiversity as a function of cropping systems. And my take-home message from this talk is it is very important to practice multi-cropping systems to increase the biodiversity or to at least preserve it. The third lecture we heard was by Professor G.S. Dhingra, and who shared his vast experience of working with poroid and non-poroid fungi, isolated from wide ecosystems, truly a classical taxonomy to the core. The last lecture was by Dr. L.M. Suresh, who spoke on the challenges of containing maize leaf necrosis, which is caused by a combined infection by mixture of viruses, and it has a great economic impact on maize productivity. Finally, I would like to thank the Professor Avaneet and the members of the organizing committee for giving me this opportunity and the honor of co-chairing this session. Professor R.S. Singh, would you like to add anything to this? Okay. If no, on behalf of Professor R.S. Singh, Chairman, and on my own behalf, I would like to thank all the speakers and the participants for a lively morning session. With these words, I would like to hand over the mic to the organizers. Thank you.
Thank you very much, sir. And I'm sure our students have learned a lot from this beautiful galaxy of stars who have deliberated so much on the mycology. Being a cytogeneticist today, somewhere in the back of my mind, I think I also have a question. Should I have gone into for mycological research? Like that, those questions are still coming up now. But with that, that we were supposed to have a break of about 15 minutes. If it is okay with everyone, since we've already gone beyond our time limits, Could we proceed to the next session? And we can have a cup of tea at our places simultaneously, along with the next session, sir?
Yeah, that's fine.
We can proceed.
With the permission. Thank you so much, sir. Thank you so much. Now we will go in for the 10th session of the conference for the Young Scientist Award lecture. And I would like to invite Dr. Geetika Sarhandi to kindly take over as the moderator for the next session, please. Over to you, Geetika ma'am.
Thank you, Dr. Aneet. Thank you so much. I first thanks again the organizing committee for giving me this opportunity second time on the last day, and that is for the very vibrant session, that is for the Young Scientist Award, in which budding mycologists, they're going to present their work, and I first give them good luck that they will present and compete well and get the award from this. Now, before proceeding further, I first like to introduce to this conference our chairperson and co-chairperson for this session. First, the chairperson is Dr. V. Shanmugai. He is assistant professor at Department of Microbiology and Technology, School of Biological Sciences, Madurai Kamraj University, Kamraj.
Dr.
Yes, sir.
Dr. Bhuvaneshwari, yes, Bhuvaneshwari is the chairperson.
Okay, sir. I have chairperson as this one, and co-chairperson is Bhuvaneshwari. No problem, sir. I will repeat it. So sorry, sir, I have the
person, and Shanmuga will be the co-chairperson.
Okay, sir. I have chairperson is Shamugai, and no problem, sir. Sorry, ma'am, Bhuvaneshwari ji. I'm extremely sorry. So I again repeat, but welcome you as a chairperson of this session for Young Scientist Award, Dr. Srinivasan Bhuvaneshwari. She is the assistant professor at Department of Botany, Bharati Women's College, Chennai, Tamil Nadu. And her area of interest are aerobiology, microbiology, mycology. So like our session, vibrant session, she is also very variant research interest to which is going on aerobiology, microbiology, and mycology. Welcome, Ma'am Bhuvaneshwari ji, for this session as a chairperson. To us, along with Bhuvaneshwari ma'am, there is a co-chairperson, Dr. V. Shanmugai. He is assistant professor at Department of Microbial Technology, School of Biological Sciences, Madurai Kamraj University, Madurai. His area of research are also very variable. Secondary metabolites from rhizosphere of medicinal plants to defense-related enzymes to bio-control agents for plant diseases to plant growth hormones from bacteria and fungi. And in her bag of achievements, he has he done his postdoctoral fellow in Department of Biology, Italy, and he got Young Scientist Award in the Federation of European Microbiologists, Geneva, Switzerland. And he also completed one of his research project funded by UGC from 2011 to 2014. So I welcome you, sir, also as a co-chairperson for this Young Scientist Award session for today. In this session, if I'm not wrong, there are going to be about 11 presentations in this session. And I hand over the stage to you now, chairperson. Yes, sir. Yes, Raman, sir.
1 minute for me. 1 minute.
Yes, sir. Yes, sir. No problem. No problem, sir. No problem.
Yeah, I request the chairperson and co-chairperson to evaluate the candidates based on 100 marks. 100 marks. 100 marks. Yeah. And you have to give one topper, only one topper candidate, one candidate for the award, and restrict the time for 10 minutes because it is an award lecture. The time is also important. Thank you, Dr.
Okay,
Geetika.
Thank you, Dr. Ramanji. Thank you so much. So, Madam Chairperson and Dr. Co-Chairperson, I hand over this stage to you for this Young Scientist Award session. Please, ma'am, proceed. Thank you, ma'am. Thank you so much. Ma'am Bhuvneshwari ji, unmute. Ma'am, please unmute yourself.
Okay.
It's okay.
First of all, I would like to thank MSI Secretary, Professor Raman sir, and the organizing committee members, Department of Botany, University of University of Patiala, for giving me this opportunity to chair a very prestigious award session, the MG Trimalachar Young Scientist Award lectures, and I'm very much thankful for honoring me to chair this session. Thank you. First, I would let us start this session with the first speaker, B.N. Narendra Babu.
Yes, ma'am.
So I call upon B.N. Narendra Babu.
Thank you, ma'am. Very good morning. Can I share the screen?
Sure, you can do it.
A very good morning to everyone. Myself, Narendra Babu, BN. I'm working on antifungal activity of the pigments produced by Aspergillus nudulens under the guidance of Professor S. Shishupala. The natural pigments are produced by plants, animals, and algae and fungi. The synthetic pigment causes detrimental effects on effects such as cancer and other allergic reactions to the human beings. So there is a more focus pertaining to the microbial pigments. So among the microorganisms, fungi are known to be a dominant pigment producers. These, apart from using as a coloring agent, these pigments also exhibit a various biological activity. So in our studies, our objective is to determine the antifungal activity of the pigment produced by Aspergillus nudulens. This fungus was isolated by from the soil using a potato-dextrose agar medium. So, and the pigment production was done by using a potato-dextrose broth, and these pigments were extracted by using ethyl acetate as a solvent. Both cellular and secretory pigment were extracted and tested for their antifungal activity. There are many assays were conducted to test the antifungal activity. One among these is the conidial germination assay. In this assay, the target fungi used are Alternaria, Carularia, Fusarium, and Cladosporium species. 1.5 mg of crude ethyl acetate extract, pigment extract was dissolved in 1.1% of DMSO, and controls 0.1% DMSO, and sterile water was used, and the target spores was mixed with the pigment solution and kept in the moist chamber for 15 to 20 hours. Then the conidia were observed for the production of germ tube in different microscopic field. For each treatment, a minimum of 100 conidia were considered. Then the conidial germination percentage for each treatment was calculated. The germination inhibition over the control was also calculated using the formula germination in control minus germination in the treatment divided by germination in the control into 100. Then the disc diffusion assay was conducted for this. Candida albicans and Cryptococcus cultures were obtained from National Center for Microbial Resources, Pune, where the selected candidates were swab inoculated on the sabrodextrose agar medium. So, and the pigment loaded with the disc were placed equidistantly on the inoculated plate. The fluconazole was used as a positive control, and ethyl acetate was used as a negative control. And these plates were kept in the 37 degrees Celsius in an incubator. Then we conducted the bioautography techniques, where we have used 3 mg of crude ethyl acetate extract and loaded on the TLC silica gel plate and allowed to evaporate. And over this, the thick suspension of spore suspension of Alternaria, Carularia, and Cladosporium in separate were made in the potato-dextrose broth and sprayed onto a TLC plate using an atomizer. Then these plates were kept in the moist chamber. Then after this crude one, the thin layer chromatography bioautography was done to check whether the separated fraction having the antifungal components or not. For this, the crude pigment were allowed to separate using an ethyl acetate hexane solvent system, and the separated plate was then allowed, then sprayed with the fungal spores and incubated in the moist chamber to see the zone of inhibition. The results clearly indicated that in the spore germination assay, the secretory pigment from Aspergillus nidulans showed very inhibitory activity against Cladosporium species and Carvularia species, whereas there is no inhibition was observed against Alternaria and Fusarium species. Whereas the cellular pigment did not show any of the inhibitory activity to the target fungi tested. This is the disc diffusion assay against the Candida albicans. The secretory pigment of the fungi Aspergillus nidulans show as slighter inhibitory activity against the Candida species. The cellular pigment from the Aspergillus nidulans show a slight inhibitory activity against Cryptococcus species, and both the cellular and secretory pigment showed a slight inhibitory activity against the Cryptococcus species. Then the biotography was conducted. This is the crude one. The secretory pigment from the Aspergillus nidulans showed a clear zone of inhibitions against the target fungi, that is Alternaria species. This is against the Cladosporium species. The cellular pigment from the Aspergillus nidulans did not show any inhibitions against the Cladosporium species, whereas the secretory pigment showed an inhibition activity against the Cladosporium species. This is the fluconazole, the positive control we have used, and this is the ethyl acetate. This is against the Carvularia species, where both cellular pigment, this is the cellular pigment from the Aspergillus nidulans, and this is the secretory pigment from the Aspergillus nidulans. The secretory pigment showed more inhibitory activity against the Carvularia species. In biotography, the secretory pigment showed inhibitory activity against Alternaria, Carvularia, and Cladosporium species, whereas the only crude cellular pigment showed inhibitory activity against the Carvularia species. So for further studies in the TLC biotography, we have used only cellular pigment for the against the Carvularia species. This is the plate which is separated by using ethyl acetate and hexane solvent system. The same plate was sprayed with the spores, Carvularia spore suspension. After the incubation period, the brown red pigment having the 0.73 RF value show inhibitory activity against the Carvularia one. This is against the Alternaria species. Here the brown brick red pigment of having 0.73 and 0.80 showed up activity against the Alternaria species, and the slight yellow pigment showed inhibitory much inhibitory activity against the Alternaria species. This is against the Cladosporium species. So we observed more number of fractions were shown the inhibitory activity against the Cladosporium 0.20, 0.51, the RF value having 0.51, which is non-pigmented but showed inhibitory activity, and 0.59, 0.71, the brick red color pigment, and it is 0.787, it is in light yellow pigment one, and 0.96, which is having a yellow pigmented one, with the 0.7, the RF value having 0.71 showed a maximum zone of inhibitions against the Cladosporium species. Then again, it is against the Carvularia species. The RF value, the brick red of having RF value having 0.71 and 0.81 showed and 0.95 showed an inhibitory activity against the Carvularia species. Among these, the RF value having 0.95 showed a maximum zone of inhibitions against the Carvularia species. The discussion in the present study, the pigment producing Aspergillus nidulans showed antifungal activity against the target fungi. The Ascomycetes fungi belongs to the Monoscus, Penicillium, Fusarium, etc. are generally known to produce various kinds of bioactive compounds. The secretory pigment from Aspergillus nidulans showed an antifungal activity against Candida albicans and Cryptococcus species in a disc diffusion assay. The Saravanan et al. Reported the antifungal activity of the pigment from Aspergillus stomaceri against Candida and other fungal species. The TLC bioautography suggested the different detection of bioactive pigment fraction against the target fungi. Bioautography is one of the screening method commonly used for the detection of antimicrobial activity. From this, we can conclude that the specific antifungal activity of the pigment from Aspergillus nidulans was detected. Variation in antifungal activity with respect to a different target fungi and differences in antifungal activity between the cellular and secretory pigment are of significance in biotechnological applications. If these active fractions are found
1 minute. 1 minute to go.
Then they may be used in food and pharmaceutical industry. These are the references which I have used. Thank you, Anandal. Thank you, MSI, for giving this wonderful opportunity to share my research.
Okay. Are there any questions from the audience?
Look, you have done the antifungal activity from the pigments.
Yes, sir.
What is the actual application? Because you have not tested what type of mycotoxins are produced by this, because large number of compounds are already reported from Aspergillus nidulans. What way it will be novel?
Sir, if the target by using TLC's bioautography, we can able to know that which pigment fraction having the antimicrobial activity. If that pigment was found to be non-toxic to the humans, then that may be applicable. Then they may be used in food and pharmaceutical industries.
You see, lot number of toxins are also produced by Aspergillus. So you have to
Yes,
first
sir. We have to check the toxicity, sir. We have not yet checked the toxicity. We have to check the toxicity, sir.
So in first case, you have to check the toxicity of the human cell line, plant cell line, then you can say it has some biotechnological application. Till
Yes,
that time, there is this just like other screening.
Yes, sir.
Okay. Okay, I just want to ask you one question, Narendra Babu.
Yeah,
why did you choose Aspergillus nidulans? What's the reason for choosing Aspergillus nidulans?
Ma'am, we have isolated from the natural sources. We have not taken particularly Aspergillus nidulans. We have isolated other fungi.
What is the natural source from you?
The soil was
from
used.
Soil. Okay.
Yes, ma'am.
What type of soil?
Ma'am, we have not taken particular type of soil, ma'am. In and around the Davangere, we have chosen different soil types, clay
Okay.
and all.
Okay, in soil, the most dominant one is Aspergillus niger. And why did you choose Aspergillus nidulans?
Because it was produced when we screened the fungi for pigment production. We got this was Aspergillus nidulans was a major pigment producers among others.
Okay.
So your main target was to produce pigment or to get antifungal? What was your target?
First, I have to get the pigments. Then with that pigments, I checked out the antifungal activities.
So what is the advantage of the pigment having antifungal activity?
This can be sprayed onto if the mainly we have chosen the plant pathogenic fungus for the target organisms. So if the active compound is against that organism, if you spray onto the plant material, that there is a chances for reducing the diseases.
Okay, but what is the role of pigment then?
That pigment is only having which pigment is having the antifungal activity, then that pigment can be produced in a bulk quantity and sprayed onto the plant materials.
Your process should be very simple. Take a culture, take antifungal screening and use it. There is no need to have a culture with pigment for antifungal activity in particular.
Okay. Thank you, sir.
Are there any questions from the audience? As there are no questions, I move on to the next speaker, Gagan Barar from Punjab University, Patiala. Domestication of wild edible agaric, Macrocybe gigantea. Is there?
Yes, ma'am.
Gagan? Okay. You can proceed.
Very good morning to all of you. I'm here to present domestication of wild edible agaric Macrocybes argentea. As we all know, population is increasing day by day, and the ever-increased world population puts great pressure on the food sources, as the result of which large fraction of population faces challenges of malnutrition.
Kindly share your screen, Gagan.
Yeah.
Okay.
Press
Okay,
F5.
now it's visible. Start.
Press F5 for full screen. Yes.
I'm here to present on topic domestication of wild edible agaric. Very good morning to all of you. I'm here to present domestication of wild edible agaric Macrocybes argentea. As the population is increasing day by day, the ever-increased world population puts great pressure on the food sources, as the result of which large fraction of population faces challenges of malnutrition. This necessitates the search of an alternate food resources having a great nutritional value and enormous health benefits. Mushroom is one such food substitute with large potential, serving multifarious nutraceutical functions along with large nutritional value. Therefore, it is need of art to develop less laborious and cost-effective strategies to cultivate such type of mushrooms for human welfare. Macrocybes argentea is a one such edible mushroom. Genus Macrocybe belongs to family Tricholamataceae. Earlier species of Macrocybe were placed within the genus Tricholoma. Later on, it was segregated in genus Macrocybe on the basis of presence of clamp connections and absence of ectomycorrhizal associations. This genus is known by 7 species world over. From India, 5 species are known, from which Macrocybes argentea, Macrocybes lobensis, Macrocybes crassa are edible. They are typical in possessing exceptionally large fleshy carpophores that often found growing in large cespitous clusters. Macrocybes argentea was found growing wild in a park in Urban State Phase 2 near Punjabi University campus, Patiala. It grows in large cespitous clusters having a convex shaped cap with a slightly depressed center. It has free gills in young stages and later on it becomes adnexed in mature carpophores. Pileus has a enrolled margins and it always grows in cespitous clusters. It has very large, robust sized carpophores with thick flesh. This collection was made in 2017 August. Later on, this mushroom was again collected in 2018 from the same place. For understanding the best conditions for the vegetative growth of the fungus, evaluation of various physical and biochemical parameters was done. In physical parameters, we evaluated the different solid and liquid media, effect of temperature, pH, and light and darkness on the mycelial growth of Macrocybes argentea. And in biochemical parameters, we evaluated the effect of different carbon sources, nitrogen sources, effect of vitamins, trace elements, and growth regulators on the vegetative growth of the fungus. First, we evaluated the different solid media for vegetative growth of Macrocybia gigantea. In this, we evaluated the 15 different media. The composition of different media was according to the given by Newton 1969. After that, mycelium was inoculated in the flasks and it was kept in the incubator. Different 15 media prepared were these, in which malt extract agar shows the maximum vegetative growth on the basis of the colony diameter in centimeter, which was observed on daily basis. This histogram shows the same results, in which malt extract agar shows the maximum growth and CDA Japek dox agar it shows no growth, and in Eliot agar it shows the minimum growth. This is photograph shows the basal medium selected based on the colony diameter, in which it shows the maximum growth. That was the malt extract agar. Later on, we evaluated the different liquid media for vegetative growth of Macrocybia gigantea. In this, we took 16 media. The composition of each media was taken according to the Newton 1969. The inoculation was done, and we took 3 replica of each medium. And in this, glucose peptone medium was selected as the best medium, in which it shows the maximum growth. In this case, we took the dry weight of the mycelium in mg per ml. This photograph shows the basal medium selected on the basis of the maximum vegetative growth. That was the glucose peptone medium. And this histogram shows the same results, in which glucose peptone medium shows the best maximum vegetative growth and WMSM shows the minimum vegetative growth. Later on, we determined the incubation period for Macrocybia gigantea. After evaluation of best solid and liquid medium for the mycelial growth, we came from the incubation period, in which we took the maximum vegetative growth of Macrocybia gigantea. It was determined so that optimum mycelial growth can be checked at on which day it occurs maximum. This experiment was performed for 38 days, in which it shows the maximum vegetative growth on 33rd day, and after that it starts decreasing day by day. So we took the 33rd day as the best day to check the maximum vegetative growth. After that, we evaluated the effect of temperature for mycelial growth, in which in solid media we took malt extract agar medium, which was selected as best from the various solid media. In this, we took 5 temperature, 15 degrees Celsius, 20 degrees Celsius, 25, 30, 35 degrees Celsius, and 40 degrees Celsius, in which it shows the maximum growth at 30 degrees Celsius temperature. And this histogram shows the effect of the different temperature on the mycelial growth in malt extract agar medium. At 15 degrees Celsius, it shows no growth. After that, this same temperature was evaluated for the vegetative growth in liquid medium. That was the glucose peptone medium. In this, we took the basal medium as a glucose peptone medium, and we incubated it at different temperature like solid medium, same as a solid medium like 15 degree Celsius, 20 degree, 25 degree, 30 degree, 35 degree, and 40 degree Celsius. In this, it shows maximum growth again in 30 degree Celsius temperature. This histogram shows the same results, in which it shows no growth at 15 degree and 20 degree temperature and 40 degree temperature. This photograph shows the maximum growth observed at the 30 degree temperature in the basal liquid medium. That was the glucose peptone medium. After evaluation of temperature, we came for different effect of different pH on the vegetative growth of Macrocybia gigantea in solid medium. In this, we adjusted the pH with the help of buffers, citrate buffer and phosphate buffer. And after that, we adjusted the pH with the help of one normal NaOH and one normal HCl. After evaluation of different pH, we observed that it shows maximum growth at pH 5, and at basic pH, it shows no results. At extreme acidic pH, that was the 3, it shows no growth. This histogram shows the results at which it shows the maximum growth at pH 5 and at extreme acidic pH 3 and at basic pH from 7.5 to 10 pH, it shows no growth. And this photograph shows the maximum mycelial growth at pH 5. In this, we also took the colony diameter on centimeter basis. After that, pH level was evaluated for the vegetative growth in liquid medium. We took glucose peptone medium as a liquid. In this, it shows the maximum vegetative growth and 5 pH, at 5 pH, and in this also, at basic pH, it shows no growth. This histogram shows the results in which it shows the maximum growth at 5 pH, and in basic pH, it shows no growth, and at 5 pH, it shows the maximum growth. On the basis of the dry weight of mycelium, we observed this. After that, we check the effect of the darkness and light on the vegetative growth in both solid and liquid medium. In this, we took the, we gave the different conditions of light and dark, and we observed that in darkness, it shows the highest growth as comparative to light conditions in both solid and liquid media. This histogram shows the results in which in dark, it shows maximum growth as compared to the light conditions. Later on, we came to biochemical parameters. In this, we evaluated the different effect of different carbon sources for the vegetative growth. We selected basal glucose peptone medium as a liquid.
Your time
medium.
is up. Your time is up.
In this, glucose is the carbon source, so we replaced the different glucose with the different carbon
You can
sources.
conclude your session, Ma. Your time is up.
In this, we took 2 controls in which no carbon source was present, in the other control 2 without any carbon source, in which it shows the maximum growth at the control 1, that was the with glucose. Same was done with the different nitrogen sources. In this, we replaced the peptone with the different nitrogen sources. In this, we took 2
Please
controls.
go to the conclusion slide, Ma. Already it's time.
In this, we took the first control with peptone and in second control without any nitrogen source. In this, it shows the maximum growth in yeast extract agar, and in this at aspartic acid, acrylamide, cysteine, glycine, urea, it shows no growth. After that, we evaluated the effect of supplementation of the basal medium with different trace elements for the vegetative growth in glucose peptone medium. In this, we took the basal medium. After that, it was supplemented with the different concentrations with different trace elements. In this, we took the 5 elements and their mixture was also taken. In this, we took 3 concentrations of each element and their mixture. That was the 1 ppm, 5 ppm, and 10 ppm. In this, one control was taken in which no supplementation of trace element was done. In this, it shows the maximum growth at the magnesium 5 ppm. It shows the same results. At magnesium 5 ppm, it shows the maximum growth. After that, evaluation
Gagan,
of
please conclude, Ma. It's already time.
effect of different vitamins was observed. In this, we took different concentrations of vitamins and maximum growth was observed in the medium, which was supplemented with 50 ppm vitamin C. After that, we evaluated the basal media with different growth regulators with different concentrations, in which it shows the maximum growth at the indole butyric acid at the 40 ppm. This histogram shows the same results. After that, we evaluated different grains at different boiling points for the spawn production, and it shows maximum growth at wheat grains for spawn production at 40 minutes. After this histogram shows the same results. This is the fully colonized spawn packets. After that, evaluation of various agro-wastes for mycelial colonization was determined, in which it shows the maximum growth at wheat straw. This histogram shows the same results, in which it shows the maximum results at wheat straw. Conclusion: work was started with an aim to cultivate this mushroom, so various parameters were evaluated for vegetative growth. It was concluded that it shows maximum vegetative growth on malt extract agar and glucose peptone medium at temperature 30 degrees Celsius and at pH 5. Darker conditions promote more growth than light conditions. Maximum mycelial growth is observed when glucose is present as carbon source. And it was also observed on maximum vegetative growth when medium was supplemented with vitamin C 50 ppm, IBA 40 ppm, and magnesium 10 ppm. Wheat grains at 40 minutes promote the vegetative growth, and wheat straw is the best for the food body production. So food body will be further produced on the wheat straw. Thank you.
Are there any questions?
Already the time now is up. You can proceed.
Okay, as there are no questions and the time is running short, we can go to the next speaker, Sankrita Gonkar from Goa University, Goa.
Yes, ma'am, I am here.
Okay, you can share your slide, ma.
Ma'am, I am not able to share my screen because she has not.
She has to go down. The previous speaker has to close down the slide.
Gagan, please close down your.
Gagan,
stop sharing. Stop sharing.
Remove your screen. Stop sharing. Exit the meeting. Gagan, exit the meeting.
Yes, sir.
Exit the meeting. Leave.
Somebody can assist her.
Leave the meeting.
Yes, sir, there's somebody on the way.
That will help.
We'll help, sir.
Respected chairperson, co-chairperson, senior scientists, and friends, good morning to all. I am Sankrita Gaonkar, studying at Department of Botany, Goa University, under the guidance of Professor Bernard Rodriguez. So I am going to present my work on the topic entitled Effects of Bioinoculants on the Growth and Phosphorus Uptake in Rhizophora mucronata Seedlings. Mangroves form a specialized ecosystem in the intertidal zones, being adapted to fluctuating salinity and extreme tides. These are highly productive ecosystems of tropical regions. However, the nutrients like nitrogen and phosphorus limit their growth and development. Therefore, the microorganisms that can solubilize and mobilize phosphorus play an important role in phosphorus nutrition. So, one of them are arbuscular mycorrhizal fungi, which form symbiotic relationship with the plants, thereby promoting phosphorus uptake. Another type of microbe like phosphate solubilizing bacteria can help in solubilization of insoluble phosphorus in the soil. And hence, using these microbes as bioinoculants can enhance the growth of the plants. Thus, the objective of our study was to examine the influence of single and dual microbial inoculation on plant growth, biomass, and phosphorus nutrition in Rhizophora mucronata. So most of the phosphorus present in the rhizosphere is unavailable for the plants as it is bound to calcium in alkaline soil and to iron and aluminum in acidic soil, making it insoluble. So the organic acid secreted by PSPs can solubilize bound phosphorus, converting it to soluble form. And AM fungi form hyphal network in the rhizosphere, which can help in mobilization of the soluble phosphorus. So based on the literature survey, there are only few studies pertaining to diversity of AM fungi in mangrove habitats, and also the studies on the effect of bioinoculation of the growth of mangrove plants are scarce. Shorao Island of Goa has around 250 hectares of mangrove cover, and for our study, the samples were collected from 70 mangrove species, out of which 11 were true and 6 were associate mangroves. The mangrove plants were identified using the following manuals. Soil analysis was carried out by standard methods, and the results showed that Shorao mangrove soil was acidic in nature with increased salinity in salinity levels in true mangrove areas, and the phosphorus level was low in both true and associate mangrove areas. Root colonization was estimated using trypan blue staining technique, and AM fungus spores were isolated using wet sieving and decanting method. To multiply the spores recovered from field, trap and pure cultures were prepared, and these spores were identified using the standard manuals and referring to the INVAM website. Spores were identified based on the color, size, ornamentation, wall layers, and hyphal characters. The person colonization, isolation frequency, and relative abundance were calculated using the following formulae, and statistical analysis was performed using SPSS software. Colonization was found to be maximum in Thespesia populnea, which is an associate mangrove, whereas it was least in true mangrove Avicennia marina. Increased colonization rates in associate mangroves could be due to their distribution in the landward areas of mangrove habitats where inundation and salinity levels are low. Besides, some of the true mangroves showed higher rates of colonization. However, the spore density was maximum in Cereops tagal, which is true mangrove, whereas it was lowest in an associate mangrove, that is Acrosticum aureum. Higher spore density in true mangrove could be due to the host species and the environmental conditions maybe. And among the 17 mangrove plant species investigated, Acalospora dilatata showed maximum relative abundance, followed by Rhizophagus interradices. Hence, these two AM species were used for the further studies. Apart from AM fungi, phosphate solubilizing bacteria were also isolated from mangrove plants of Shoram Island using serial dilution plate method on Picoskaya agar medium, and the halogen forming colonies were purified by continuous streaking. And biochemical analysis were carried out using the standard methodologies of Burgess method. Following biochemical tests were performed, and based on these tests, bacterial isolates were tentatively identified as Bacillus species. The molecular sequencing of the bacterial isolates was carried out at RGCB Kerala, and the sequences obtained were analyzed using BioEdit and Mega software. The sequences were deposited in NCBI Gene Bank, and the results of molecular analysis indicated the close relationship of PSP1 with Bacillus subtilis and that of PSB2 with Bacillus halotolerans. Also, the qualitative analysis of phosphate solubilization was performed on Picoskaya's bromophenol blue agar medium, and the solubilization efficiency was calculated using the following formula. Phosphate solubilizing efficiency was more than 3 folds higher in PSB2 than that of PSB1. To further confirm the solubilizing efficiency, quantitative analysis was carried out using vanadium-molybdate phosphoric yellow color method, and the solubilization of tricalcium phosphate would have occurred due to the production of organic acid by the bacterial strains, which was confirmed by a significant drop in the pH of the liquid medium, and the PSP2 could solubilize higher amounts of insoluble phosphates in the quantitative estimation as well. Then the screening of the effective bioinoculants was performed by using AM fungi and PSBs in different combinations. Then the several growth parameters were analyzed, and mycorrhizal dependency was also determined. All the vegetative parameters of Rhizophora mucronata varied significantly in comparison to the control treatment. Plant height was maximum in treatment 7. And these pictures show the difference in growth of control and inoculated plants. The treatments with consortium of Rhizophagus intraradices, Acalospora dilatata, and the PSB significantly enhanced the total biomass of Rhizophora mucronata. And the mycorrhizal dependency values reached maximum in treatment 7, indicating the dual inoculation of plants with AM fungus species and with PSB could produce greater biomass of Rhizophora mucronata seedlings. Chlorophyll content was analyzed using Anand's method. Total chlorophyll was high in treatment 7, which could be due to enhanced plant phosphorus nutrition. Then for the estimation of phosphorus, plant samples were digested and the analysis was done, was carried out using the following method. And the phosphorus content in leaf was high in treatment 7, which could be due to the synergistic relationship between AM fungi and PSB, which enables AM fungi to translocate phosphorus that is solubilized by the PSB. The root colonization was also found to be increased highest in treatment 7, and a highly significant correlation was found between the total chlorophyll and leaf phosphorus, indicating their effect on each other. It is well evident from the present work that single and dual inoculation using AM fungi and PSB enhance the growth and uptake, phosphorus uptake in Rhizophora mucronata plants. The AM species as well as PSB isolated from the mangrove ecosystem can grow and thrive in saline conditions, hence can assist plants in stress tolerance during the extreme environments. Also, the bioinoculation can be employed in restoration of mangrove ecosystem, and these can be used as biofertilizers as well. Thank you for patient listening. I thank MSI for giving me the opportunity to present my work.
Are there any questions from the audience? Good presentation,
Nice
Gagan.
presentation. Yes.
Thank you, ma'am.
Presentation. Well
Then we will move on to the next speaker, that is Natasha Barnens, who's going to talk about crude oil hydrocarbon degrading fungi isolated from marine environments as potential bioremediation agents. Natasha is here.
I think if he is not there, you can move to the next lecture.
Natasha, kindly unmute yourself, Natasha.
She's there?
Yes, sir, she's there.
She's there, okay.
Natasha, you are not audible, bache.
Unmute.
Natasha, please unmute.
Ma'am, I think she's having some technical problem. We can proceed with the next. She may be.
Okay.
Dr. Avneet, I think she is not with the system audibility. उनको log out करके log in करे दोबारा और system नाल audio system
Okay.
Okay,
so we'll move on to the next speaker so that she can present in the last as the last speaker. We'll give her some time. Okay, the next speaker is Taranjeet Singh from Punjab University, Patiala.
Good morning to all. Ma'am, I am audible?
Yeah, you're audible. You can proceed.
Okay, let me share my screen first, ma'am. Yeah, good morning to all. Just wait, ma'am, it's being... Yeah, good morning to all. Today I'm going to present my lecture for the young scientists who are on the topic that is purification and characterization of fungal endonuclease for the preparation of fructooligosaccharides from. In this, we first purify and characterize our desired enzyme, that is endonuclease, and this purified enzyme is further being used for the preparation of fructooligosaccharides, which is a well-known prebiotic. So starting with the purification, for the purification, isopropanol precipitation has been carried out, and the different concentration of isopropanol, that is around 40 to 90%, has been taken. And it has been found that at a concentration of 70% isopropanol, the maximum recovery of 81.83% with the purification of 0.96% was obtained. Now, after performing isopropanol precipitation, then iron exchange chromatography has been carried out. In this, for the iron exchange chromatography, the E cellulose column has been taken, and in the fraction 15, 16, and 17, maximum enzyme activity was found out. Moreover, in these fractions, the maximum protein content has been obtained, and these fractions has been eluted out with the help of a 0.2 molar NaCl. Now, after performing the ion exchange chromatography, these three fractions, which is having the maximum enzyme activity, have been pulled out and further being used for the purification using the size exclusion chromatography. The fraction 12 obtain the maximum enzyme activity, and this fraction has been rooted out with the help of a 0.1 molar phosphate buffer, which is having a pH of 6.5. After performing the side-stream chromatography, the molecular weight of purified enzyme has been found to be 53.7 kilodalton. Then this is the purification summary of all the purification steps. After performing the purification on all the three columns, finally we get the purified enzyme, which is having a specific activity of 57.86 with a purification of 11.9%. Then after performing the purification, the molecular weight determination has also been carried out. And for this, the SDS page analysis has been carried out. In the lane 5, the presence of a single band shows that the protein which we have get is the monomer. And for the analysis that that our protein is being not been included with other impurities and not, the native analysis has also been carried out. In the lane 5, the presence of a single band shows these results. Then after performing, then another technique for determination of the molecular weight is the regression of a log molecular weight with that of relative mobility. In this, the red dot shows that the purified enzyme has been purified, and from this graph, the molecular weight of purified enzyme was found to be about 53.45 kilodalton. Then there is a determination of the optimum pH and pH stability studies. The optimum temp pH of the purified enzyme was found to be 5.5, and the enzyme was found to be at show the maximum activity at the temperature of 55 degrees Celsius. Similarly, for the pH stability range, enzyme was found to be stable at a pH range ranging from 4 to 7, and the enzyme was found to be stable at a temperature of 50 to 70 degrees Celsius. Then is the trace metal effect of trace metal elements on the enzyme activity has also been carried out. In this, the calcium chloride and magnesium sulfate was found to enhance the enzyme activity, so these have been considered as an activator. Whereas the copper sulfate, iron, cadmium, and cobalt and other salts found to decrease the enzyme activity, so these are being considered as an inhibitor of the enzyme activity. Then the thermodynamic inactivation parameter studies has also been carried out, and this is the T half value and D value has been calculated. The T half value has been decreased from 55 to as we rise the temperature. Similarly, the D value was also decreased with the increase in temperature. Moreover, the very less difference between the delta H and delta G value showed that the purified enzyme has been thermostable in the range from 50 to 70 degrees Celsius. Then the kinetic parameter characterization has also been carried out. In this, the Km value for the inulin strategos has been calculated. The Km value for the inulin was found to be less than that of strategos, which means that the enzyme is being more effective toward the degradation of inulin than that of strategos. And similarly, the high value of the Vmax and Vmax ratio oblique Km and the Kcat value has also been calculated. The high value of the enzyme for the inulin justifies its potential for the inulin than that of the strategos. So this is a paper which is being published by our group, and it is being published in the International Journal of Biological Macromolecule, which has an impact factor of 5.16. Then after the purification of enzyme, this purified enzyme has been used for the preparation of the fructooligosaccharides in a batch system. And for this purification and this preparation of fructooligosaccharides, stats have been used, that is the RSM technique has been used. And for this, the independent variable like inulin enzyme load, hydrolysis time, and agitation speed has been optimized. The inulin load has been optimized from 3.8 to 8%. Enzyme load has been optimized from 25 IU to 75 IU. Hydrolysis time has been varied from 5 hours to 15 hours, and agitation speed has been varied from 83 RPM to 167 RPM. So based on these results, the CCRD design has been obtained. And in this, we found out that at the run 5, the maximum fructooligosaccharides yield of 99.19% was obtained at a concentration of 7.3% inulin with an enzyme load of 65 IU, hydrolysis time of 13 hours, and under the agitation speed of 100 RPM. Then based on this, the NOVA analysis has also been carried out. In this, we find out the lack of it was found to be not significant, which justifies that the model which we have selected is accurate. Moreover, the p-value of all the parameters which we have selected was found to be significant. And then is the goodness of fit value for the stats model. In this, we found that the R-square value was found to be very much close to the 1, which justifies again that the model which we have selected has been good. And moreover, the difference between the adjusted R-square and predicted R-square was less than 0.2, which again justified that the model which we have selected again fitted and authenticated. Then next is the RSM plot, which shows the interaction between the different independent variables on the fructooligosaccharides yield. Then is the TLC determination. In this, the picture one shows the TLC chromatogram, and this is the TLC densitogram. Here the GF2 represents the castos, GF3 represents the nistose and GF4 represents the fructophiloside nistose. Basically, these are the different degree of polymerization fructooligosaccharides. And then is the validation parameters have also been calculated. In these are the runs which has been predicted by the model in order to justify that the model which we have selected is been accurate or not. So in this also the experimental value and the predicted value are very much close to each other, which again justifies that the model is accurate and validated also. Then this is a TLC representation of the different runs which has been predicted by the model. Here also the GF2, GF3 and GF4 represent the Castro, Nestor and fructophiloside nistose. Then again is the, now after the, in the earlier slide I have shown that the maximum fructooligosaccharides yield was obtained in the fifth run, which is about 99.19%. And in order to
2 minutes to go.
In order to determine whether how much of the degree of polymerization fructooligosaccharides has been obtained in the total fructooligosaccharides yield, we use the HPTLC technique, and from this we have found that in the run fifth, 99.19% of total fructooligosaccharides contain 33.85% of esters, 24.5% nistose, and 7.26% of fructophiloside nistose. Also, the other degree of polymerization fructooligosaccharides has also been present in this fraction, which is about 33.58%. Then this is also the presentation of the different fructooligosaccharides yield, which has been obtained in the different run as predicted by the model. So this is another paper which we have published by our group.
Taranjeet, you have 2 minutes.
Yes,
you can
ma'am.
go
Yes,
for
I'm
conclusion.
concluding my work. I'm concluding my work. Just wait, ma'am. So this is another paper which we have published in another reputed journal. So these are the conclusion of my work. The three-step purification technique has been obtained, which show that the purification process which we have used is the up to mark. The vibrant stability of the pH and temperature show that the biocatalysts which we have used have application at industrial scale. Moreover, the KMBF values are also determined, which show that the catalytic selectivity of an enzyme towards the different biocatalytic processes. And then the endonulins which we have purified have also a potential for the fructooligosaccharides production, which is a well-known prebiotic in the today world. And the result obtained in this study, which can also help us to develop a large-scale process. So this is my presentation for the award lecture. Thank you.
Thank you. Are there any questions?
Excellent work, Taranjeet. Well done.
Thank you, sir. Thanks a lot.
Are there any questions? Okay, Taranjeet, I just want to ask one question. Why did you choose Aspergillus tiranti? What is it? From where did you get that fungus for your study? Can you hear me, Taranjeet? Taranjeet, can you hear me?
Ma'am, there is a problem of electric supply in Patiala in our campus. I think Taran is not having the backup.
Okay.
So that is why his video is still, we can proceed with the next one, ma'am.
Okay, next speaker. We'll go on to the next speaker. That is Mahendra Jeet Kaur from Punjab University, Chandigarh, India. Mahendra Jeet Kaur.
Maninder, you can start.
You can share your slides. Unmute, ma. Unmute.
Maninder, you are not audible, beta. You are not audible.
Is it a power problem there?
There looks some technical snag, sir.
Yeah, I think then any other paper from elsewhere can we see in the meantime?
No, this is Punjab University.
I know.
Yes. Shared her screen, sir.
That I'll stop, ma'am. We can stop her screen and we can move on to the next one.
Okay, okay, sir.
Correct.
So I call upon Apurva Shavan from Goa University. Apurva Shavan, Goa University.
Yes, ma'am, here.
Okay.
So Maninderjeet, you're ready?
Hello.
Maninder, your audio is not proper, beta.
Now who is available? We can ask them to.
Okay, okay, sir.
I think Maninder will be, she's there, but it's not, I think so, yes, now.
Apurva, you can start.
Apurva Shavant, you can start. You can start sharing the slide. Are you there?
Yes, ma'am. Hello. Yes, ma'am.
Apurva?
Yes, ma'am. Yes. Hello.
Apurva?
Hello. Yes, ma'am. Hello.
Hello.
Apurva is coming or slide is
Yes, sir.
loading.
Yeah.
Hello. Yes, sir.
Yes, you can start.
Yes. Ma'am, am I audible?
You are audible.
Audible, but your screen, yes, both you should.
Person, mycologist, and friends. Good afternoon. I am Apurva Savan, PhD student from Department of Botany, Goa University, working under the guidance.
Your screen is not seen. Presentation.
Apurva, I think there is some problem with the connectivity issue at your place.
Shall we go to the next speaker, sir?
Yes, ma'am, that will be better.
Okay, the next
While the
speaker is.
next speaker joins, I just want to request the young scientists to please remain calm. Such technical glitches may occur. You should not get panicked and stressed with your presentation. All the best. You all will do well.
Thank you, madam.
We'll go on to the next speaker, Kolam Sharda Devi from University of Delhi, New Delhi. Effect of Rhizophagus intraradicis on the chemical profile of Ocimum caniflorum essential oil. Please proceed.
Sharda Devi is there. Yeah, she is coming.
Good afternoon, dear dignitaries, revered chairperson, co-chairperson, and all those who are present in this virtual meet. I, Thoktum Sarda, from Delhi University, would like to present my work on the effect of Rhizophagus intraradicis on the chemical profile of Ocimum teniflorum essential oil. Ocimum teniflorum, Ocimum teniflorum, synonym Ocimum sanctum, or commonly regarded as the queen of herbs in Ayurveda, is immensely valued for its medicinal properties. Some of its pharmacological properties include its antioxidant, anti-inflammatory, and antimicrobial activity. As a result, they have paved their own way in various industrial sectors such as food, cosmetics, and pharmaceuticals. Consequently, various commercial products of this plant, such as its essential oil or their leaves, dried leaves in the form of green tea additives or herbal tea and herbal powders, are commercially available. And recently, they have gained much interest from the Western market. India stands proud as a major exporter of tulsi and its products. Major exporter of tulsi and its products in different countries all around the globe. In the year 2019, according to India Today, a report of India Today report documented that the export value of Ayurvedic and herbal products from India has increased from $355 million in 2015 to $446 million in 2019. So the increase in demand of this plant and its products compelled the adoption of an appropriate culture practice for cultivation of this highly sought after plant. In this regard, avascular mycorrhizal fungi offers a promising potential in sustainable cultivation of this plant and also improving the productivity. The role of avascular mycorrhizal fungi in harvest yield and on the metabolite accumulation of various aromatic and medicinal plants have been increasingly realized in the past 2 decades. Surprisingly, from an extensive literature review from my side, what I could decipher was studies pertaining to Ocimum teniflorum in relation to AMF are just limited to assessment of the diversity of avascular mycorrhizal fungi in the Ocimum sanctum rhizosphere, or assessment of the response of Ocimum sanctum to different avascular mycorrhizal fungi. So in order to exploit this sustainable AMF technology in the cultivation or in the sustainable cultivation of this commercially sought after plant, I have laid down my objective, and the objective read as effect of Rhizophagus intraradices colonization on 2 genotypes of Ocimum teniflorum in terms of harvest yield and essential oil concentrations that are of commercial importance, and composition of essential oil and phytochemical constituents that are of therapeutic importance. For conducting a pot experiment, seeds of 2 high-yielding genotypes of Ocimum teniflorum were procured from NBPZR, and they were inoculated with Rhizophagus intraradices, a pure inoculum of Rhizophagus intraradices procured from dairy. Colonization was checked at the time of transplantation as well as harvest. And in comparison between the 2 genotypes, genotype B showed comparatively higher colonization percent at both transplantation as well as harvest. Once the colonization has been confirmed, the effect of AMF colonization on the commercially important parameters of this plant, that is harvest yield and the EO concentration. AMF colonization in roots led to increase in the leaf biomass in body genotypes, and similarly, essential oil concentration also increased in body genotypes. So from this, what can be deciphered is that colonization of colonization of this plant roots with AMF leads to double promotion of this plant from the commercial viewpoint. Since the essential oil is biosynthesized and accumulated in the pelted glandular trichome of the plant, trichome density was also estimated to validate the increased essential oil concentration, and as evident in this graph as well as in the scanning electron micrographs, AMF colonization led to a significant increase in the trichome density. Now, looking beyond the commercially important parameter of these plants and looking into the therapeutically important parameters, that is the composition of the essential oil and the active principles, the effect of Rhizophagus intraradices colonization was assessed based on the GCMS analysis. and the metabolite profiling of the essential oil extraction of genotype A showed that in non-mycorrhizal plants, 23 compounds were identified, and whereas in mycorrhizal plants, 21 compounds in total were identified. So from the analysis, what we could decipher was the essential oil of Ocimum tenuiflorum is a concentrated heterogeneous mixture of terpenoid derivatives and phenylpropanoids, and of which sesquiterpene and phenylpropanoids are the major contributor. The same trend of decreasing number of compounds detection in mycorrhizal plants was also observed in genotype B. And sesquiterpene and phenylpropanoids were also the major contributor in the essential oil composition here as well. This Venn diagram shows an overview of all the compounds and how the composition of essential oil differ in two genotypes in response to mycorrhizal colonization. Out of the 54 volatile compounds that were identified, 13 compounds were common in both the treatments of both the genotypes. And what is interesting in this finding is that these 13 compounds and this common 13 compounds encompasses the compound of our interest that are of medicinal importance. To list them, they are eugenol, germestrin A, beta-alanine, and beta-caryophyllene. While eugenol is a phenylpropanoid and is the active component of Ocimum tenuiflorum essential oil, beta-alanine, beta-caryophyllene, and germestrin A are terpenoids. So regardless of the genotypes, the content of these active compounds increase in response to AMF, though the extent of the increase vary. So, and it is still a gray area to decipher the deciphering of the underlying mechanism in this. The difference in the extent of increase is yet to be deciphered, and we are still working on it. So, coming from the, apart from the essential oil, the leaves of Ocimum tenuiflorum are also commercially regarded owing to their antioxidant potential. So the effect of Rhizophagous intrarhizosis colonization on the antioxidant properties were identified, and for this the leaves were taken in two sets, fresh leaves and processed leaves. Processing of leaves was done according to green tea, processing of green tea as described by Ahmad and Stepp. Since phenol, flavonoid, and polyphenol content are the
Kapkom, you have 2 minutes. Please conclude.
The key indicator of the overall antioxidant potential of plants. These findings of the study found that mycorrhizal colonization led to increase in the in phenol, flavonoid, and polyphenol contents in both the genotypes, and comparatively fresh leaves showed higher content of phenol, flavonoid. Following this, antioxidant potential was also determined, and it was found that regardless of the genotypes, mycorrhizal colonization increased their total antioxidant potential, and comparatively, in fresh leaves showed higher antioxidant potential. So to sum up the findings of this study, I would conclude with the statement that AMF augmented biomass and essential oil yield resulting in the increasing the commercial value of the plant, and the enhanced concentration of active principles and the boosted antioxidant potential of the plant resulted in the increased therapeutic value of this plant. This is a publication that comes from this objective. With this, I would like to conclude my presentation. Thank you, one and all.
Are there any questions? Okay, have you tried with any other plants apart from Ocimum?
No, ma'am, actually, I started with two species of Ocimum, Ocimum tenuiflorum and Ocimum basilicum, but for now we are strictly restricting it to Ocimum tenuiflorum only.
Okay. Okay, ma'am, thank you.
Thank you so much.
So we can go to the next speaker.
Okay.
Yes.
Anik Sarkar. University
Yes,
of
ma'am.
Calcutta.
Yes, ma'am. I am here. Am I
Yeah,
audible?
you can proceed. Yeah, you're audible.
Okay, okay. Uh, Good afternoon. Myself, Onik Sharkar from Department of Botany, University of Calcutta. I am pursuing my PhD under the supervision of Professor Krishnendu Acharya. Here today, I am going to present my work entitled Nitric Oxide, a Signal Mediator in Chili Alternary Alternata Interaction. As we are all well known about the importance of chili, chili is one of the important cash crops grown for its pungent fruits. It is the rich source of different types of vitamins, minerals, and proteins. India is now become the major producer and exporter of chilies. Among the different horticultural crops, chilies are providing 1.5% share of country's GDP. Different types of stresses are associated with significant reduction in chili production. Among the different types of biotic and abiotic stresses, alternaria leaf spot disease is nowadays emerging. Till today, very few reports are available regarding the chili alternary alternata interaction. To protect this pathogen, it is an urgent requirement to understand detailed mechanism of pathogenesis. For this purpose, two chili cultivars, one tolerant Beldanga cultivar and one susceptible bullet cultivar were selected, and pathogen Alternaria alternata was procured from Agarkar Research Institute, Pune, India. Disease index study showed that the leaf spot symptoms were developed on both the cultivars, but in case of susceptible cultivar, the symptoms were more prominent than the tolerant one. Here you can see. And observation of pathogen progression in both the cultivars clearly demonstrated more profuse growth of pathogen on the susceptible cultivar. Callose, one of the most important beta-1,3 glucan polymer that forms temporary cell wall against the foreign invading phytopathogens. Aniline blue staining of callose deposition showed that in response to pathogen, the tolerant cultivar showed higher amount of callose deposition than that of susceptible bullet one during the period of pathogenesis. Defense-related enzymes protects plants in various ways against the different types of stresses. Among these defense-related enzymes, phenylalanine ammonia lyase is considered as key enzyme in phenyl propanoid biosynthesis pathway. Peroxidase and polyphenol oxidase, they are involved in lignification and cell wall integrity. Here, in response to the pathogen, in case of tolerant Beldanga cultivar, higher activities of different defense-related enzymes were observed than that of the susceptible one. Like the defense-related enzymes, the antimicrobial and antioxidant compounds as total phenol and total flavonoid showed higher accumulation in tolerant cultivar in response to the pathogen invasion. In contrast, in response to the pathogen, chlorophyll degradation was much more higher in case of susceptible cultivar during the entire pathogenesis period. Stress marker lipid peroxidation indicates the extent of membrane damage. Here, in response to pathogen, higher amount of lipid peroxidation in susceptible cultivar marks the higher membrane injury than that of the tolerant cultivar. Histochemical visualization of cell death and hydrogen peroxide and reactive oxygen species generation were performed by using DAB, DCFDA, and trypan blue staining. We found that in susceptible cultivar, at every time point of pathogenesis, ROS production and concomitant cell death were higher than that of the tolerant cultivar. Semi-quantitative RT-PCR analysis of different defense-related genes and pathogenesis-related proteins showed differential patterns in their expression levels in both the cultivars in response to the pathogen invasion. Now, the question is, what is the function of nitric oxide? In response to this answer, we have checked the nitric oxide status in both the cultivars. And we found that during the different time points of pathogenesis, the tolerant cultivar showed higher amount of nitric oxide generation than that of susceptible cultivar. The biochemical results of nitric oxide generation was further confirmed by real-time nitric oxide generation using DAF2DA dye. This dye is a membrane permeable dye, and this dye can bind nitric oxide directly, and it will give a brilliant green fluorescence. So like the biochemical results, here you can see that the tolerant cultivar showed higher amount of nitric oxide generation in response to pathogen than that of susceptible bullet cultivar. In order to find the signaling role of nitric oxide, the plants were treated with nitric oxide modulators, and sodium nitroprusside or SNP was taken as the nitric oxide donor, and CPTIO was used as nitric oxide scavenger. We found that during the nitric oxide surplus condition, that is the SN-treated sets, the activities and accumulation of different defense-related enzymes were higher in both the cultivars, but in nitric oxide depleted condition, all the defense parameters were compromised. Like the biochemical results, nitric oxide content was orchestrated in both the cultivars in response to different types of modulator treatment. The overall story can be summarized by this depictive map. The higher amount of nitric oxide generation in tolerant cultivar is directly correlated with increased defense responses in against the alternata invasion. Lastly, I would like to conclude that nitric oxide function as signature molecule during chili alternata interaction. Recently, this study has been published in Physiological and Molecular Plant Pathology. Thank you.
Are there any questions? Okay, then we will move on to the next presentation by Pooja, University of Delhi.
Yes, ma'am. Hello.
Yeah.
Am I audible, ma'am?
Yeah, you're audible.
Shall I start, ma'am?
Yeah, you can start.
Good afternoon, everyone. I am Pooja from University of Delhi, and I am here to present my work titled Diversity of Secreted in Xylem Genes and Phylogenetic Relationship Among the Members of Fusarium oxysporum Species Complex. The pathogen that I work on is Fusarium oxysporum. It's an anamorphic species which causes vascular wilt, stem, root, and crown rot diseases in over 120 plant species. The pathogen infects the host via root, results in xylem blockage that is characterized by xylem discoloration, leading to wilting of plant and eventually plant death. The pathogen can be characterized in the basis of former specialist Lycopersicea is specific to tomato, whereas former specialist Cubans is specific to banana. These former specialists is a particular cultivar of plant host. For instance, in Cubans, there are 4 different races of the pathogen that are specific for 4 different cultivars of banana. Together, these former specialists and races forms the Fusarium oxysporum species complex. A major outbreak of Cubans was observed on banana, which is commonly termed as Panama disease, which led to huge losses of banana production. So considering the economic impact of this pathogen, it has been positioned as the 5th most significant phytopathogen. The system that I particularly work upon is safflower and former species Carthamai pathosystem. Safflower is an oilseed crop that is grown in India in majorly Madhya Pradesh, Maharashtra, Telangana, and Karnataka. Oilseed production in all these states is majorly compromised due to Fusarium wilt of safflower. The disease is prevalent in all states with incidence of up to 80% in the Indian Deccan Plateau region. These slides communicate that in India. The point here to note is that how a pathogen that shows a broad host strain is able to infect a particular species. The answer lies in the genome of the pathogen shows presence of specific pathogenicity-related genes called as secreted in xylem Until now, 14 genes have been identified in tomato-specific pathogen, former specialist Lycopus. Several roles have been attributed to these genes, which includes role as avalence factors and role as effectors. Their role in conferring host specificity in two pathogens, but the role that I will be focusing on in my presentation is their role as markers in race and former specialist identification and discrimination. So if these genes can be used as diagnostic markers for identification, then we hypothesize that the area of 16 should vary between different former specialists of the Fusarium oxysporum species complex. So based on that, the first objective was laid down, which was to investigate the diversity of secreted in xylem genes in different former specialists. For this study, all the isolates that are mentioned in the table were used and were procured from different institutes. The DNA was isolated using the CTAB extraction method of DNA, and the DNA was subjected to amplification using primers that were specific for 6 genes. These are the results for PCR amplification. These pictures represent bands that are obtained for a particular gene in a particular former specialist. Based on these gel pictures, a table was created where the presence was demonstrated using plus sign and the absence of 16 was demonstrated using minus signs. It is evident from this table that the area of 16 is variable among the former specialists. With Lycopus in which the genes were discovered, 14 genes are present, whereas in former specialists like Gladuli, only 3 genes are present. It can also be inferred from this table that Fusarium species other than oxysporum do not show presence of 16. So this corroborates with the hypothesis that these genes can be used for identification of Fusarium oxysporum pathogens. Based on the presence and absence of 6 genes, cluster analysis was also constructed, where we observed that most former specialists which show presence of 6 genes cluster together in clade S1, whereas former specialist Carthamai, instead of showing presence of 16, clustered together with Fusarium solani. This is an observation which is different from hypothesis, so we are working upon it for further validation. The second major feature of the members of Fusarium oxysporum specialist complex is the polyphyletic origin. Isolates classically in a former specialist should show only polyphyletic or paraphyletic. This is also attributed to the horizontal transfer of 6 genes from a pathogenic to a non-pathogenic strain. There are several endophytic strains that are documented to colonize asymptomatic plants, and they show the potential that they can acquire these genes horizontally and transform into a new pathogenic lineage that can prove to be a serious concern for resistant line breeding. Thus, to evaluate the nature of their origin, the second objective was laid down, in which we evaluated the interspecies relationship between Fusarium oxysporum and former specialists based on internal transcribed spacer region and translation elongation factor gene data sets. These are the isolates that we included to the study, and in this table, so based on those sequences, trees was created. These are the results for objective 2. This is the tree that was obtained from ITS data set. Here we can see that most of the former specialists are clustering together in major cluster IT1, and Fusarium species are clustering together in minor clusters IT2 and IT3. The second tree was obtained using translation elongation factor 1 alpha gene. Here we can see that most of the former specialists, similar to ITS data results, are clustering together in major cluster T1, except the pathogen Fusarium oxysporum carthamai. These results are corroborating with the results from cluster analysis, but not from the ITS analysis. That's why, to resolve this incongruency, we generated a concatenated phylogenetic tree. And here also we obtained results similar to translation elongation factor alpha gene data sets. Most of the former specialists were clustering together in the major clade C1, whereas former specialists like Patiala and Carthamai were clustering together in minor clades. Here we can see that most of the former specialists are showing independent lineages, indicating that they must have evolved independently during the course of evolution. So summarizing my results from both the objectives, I can say that the investigation of 16 diversity revealed that every former specialist have a different area of 16s, and then they can be used as diagnostic markers for identification of the pathogen. The phylogenetic analysis hinted towards the polyphyletic nature of the pathogen, suggesting that there is complexity in the origin of pathogenic lineages. The trees that were obtained through cluster analysis on the basis of presence and absence of 6 genes and concatenated traits were in concordance and placed the pathogen Fusarium oxysporum parthami in support to Fusarium species. With this, I will conclude my presentation and would like to thank the organizers of MSI for conducting the conference online successfully. Thank you.
Any questions from the audience? Why did you choose Fusarium oxysporum?
Fusarium oxysporum, we actually started with the pathogen. We are going through the major limitations on the cultivation of safflower in India, because when we looked at the data of safflower production, India was the major producer in 2014 for safflower seeds. But after that, it was taken over by other countries. Even though we have the largest area under production, we do not have the equivalent amount of yield. So by looking at the limitations of safflower production, I worked upon this pathogen, Fusarium oxysporum, and I was doing literature survey. I realized that this pathogen is something which is not has been dealt till now and needs to be taken further.
Okay. Okay. Thank you. Are there any questions? As there is no question, we go on to the next.
Yeah, Pooja, just a minute. Pooja,
yes,
have you seen these fungi under microscope?
Yes, sir.
Okay. Did you see some differences in these pharma species?
So there is no morphological difference in the Forma specialis. There is some differences when it comes to pigmentation, but only for a few of them.
Because they are all clustered together in ITS. So
yes, sir, they all clustered together in ITS. Yes, sir. So there is no morphological differences, and it's being a polytypic species.
Yeah, even okay, but good work you did. All the best.
Thank you so much, sir.
Definitely, the conidial size of the Fusarium differs from species.
Yes, ma'am. The conidia size differs.
Did you measure that?
Ma'am, I have not measured it, but I have observed that
even though it appears same, all the Fusarium species resembles the same. The conidial size and the macroconidia and the microconidia, yeah, it differs.
Yes, ma'am.
But there is also enormous plasticity in their size. You know, they may change if you grow them in different culture medium. They again show differences in their. It's enormous variation they show. Yes. But it is Fusarium is a wonderful fungus to work, even from a taxonomic point of view. All the best for your future work.
Thank you so much, sir.
Thank you, Pooja.
Thank you, ma'am. Thank you.
Okay, we go on to the next presentation by Sudeshna Nandi, University of Calcutta.
Hello.
Yes, you're audible.
Am I audible?
Yeah, you're audible.
Okay. Hello. Good afternoon, everybody. I'm going to deliver us my presentation on the topic astracurcurol potentiates apoptosis, autophagy, and attenuate cell migration via fine-tuning the AKT signaling in human lung cancer cells. Among all the cancers, lung cancer is one of the preeminent health intimidating disease that occurs in worldwide, and it have a high mortality rate both in males and females. Our Moderna target therapies have several complications, which includes side effects, lower stability of the drug, huge cost with limited treatment facility. So an attempt has been made by various researchers and scientists to establish a novel anti-cancer drug, preferably from the natural resources, as they have very lesser toxicity. So in this scenario, medicinal mushrooms are coming out as an encouraging substitute to chemotherapy, and their exploration are opening much more new modern routes in mycomedicines. To fulfill this purpose, here we have chosen a wild edible gastromycetes named as Astreus hygrometricus. The purpose of choosing this particular mushroom is that previously in our laboratory, this mushroom has shown immense biological activity. So we made an attempt to isolate and formulate a novel bioactive compound named astracurcurol from Astraeus hygrometricus for successfully targeting the cancerous cells. Looking at the first figure, here we can see that astracurcurol treatment has inhibited the proliferation of the A549 cells as well as altered their fusiform shape. Along with it, it also increased the dead floating cells. Next, WST assay was performed. This assay showed that astracurcurol has inhibited the cell proliferation in a dose-dependent manner and helped us to calculate the IC50 value, which comes out to be 69.42 micromolar. To our interest, on treating the PBMC cells with astracurcurol, it has substantially increases the survivability of this cell without any cytotoxicity. To conclude, to go in accordance with our previous findings, we have done the colonogenic assay where astracurcurol was found to prevent the formation of colonies in a dose-dependent manner. So, to ascertain that whether this decline in cell viability was due to the apoptosis, nuclear morphology analysis was done with DAPI. Here we can see that in control set, the cells have their nuclear morphology and they have a homogeneous blue staining, whereas in the treatment set marked with the pink arrows, they shows the apoptotic cells which are comparatively smaller in size. They have fragmented nuclei, condensed chromatin, formation of apoptotic body and membrane blebbing. This was further justified with the micrograph of SEM which shows the similar results. Next, the astracurcurol treated cells were double stained with acridine orange and ethidium bromide. This green fluorescence represents that live cells. This yellowish orange fluorescence represents the early apoptotic phase, and this orangish yellow to orangish red fluorescence represents the late apoptotic phase. Further, to quantitatively assess the process of apoptosis induced by astracurcurol, annexin 5 pi banding assay was done, and here we can see that in control set the population was in the first quadrant, which gradually shifted to the second and third quadrant in the treated set, which implies the presence of early and late apoptotic cell population. Astracurcurol was also found to arrest the cell cycle of A549 cells at G0 G1 phase with a significant peak of sub G0 stage. So to rule out whether this astracurcurol induced apoptosis was due to extrinsic or intrinsic signaling pathway and to observe the possible role of AKT in this pathway, astracurcurol treated cells were subjected to Western blotting, and as you can see that treating the cells with astracurcurol has downregulated the phosphorylation of AKT. This downregulation has further downregulated the expression of MDM2 and which is followed by the upregulation of phosphorylated P53. This changes in the upstream protein imbalances the apoptotic and anti-apoptotic markers, thereby causes the upregulation of BAX and downregulation of BCL2, which is the conclusive effect of the cell. So this was further validated by the molecular docking, which is done with astracurcurol and human BCL2 protein, and astracurcurol have shown an encouraging binding energy of minus 8.7 kilocalorie per mole. Extending our study, we observed that this changes in the ratio of BAX and BCL2 has disrupted the mitochondrial membrane potential. Thus, there is a shift in the histogram peak towards the left-hand side. This depolarization of mitochondrial membrane has further causes the release of cytochrome C from mitochondria to cytosol, which is as evidenced from this immunohistochemistry data and Western blotting. Afterwards, the impact of astracurcurol was also observed in the downstream effector of cytochrome C, where we can see that astracurcurol has causes the cleavage of caspase 9, caspase 3 and PAR. So all these findings together confirms that astracurcurol induced apoptosis is governed by mitochondrial mediated intrinsic pathway. Apart from this, astracurcurol was also found to enhance or elevate the expression of some of the prominent markers of death receptor pathway. As we can see that it has upregulated the expression of FAS, FAD both through quantitative real-time PCR as well as through Western blotting. It also induces the cleavage of caspase 8. Thus, it hints that astracurcurol induced apoptosis was not only due to the intrinsic signaling pathway, but also due to the death receptor extrinsic pathway. Apart from this apoptotic property, astracurcurol was interestingly found to trigger autophagy in lung cancer cells. Here you can see that on treating the cells with astracurcurol and staining them with acridine orange, which is a lysosomal staining dye, we can see there is a strengthened red fluorescence in a dose-dependent manner with the formation of acidic vesicular organelles. The formation of acidic vesicular organelle is a prominent hallmark of autophagy. This microscopic images was further validated by flow cytometric analysis, where it shows that there is an increase in the fluorescent intensity of red dye or acridine orange dye, which confirms the formation of autophagic vesicles. Next, to observe the differences in the autophagy related markers, we observe the upregulation and downregulation of some of its major proteins. Here we can see that there is upregulation of Beclin-1 and upregulation of ATG7, whereas there is a downregulation of P62. All this data together confirms the activation of autophagy in lung cancer cells. We know that PI3-AKT-mTOR signaling pathway is usually negatively administered the activation of autophagy. So interestingly, our data here shows that application of astracurcurol has attenuated the phosphorylation of mTOR PI3K, and as I have shown already, it has also downregulated the phosphorylation of AKT. So all this together revealed that there's a possible involvement of PI3-AKT-mTOR signaling pathway in the astracurcurol-induced autophagy activation. Apart from its apoptotic property, we are next interested to observe its anti-migratory potential towards the lung adenocarcinoma cells. So first assay, which is a wound healing assay, here we can see in control cell after 24-hour treatment with astracurcurol, this wound has completely closed, whereas in the treated set in a dose-dependent manner, astracurcurol has inhibited the migration of these cells. And interestingly, the migration has been inhibited at a dose lower than the apoptotic dose. This assay was further validated by a next assay, which is a transwell assay. Here we can also see the similar results, that in comparative to control cells, there was a less amount of cell migrated in the lower part of the transwell. This is further justified with the Western blotting and quantitative real-time PCR, where it shows that astracurcurol has downregulated the surface antigen CD44, as well as it prevented the expression level of matrix metalloproteinase 2 and 9, which are the major protein required for the invasion of the cancerous cell. Finally, we have seen that astracurcurol has also successfully repressed the tumor development in ex ovo xenograft model, which is in accordance with our in vitro studies. So here my entire study has been presented is this schematic diagram, where it shows the pivotal role of the astracurcurol in promoting apoptosis, autophagy, inhibiting the migration and the tumor regression. Also, here we can also see that AKT plays a major role in this pathway, as AKT, by inhibiting the AKT PI3K signaling pathway, which is usually a cell survival pathway, not only trigger apoptosis, but it also activates the autophagy. So we could tell that astracurcurol might emerge as a very good candidate for treating the lung cancer cells, and accessibility of this particular compound, astracurcurol, might extend the survivability and reform the life quality of lung cancer patients. Thank you very much for your attention.
Are there any questions?
From which specific mushroom did you isolated this astracurcurol?
Astrius hygrometricus.
Okay. What is the possibility of this particular specific chemical in Ganoderma and other, say, Trametes versicolor?
Sorry, sir. Can you repeat the question?
Other medicinal mushrooms which are being, because Ganoderma is being used extensively against cancer. There is a PSK drug which is isolated from Coriolus versicolor. You didn't compare the results of this particular astracurcural which you isolated from Astrias with those?
No, actually, we have not compared it with the other mushroom extract or any pure compound, but we compared the anti-cancer efficacy of this particular compound with a very commercially available drug, that is doxorubicin, which is used for treating the cancer. We have compared its activity with doxorubicin.
Okay. Otherwise, from Trametes versicolor, PSK is one of the well-known component which is used against cancer.
Yes, sir.
Okay, thank you.
Where from you got this fungus? Where from you collected this? From Calcutta? Is it?
No, this has been collected from Bakura. Bakura, West Bengal.
Okay. Okay.
Is it being used by local people there?
Yes, it is. It has highly been used by the local tribal people. They take this mushroom as their day-to-day functional food, and it has also noticed that when we have learned this from the tribal people only, that it also that not only they take it as a food, but it also have a very vast medicinal activity.
Okay, thank you. Nice presentation.
Did you purify the astrocurcural?
Yes, ma'am. This has been isolated through column chromatography method, and then it has been the structure has been characterized.
Okay.
It's a good work.
Yes. Okay. Nice presentation. Okay, well, then we go on to the next speaker. Tahira Akhtar from Abdur Rahman Crescent Institute of Science and Technology. Wonderful.
Is there?
Yes.
Tahira Akhtar?
Yes, sir. Yes, ma'am.
You can proceed.
I'm here only. Ma'am, shall I start?
Yeah, you can. You can proceed. You can share your screen.
Yes, ma'am. Ma'am, is my slides visible?
Yeah,
it's visible.
Okay. Thank you, ma'am. A very good afternoon to one and all present here. I am Tahira Akhtar from BS Abdur Rahman Crescent Institute of Science and Technology, Chennai. And present, I am pursuing my PhD under Dr. S. Hemalata. So the work which I'm going to present here is about the efficacy of the endophytic fungi mediated silver nanoparticles against pathogens and multi-drug resistant E. coli strains. The objectives of the work.
Full screen.
Sorry, sir.
Make it full screen.
Okay, sir.
Press F5. Okay.
Sir, now is it fine?
Yeah.
Okay, thank you, sir. So for the objectives of the present work were endophytic from different plant sources and the identification of the endophytic fungi. Then the second step was the fermentation and extraction of the bioactive compounds from the endophytic fungi. Then phytochemical analysis of bioactive secondary metabolites, isolation, purification of and identification of the secondary metabolites. Then synthesis of the nanoparticles from the endophytic fungi. Then characterization by various various techniques like UV visible and EDX, then the antibacterial and the antibacterial activity of the silver nanoparticle against the ESBL and pyocyanin producing bacteria, and the antifungal activity of the silver nanoparticle against the pathogenic fungi. Then anti-cancer activity of the silver nanoparticle by using endophytic fungi, synthesis of silver nano from different plant extracts also as well. So as we all know, the endophytic fungi refers to the fungi which are residing inside the healthy tissues of the plant, and as such endophytic protect their host from the infection and various conditions, creating bioactive compounds. The
it is not well known that how endophytic can secrete the bioactive compounds. So it may be because of the metabolic interaction of endophytic with its host, which may favor the synthesis of biological active secondary metabolites. Then endophytes are novel source of secondary metabolites with potential application in industry, in pharmacology industries and other industries as well. Fungi have been detected as a source of endophytic fungi. Then these are the four plant species which I have used for my research work, and these all the four species I have collected from the university campus. The first one was the Solanum nigrum, black nightshade. Then another was the Plankthrus amboincus. Then third one is the Oxalis corniculata. Then Catranthus. All the four species are having really good medicinal value. This is the here, this is the Catranthus rosas, because here I am not going to present all the data. Here I am only to present about the Catranthus rosas, and this is the Catranthus rosas from which I have isolated the endophytic fungi. I have isolated 4 or 5, but I am showing the so which I am choosing is the Botryospora rhodata from the Catranthus. So this is the schematic representation of the endophytic fungi isolated from the Catranthus rosas. The endophytic fungi was isolated from the Catranthus and was identified on the basis of morphology as well as the molecular as well as the molecular analysis. Then after that, the genomic DNA was isolated by the CTAB method, and the RTS region was amplified by PCR by using the universal primers, and the species was identified by NCBI blood. So after NCBI, we came to know the fungi is Botryospora rhodata. Then this is the phylogenetic tree which shows the relation of the isolated fungi of which we are using for the synthesis of the and closely related strains from the NCBI gene bank. And we have isolated several bioactive compounds from these endophytic fungi. For this, we have for to obtain the bioactive compounds from the endophytic fungi, we inoculated on the potato dextro broth for the 21 days to obtain the secondary metabolites, and to synthesize silver nanoparticles from the mat, we inoculated for 7 days. After inoculation, mycelium vat was removed from the liquid feeder and crude extract were filtered and stored at -4 degrees centigrade. So after that, the qualitative and qualitative analysis of the bioactive compounds secreted by the endophytic fungi was done by different. So alkaloids, flavonoids, these were the bioactive compounds present in the secondary metabolites. And this is the graphical this is the representation of the bioactive compounds present in the secondary metabolites. Then after we have synthesized the silver nanoparticles from the endophytic fungi. For that, these are the steps which we have used. So first we have the PDA broth in the shaker. Then we have after 7 days we have separated the mycelium mat and washed it several times with the water. Then 80 grams of the wet shaking for 24 hours. After 24 hours, the fungal biomass was removed by the filtration, and the cell filtrate was added. With the cell filtrate, 1 millimolar silver nitrate was added, and after 24 hours color change was observed. And further, the silver nanoparticles were characterized by the UV spectra, SEM, TEM, and EDX. Mainly these nanoparticles were utilized to check the antibacterial, antifungal, and anti-cancer activities. So this is the fungal mat. This is the 7 days fungal mat on the PDA broth. After that, after 7 days, we have removed the mycelium. We have removed the mycelium mat and inoculated in the distilled water after several washes by distillation. Then this is after for 24 hours in the shakes are 48 hours. So after 48 hours, we have filtered. So finally, the filtrate was added with the silver nitrate, and finally silver nanoparticles were synthesized. So after that, the first observation whether the silver nanoparticles has been synthesized or not. So UV were taken, and the which shows the SPR peak was at 4F, which gives the confirmation that silver nanoparticles has been synthesized. So after that, FTR analysis, sorry, FTR spectra was also done, which displayed the functional groups present in the silver nanoparticles and the interaction of the silver nanoparticles with other bioactive. These were the functional groups present in the silver nanoparticles. And after that, EDX was also done. EDX reveals the composition of silver nanoparticles, and which shows a strong signal in the silver region and confirms the formation of microsilver with the silver, like other element signals such as carbon, oxygen, potassium were also recorded, which may be due to the other bioactive compounds present in the fungal cell filtrate, which we have used for the synthesis of nanoparticles. Then after that, this is the SAM diagram. We showed the formation of highly aggregated small silver nanoparticles, and then it was followed by the TEM. We showed the shape and size of the silver nanoparticles. After that, said pattern of microsilvers, which displayed functional group. Sorry, the said pattern revealed the nature of the silver nanoparticles, and they were in amorphous in nature. Then these nanoparticles were utilized for the antibacterial to check the antibacterial activity of the silver nanoparticles, and against different bacteria like Pseudomonas aeruginosa, Salmonella typhi, Proteus, Malavirus, Abomini, MRSA, E. coli. Then this is the control which we have used as antibiotic, and these are the different nanoparticles which were used to check the antibacterial again. Effect of nanoparticles were check on the growth of the producing E. coli also, which shows which inhibits the nanoparticles inhibits the growth of the E. coli in a dose dependent manner. So from this conclusion, significantly these micro nanoparticles significantly inhibited the growth of the bacteria also. Then after that, these nanoparticles were used for used against the fungal species, plant pathogens. This was utilized for the plant pathogens also. So R. solani, Fusarium, Gramillonium, Aspergillus niger. So these nanoparticles they inhibit the radial growth of the pathogenic fungi, and from this we can conclude that these silver nanoparticles can be used for the as well as anti-cancer activity in the future, sir. Thank you, sir. Hello.
Okay. Thank you. Are there any questions?
Hello, sir.
I think in the absence of questions, you can move the chairperson.
Okay, sir. I will do it. I just want to ask you one question, ma'am. Did you identify only one fungus? Did you isolate only one fungus?
No, ma'am.
As an endophyte?
No, ma'am. We have isolated many fungus, but here I am showing it one fungus only, ma'am.
Okay. Can you tell me what are the other fungi which you isolated?
Ma'am, one is Lasodiplodia.
Okay.
Oh, 1 second, ma'am. Sorry, ma'am.
Okay, ma. Thank you.
Ma'am, total 4 fungus we have isolated. One is Cytosporia rostrata. Then another is Botryospora rhoda. Then third is Lassodiplodia pseudothrome. Then another one is Daldinia escorte.
Okay, ma. Thank you.
Hello.
So we go on to the next.
Madam.
Yes.
Madam, one more doubt, madam. I am Dr. Ambika from Putti Pushpam College, Tanjavur, Tamil Nadu.
Okay, sir.
I wanted to have one clarification with regarding the silver nanoparticles.
Sure.
Silver nanoparticle.
Yes, sir.
How can you are confirming the silver nanoparticle from your metabolites?
Sir, actually, metabolites, the filtrate which we are getting from the mycelium mat, we are adding the silver nitrate. After that,
Okay. I understand what you are presented in your very nice presentations. But as far as your silver nanoparticles, how can you are confirm whether it is the silver nanoparticle or zinc oxide nanoparticle? How you are confirming?
with the help of the characterization, sir.
Yes, characterization.
Yes,
what are
sir.
the characterizations you are following?
So initially we did the UV, then FTR, then EDX, then XRD. I have shown only little here, SEM, TEM, then these are the characterizations, sir. Like FTIR with EDX also we can come to know whether silver is there, whether silver nanoparticle has been synthesized or not, sir.
Thank you.
So characterization.
Thank you.
Okay,
Thank you.
thank you. Thank you, sir.
The next speaker is Samata Gupta. Samta Gupta, University of Delhi. Are you here?
Yes, ma'am. Good afternoon, ma'am.
Okay. Good afternoon. You can proceed, ma'am.
Shall I present my screen?
Yes, please.
A very good afternoon to the respected dignitaries and all the eminent mycologists present virtually here. Today, in this virtual national conference on biodiversity and biotechnology of fungi, I'm here to present my work, that is colonization by arbuscular fungi reduces arsenic accumulation, improved growth, and more nutrient acquisition in Triticum estimum grown in arsenic contaminated soil. Now, arsenic, being a toxic metalloid, poses a serious environmental threat and contaminates groundwater and soils. As per a recent report of Times of India, around 19% of Indian population drink water heavily contaminated with arsenic. And there are concerns that excessive use of this arsenic-contaminated groundwater will also contaminate agricultural land and result into bioaccumulation of arsenic into the edible parts of food grains. Now coming to the point that why wheat? Wheat has already been demonstrated to be a significant source of arsenic exposure to humans. And in India, majority of wheat growing region coincide with arsenic contaminated wheat.
Please, they are blinking. Your slides are blinking. I don't know why it is happening.
Yes, sir.
You have to. Your slides are blinking, ma. Share it full screen.
Sure, shall I present it again?
Yeah. No, you proceed. But your slides are blinking. I don't know the reason.
Just give me a moment to fix it now.
You have any technical issues?
Yes, ma'am. I'm just switching my system.
Okay, then I'll go on with the next speaker.
Yeah,
Natasha
good.
Barnes. Natasha Barnes.
Yes,
She
ma'am.
has
Can you hear me?
you can proceed. Yeah, you can proceed.
Okay. Yes, thank you, ma'am. Ma'am, I'm sharing my screen. Can you? Can you see my screen, ma'am?
No,
Not
I
yet.
don't.
Not yet.
Yes, just a minute. Okay, is it visible?
You're sharing. Maximize. Yeah.
Make it.
Yes. Okay.
Yes. Yes.
Yes, good afternoon, everybody. Thank you for giving me this opportunity again, and sorry about the technical difficulties earlier. So I'm presenting about crude oil hydrocarbon degrading fungi isolated from marine environments as potential bioremediation agents. This work has been carried out in National Institute of Oceanography, NIO. So coming to a brief introduction about crude oil, basically crude oil is a complex mixture of hydrocarbons which consist of naphthenes, aromatics, paraffins, asphaltics, etc. And we know that crude oil has a very high economic value because it is distilled and used in a number of fuels in different kind of vehicles, including construction of roads as bitumen. Although crude oil is very economically important, there are environmental drawbacks associated with crude oil, and these are mainly because of oil spills that occur on land and water due to transportation activities majorly, and sometimes due to drilling activities. These are pictures of the Chennai oil spill in 2017, which resulted in 25 tons of oil being spilled along the coastline of Chennai. A more recent oil spill in the Indian Ocean is that in Mauritius, which was last year, and it resulted in 1,000 tons of oil being spilled, resulting in a loss of biodiversity and including marine birds and mammals. These are a few pictures of terrestrial oil spills that have occurred in India, which is in Assam, Nagaland, Surat, the tarball deposition that occurs after an oil spill event, as well as the tarballs event seen in Mumbai. So oil spills is not just a local issue, it is a global issue because oil spill is not it is not local. I mean, if there's an oil spill that takes place in Chennai or Mumbai or Gujarat, for example, these oils, because of ocean circulation and currents, can be carried to different countries and across large distances, and because of which tarballs are seen across different places. Now, in India, tarballs the reoccurrence of tarballs has been happening for the past few years along different beaches, including Goa, Gujarat, different places of Mumbai, and there is no explanation on the source of this because there have not been an oil spill close to these locations. So it has been found out that oil occurring in one particular state, the tar ball occurring in one particular state, can actually arise from an oil spill event that has happened in another region. Another example of this is the paper which was published by NIO, in which the tar balls that were found along the coast of Goa were actually associated from the Southeast Asian crude oil and not from Bombay High. And therefore, these prove that after an oil spill event, the tar balls can be carried across large distances, and therefore, oil spills are a global concern and something that should require attention. So what are the remedies available to take care of these spills? So at seas, you have three different, which is dispersion, burning, and skimming, but these are not 100% eco-friendly because burning would also result in additional pollution. Dispersion, again, just disperses the spill. It does not completely take out the spill. So what about onshore spills, that is terrestrial spills? There are 8 different available techniques for removal. Although these require a lot of labor and also are expensive techniques, so researchers are now concentrating on biodegradation agents, which are either bacterial or fungal, which result in complete mineralization of the pollutant as well as their eco-friendly approaches. So keeping this in mind, we wanted to isolate fungi which have these capabilities because we know that fungi have a higher biomass and therefore they would result in a higher conversion rate of the pollutant, as well as the fungal hyphae have the potential to penetrate deeper into these regions as compared to bacterial isolates. So our sampling locations were tar balls from a coast in Goa. Then we have the Arabian Sea, the open ocean, as well as a mangrove system in Diwar. All of these are influenced by oil spills at some point. We have isolated 51 marine isolates, which we have screened for their degradation potential. And out of these, 10 were selected because they showed maximum degradation potential and also they were the fast growers. So these were then selected for gravimetric estimation, wherein we checked the total crude oil, following which we checked qualitative and quantitative estimations using gas chromatography, which we checked two fractions, that is the alkane fraction, which is present majorly in crude oil, as well as the aromatic fraction, which is more toxic fraction. And we also checked the toxicity of these byproducts by using Artemia nocli. So coming to the results, of the 10 cultures screened, the 4 belonged to mangrove ecosystem and they were Penicillium or Aspergillus species and one was Acrimonium. 4 belonging to open ocean, they were all identified as Aspergillus species and this was by the ITS gene sequencing. And the 2 isolates obtained from tar balls belong to Fusarium species. All of these sequencing details have been submitted to NCBI and these are the accession numbers for the sequences. Coming to the gravimetric estimations, here we can see that all the isolates showed considerable degradation. However, one isolate, which is 126 and S22, they were more efficient as compared to the others in the total crude oil degradation. So then we checked the N-alkane fraction. In the N-alkane fraction by GC, the chromatograms were obtained wherein individual compound concentration was calculated and comparing the control and the test sample, we have calculated the degradation percentage. Here we can see that across the 10 isolates and the different compounds tested, these fungal compounds showed good degradation of lower molecular weight hydrocarbons as well as the higher ones, which are a little difficult to degrade because as the chain length increases, their bioavailability decreases. So they become difficult to degrade. However, these 10 cultures proved to be quite efficient. This data has also been published in 2018. Then we checked the degradation compounds in the pH section, in which we checked 10 compounds. And in this case, we have seen that these 5 cultures were able to degrade all the 10 compounds present to different degrees. So these proved to be quite good agents for bioremediation, and therefore we have next tested the toxicity. We have tested the toxicity of all the 10 isolates. However, the 5 that were good are highlighted here. So M126 and S22, which are the mangrove and the open ocean isolate, they are not very good, not recommended as bioremediation reagents because their toxicity was quite high compared with that of the control. So the other 3 isolates, the 2 from tar ball and 1 from mangrove ecosystem, are potential isolates for bioremediation agents because they resulted in at least 50% survival of the Artemia nocli. So these 3 isolates, identification as Fusarium and Acrimonium, were then checked with previous literature to find out what was the degradation percentage obtained previously and whether our cultures are comparable to previous available literature. So here we see that in all the cases, our cultures gave better degradation efficiency and therefore these can be considered as potential remediation agents. Also, their toxicity was assessed and they were not very toxic to Artemia nocli. But also we would recommend, this work is still ongoing, so we are also testing their degradation capability in soil when we put them in soil, as well as their phytotoxicity is being currently assessed, and I have not included those results here. So coming to the conclusions, these fungi were derived from various marine niches, and we have seen that they have a lot of potential, and therefore marine sources should also be considered for the isolation and biotechnological applications. These fungi can also adapt to different environments, and as we have seen, they can degrade a range of pollutants. And the 3 isolates obtained in this study, 2 belonging to fusarium and 1 to acremonium, are potential agents for bioremediation, and these should be further studied for on-site degradation. Before that, we need to perform phytotoxicity assays, which we are currently doing. So the references used for the presentation, and I would like to thank the organizing committee for giving me the opportunity to present my work on this platform, the funding agency, DBT, who has funded this work, my research supervisor, Dr. Sameer Damre, and the director, CSI National Institute of Oceanography. Thank you.
Are there any questions? Okay, you said that you have isolated the fungi from the marine sources. Did you take the soil or water?
Ma'am, for marine, we have taken the sediment, the benthic sediment, as well as the water column. For mangrove, we have taken only the soil, and for tar balls, we have taken directly the tar ball. So any sand that was associated with the tar ball, that has been analyzed.
Okay. What is the medium what medium you used for the isolation of fungi?
We have used different kind of medium. One, because it was directly open ocean, we have used just the seawater medium, which is from that environment itself, and we added a little agar for solidification. We have also used seaweed, Apex dogs, MEA, Maltese extract, and
Did you
saprophytes
check
was
the
also used.
salt concentration? Did you check the salt concentration?
Yes, ma'am. The medium was prepared in the seawater itself.
Okay.
So we have maintained the salt at 35 PSU.
Did it have the same normal growth as it was in PDA, the fungi?
Ma'am, the fungi, when isolated itself, it was isolated in medium containing salts.
Okay. It was a normal growth? Did you
Yes,
study any
ma'am.
growth studies? Did you do
We
any
have
growth
not
studies in
done.
that?
No, we have not done any growth studies. We have only done the
Because you need
degradation.
this particular fungi in if you need it as a biomass, right? You need it in huge quantity. So you need
Yes.
to go in for biomass studies also when you go in for degradation. Okay, so better
Yes,
you
ma'am. Yes.
go for the growth studies.
Okay. Yes. Thank you,
Thank
ma'am.
you. Thank you.
I think somebody is there.
So next.
So may I present my presentation now with this account?
Yeah,
I'm
you can go
Samta
ahead.
Gupta from Delhi University.
Yeah, you can go ahead. You can go ahead.
Thank you so much, ma'am. Ma'am, are my slides visible now?
Yeah.
Yes, they are visible.
It's visible.
A very good afternoon to the respected dignitaries and all the eminent mycologists present virtually here in this National Conference on Biodiversity and Biotechnology of Fungi. I'm here to present my work, that is colonization by arbuscular mycorrhiza fungi, reduces arsenic accumulation, improve growth, and promote nutrient acquisition in Triticum aestivum grown in arsenic contaminated soil. Arsenic, being a toxic metalloid, creates a serious environmental threat by contaminating groundwater as well as soil. And as per a recent report of Times of India, around 19% of Indians drink water that is heavily contaminated with arsenic. And there are concerns that excessive use of this arsenic contaminated groundwater for irrigating the crop fields will result in contamination of agricultural soil with arsenic that will ultimately result in bioaccumulation of arsenic into the edible parts of the food crops. Now coming to the point that why wheat? Wheat has been demonstrated to be a significant source of arsenic exposure to humans. In addition, in India, a majority of wheat growing area coincide with regions which are already contaminated with arsenic. Now, AMF, apart from being a most widespread symbiotic association between plant and fungi, it also beak with its host with biotic as well as abiotic stress tolerance. Now, to evaluate the efficacy of arbuscular mycorrhiza fungi in redeeming arsenic toxicity, a pot experiment was designed. For these, wheat plants, both non-mycorrhizal as well as mycorrhizal, were exposed to different levels of arsenic stress supplied as sodium arsenate. And it was found that at all arsenic levels, M plants possess better growth and biomass than their corresponding NM plants. Presence of arsenic in soil creates a competition in rhizosphere with other essential nutrients such as phosphorus. In addition, they also damage roots that affect mineral uptake, resulting in mineral deprivation in plants. Therefore, we studied mineral acquisition and translocation in wheat plants exposed to arsenic stress. The elemental profile of minerals were generated using ICP-MS, CHNS analyzer, and spectrophotometric method. And this profile was subjected to principal component analysis to evaluate the variation into nutrient acquisition under arsenic stress and AMF status. It was found arsenic concentration was strongly correlated with nutrients such as iron and magnesium. They may help in boosting the antioxidant defense against arsenic-mediated oxidative stress. Again, arsenic concentration showed a strong negative correlation with nutrients such as phosphorus, calcium, sulfur, and zinc. However, mycorrhizal plants having a higher PC1 score favors the accumulation of group 2 and group 3 nutrients and lowers the accumulation of arsenic. Similarly, the PCA analysis in wheat grains showed presence of arsenic in wheat grains negatively correlated with majority of macro and micronutrients. However, M plants having a higher PC2 score favors the accumulation of these macro and micronutrients over arsenic. This heat map shows the relative translocation of nutrients from roots to grain and a decrease of translocation of arsenic in M plants explains our previous observation in grain. Once inside the plant, arsenic interferes with metabolic processes that ultimately affects plant physiology. Therefore, we also evaluated the effect of R intraradices and arsenic stress on photosynthesis. And it was found that at all arsenic levels, M plants displayed a augmented concentration of total chlorophyll, chlorophyll A, chlorophyll B, and total carotenoids. In addition, M plants also mitigated the arsenic-mediated increase in chlorophyll A to chlorophyll B ratio. Analysis of leaf gas exchanges showed that net photosynthetic rate, stomatal conductance, and intracellular concentration of carbon dioxide was higher in mycorrhizal plants over non-mycorrhizal plants at all arsenic concentration. However, the effect of AMF status was non-significant on transpiration rate. In photosynthesis, between the two photosystems, that is photosystem 1 and photosystem 2 is most sensitive to any abiotic stress. Therefore, the efficiency of photosystem 2 was monitored in terms of chlorophyll A fluorescence, and it was found that both potential efficiency of PS2 as well as maximum efficiency of PS2 was compromised under arsenic stress. However, application of mycorrhiza fungi mitigated this arsenic-induced damage on chlorophyll A fluorescence. However, neither arsenic level nor AMF status has significantly affected the photochemical Quanchi coefficient in wheat plants. Similarly, a negative effect of arsenic was also observed on Hill reaction activity. However, mycorrhizal plants augmented the Hill reaction activity at all arsenic level, and mitigated the arsenic-induced damage on Hill reaction activity. To conclude my work, I will say that mycorrhizal plants are better when exposed to arsenic stress over the non-mycorrhizal plants. And I will sum up my study by saying that wheat plants, when colonized by avascular mycorrhiza fungi, not only show augmented concentration of micro and macronutrients, but also decrease the accumulation of arsenic into grain. However, how this differential uptake is regulated, we are still working on it. Additionally, mycorrhizal plants displayed augmented concentration of photosynthetic pigments and better leaf gases exchange, an efficient photosystem 2, and higher Hill reaction activity that altogether contributed to improved photosynthesis responsible for higher grain biomass in mycorrhizal
1 minute more.
observed
1
in
minute
present
more.
study. Okay. With this, I'll thank all the organizers of Mycorrhizal Society of India to present my work. Thank you.
Are there any questions from the audience?
Ma'am, can I?
Yes, ma'am.
Thank you so much. Thanks, Samta, for your nice presentation. I just want to know that your fungi, how and where it works on the photosystem to control this arsenic damage.
Ma'am, the first factor is this fungi is reducing the uptake of arsenic. So it already reducing the arsenic mediated damage on photosystem.
How it controls the uptake of arsenic? What is the mechanism? What could be the possible mechanism?
One possible mechanism is that it provides another route of nutrient uptake, since arsenic competes with phosphate ions in soil, and mycorrhiza provide another route in terms of its hyphae for uptake of this phosphate.
No, arsenic and phosphate, they are competing with each other for the same transporter, or
yes, for the same transporter in routes.
Phosphate shares its transporter with the arsenic.
Yes, ma'am. Since it is analogous to phosphate.
And when it enters into the system, okay, yes, phosphate, but phosphate is necessary for the system to grow.
Yes,
it is very one of the important factor. So if it is competing with phosphate, then yes, arsenic, if it enters, then how sequestration is taking place inside the?
Pardon, ma'am?
When if phosphate is not there in the system, because yes, phosphate is also uptaking.
Yes, ma'am.
You said that the fungi stops arsenic by uptaking more phosphate.
Yes, ma'am.
Arsenic and phosphate, they are through the uniport, or?
But ma'am, mycorrhizal plants provide another route. Mycorrhiza transport phosphate in form of polyphosphate ions.
Okay.
However, there is no report of polyarsenate ion transport by mycorrhizal fungi. So it is a possible method that how AMF hyphae differentiate between arsenate and phosphate.
No, you told that phosphate and arsenic, they are using same transporter system.
Yes, ma'am.
They are symport or antiport?
No, ma'am, they just compete for their transportation sites on transporter.
On the transporters?
Yes, ma'am. It's not like they are transporting all together. It's just they compete for the transporter sites.
And mycorrhiza help in more transporting the phosphate than arsenic?
Yes, ma'am.
Okay, thank you.
You're welcome, ma'am.
Thank you. So we go on to the next participant, Maninderjeet Kaur. Are you ready?
Yes, ma'am. Yes, ma'am.
You can proceed. You can proceed.
The last presentation, chairperson.
One more, sir. One more person. Apurva Sawant.
Okay, okay.
You can proceed, Mani.
Yes, ma'am. Yes, ma'am.
Maninderjeet.
One second. Ma'am, is it visible?
Yeah, you're sharing your screen, but your screen is not visible. You're sharing it.
Yes. Now is it visible, ma'am?
Yes, yeah, it's visible.
Good afternoon to all present here. So today I'm going to present a piece of my PhD work on the topic evaluation of laccase by autochthonous white rot fungus Ganoderma species under solid state fermentation for various applications in the textile industry. So as we all are aware that environmental problems has been increasing day by day. So it is a worldwide issue because the number of industries using number of chemicals which pose serious threats to our health. And from them, textile industry is the major one, which is the prime target for the environmentalist to crusade against the pollution. So globally, textile industry facing challenges owing to the pollution it causes, and various chemicals are used in the various textile wet processes. So that wet processes release number of dyes and chemicals to the environment, which pose toxic chemicals and health hazards to us. So in this regard, there is an increased pressure on the textile industry to adopt a cleaner processing methods. So hence, need of the time is to revamp the conventional process that could lead to a shift from chemicals to the enzymatic textile processing, giving rise to eco-friendly ideas. So here is the overall process which is being used in the textile industry. These are the various textile wet processes. First, they have the raw fabric, which gets desized and scored for the removal of the yellowness of the cotton fabric. The number of chemicals they were using, like H2O2, the most of them which is unspent goes to the water bodies, which pose the serious threats to our health. Instead of the chemicals, here we can use the laccase from the Ganoderma species. After that, dyeing and finishing process was there, which also by which we can, by the use of laccase, we can also use it for the denim finishing. And the unspent dyes which goes into the water, decolorization and detoxification of this can also be done by the laccase. So this is the overall idea of our study, by which we can say a laccase is a viable alternative to the conventional chemical based processes. So for the isolation of that, so for gaining the laccase enzyme, our main aim, our objectives, we designed our objectives in a way that we want the untapped flora of this Chandigarh region, so that we can get a novel fungal strain producing a very high titer of laccase, because till date there are the reports in which by the modification high titers were produced, but as such there is no fungal strain which can produce a large amount of laccase. So after that, we get identified that isolate and evaluated with the help of classical methods and the statistical methods as well. Then we have to characterize characterization of the enzyme with respect to temperature optima and pH optima, because we have to see whether that enzyme can be employed in the textile industry or not. So here I am pressed to one application of my research work. So first of all, we isolate 20 isolate and successfully isolated in the lab 8 isolates and screened for laccase production. So after, out of them, RM14 was the isolate which producing maximum laccase activity. And then we identified it as Ganoderma multipilium. So then we go for its hyperproduction by optimizing various physical chemical parameters. Along with the classical one, we also go for the statistical, and from the classical one, we get 2.4-fold increase by optimizing the parameter. Till date, there is no such report in which we got this much of higher activity. So these are the optimized parameter after which we got this much of increase in the enzymatic activity. So after classical optimization, we also went for the statistical optimization, in which we designed the placket Bowman. And out of that placket Bowman, 12 combinations resulted into the 4 influential factor was there. That is time, temperature, followed by the moisture content and the MnSO4. To optimize these influence factors, CCRD was designed. That RSM design expert software was used, which gave the 30 runs, and the run 4 give the maximum activity. And we also validated it after that, in which we have seen the predicted value is close to the validated one, which shows that the model was successfully validated. So with the statistical optimization, there is an increase of 3.4 to fold activity of the lacase enzyme, which is very high till date. So after that, we partially purified an enzyme and characterize it. We characterize on the base, the partially purified enzyme showed the maximum specific activity of 49,50,980 with 1.8 fold purification and 75% recovery. So this is the temperature profile, which is very novel, because the lacase enzyme showed optimum activity at 60 degrees Celsius. Because in the textile industry, enzyme need to be highly stable at the high temperatures as well, because the processes are done accordingly. So the enzyme was highly stable at the range of 30 to 70 up to 24 hours. And it is also a very interesting property it is having, that it is stable at 90 degree as well. So apart from the temperature profile, it is highly stable. Optimum pH showed to be maximum at 5, and the pH stability profile revealed that lacase, while highly stable in the pH range of 3 to 7, that it has retained more than 80% of the activity up to 5 hours, and at pH 4 to 6, it retained more than 60% of activity even after 72 hours, that is for the 2 days. So here I am presenting very crisp, because this is the main part of our research, as we are going to patent this technology soon, because here we get the very crucial results. Till date, there is no such report that without the use of mediator, lacase is going to decolorize major of the dyes, but we get the result that Sudan black, Ramazol brilliant blue R, indigo carmine, and Congo red. These are the major dyes which employed in the textile industry, majorly used and went into the water bodies as such. But as we can see in the pictures as well, there is a crystal clear solution we got after the addition of the lacase, like in the figure A, which is a Congo red, in the H, which is indigo carmine, and in the K, Ramazol brilliant blue, a crystal clear solution we got after the treatment without the addition of any mediators. So this is a small portion of my research work. Conclusion, this is the first report to best of our knowledge that high titer of lacase were produced by this fungus Ganoderma multipallium. And this is also found to be the first report that 100% of the major dyes used in the textile industry without the use of mediators get decolorized. So we have also collaborated with the textile industry, Ganga Textile Mills, because efforts are being done to commercialize this enzyme technology for the industrial application. So these are the published work from the research. And I'm highly thankful to the Vice Chancellor, Professor Rajkumar, Punjab University, Chandigarh, my chairman, Dr. Deepak Kumar Rahi, and supervisor, respected faculty, technical and non-technical staff, UGC for providing me the Maulana Azad National Fellowship, and my lab mates. Thank you.
Okay. Thank you. And did you do with any other fungus? Did you try with any other fungus apart from Ganoderma?
We have isolated a number of fungi. We tried, and we always also successfully got the results, but this fungus producing the maximum enzymatic activity, that's we are with this only.
Okay, did you try with any Hyphomycetus? No.
No, ma'am.
Okay, thank you.
Thank you, ma'am.
So the next presenter is Apurva Shavant. Are you ready? The last presenter.
Yes, ma'am.
Okay, you can proceed.
Yes, ma'am. Hello.
Yes.
Hello.
Can proceed.
I'm
Yeah, you can proceed.
Okay. Respected Chairperson, mycologist, and friends. Good afternoon. I am Apurva Savan, PhD student from Department of Botany, Goa University, working under the guidance of Dr. B.F. Rodriguez. Today I am presenting a small portion of our work on characterization of bioactive compounds isolated from mangrove endophytic fungi and its potential activity against A549 lung cancer cell. So mangroves, as we all are know, that they are salinity tolerant plant species, and they are associated with many microorganisms that trigger adaptive mechanisms in host and its symbionts. One of such symbiont is endophyte. So these endophytes are asymptomatic microorganisms within the plants, and its presence was described as endophyte by Henry Fredling, which was later termed as endophyte by D. Berry in the year 1866. And it was localized in almost all parts of the plant. So, especially in mangroves, which is a helophytic ecosystem, it is reported that persistence of such endophytic colonization in leaf, stem, root, flowers in almost all parts of the plant. So, such colonization it may be intercellular colonization or intracellular, or in some cases, it may be both. So here you can see in this slide that intracellular colonization, which we have localized in the roots of the mangrove plants. So which forms a highly coiled septate network of endophytic hyphae inside the roots. So this species could belong to basidiomycetes, oomycetes, psychomycetes, or even ascomycetes. Most of them belong to ascomycetes. In our study also, persistence of ascomycetes was maximum throughout the season. So as we all know that endophytes have so many applications, and many prominent scientists working in this area, and we were fortunate enough to hear so many of them in this conference. So the major work these days is on secondary metabolites. So we also tried using these endophytes to see which are the different components which are present inside this endophyte. So therefore, we had selected this study site, that is Shorao Island, Goa, and which is along the Mandovi River. And we had taken healthy mature leaf, and we had sterilized them using 3-step sterilization method by using Suryanarayanan sir's method, which is given in 1998. And then we had isolated them on a potato dextrose agar plate amended with streptomycin sulfate and penicillin G. Then plates were incubated, and they were the cultures. These are some of the cultures which we were recovered after 7 to 10 days of incubation. So our study design was based on the ethnobotanical profile of the plant, wherein literature also revealed the role of endophytes in enhancing the bioactive potential of the metabolites which are present in the host. So we had selected 5 dominant cultures which we had recovered throughout the season, and we had fermented those on a potato dextrose broth. So here you can see these were the 5 isolates which we had selected, which were isolated from true as well as associate mangroves. So it is Alternaria alternata, Curvularia, Pestalopsis, Necrospora, and Cladosporium species, and they were coded with an isolation code. So after 15 days, the fermented culture was separated by using ethyl acetate as a solvent system for the extraction of metabolites, and this was standardized. We tried to separate these compounds on a TLC plate, and then we came to know that ethyl acetate was a better solvent wherein we could separate this compound. So followed by that, for the characterization, we had used phytochemical assay as well as GCMS analysis, which was done at IIT Bombay. So GCMS analysis, this was the procedure which was followed. And then we had done anti-cancer potential. We wanted to see whether this endophytes had any role in anti-cancer potential. So the work was done at KLE, that is Cancer Research Center, Belgavi, Karnataka, using A549 lung cancer cell line, and also normal mouse fibroblast cell L929 was used. So here the viable culture suspension of 50 microliter was seeded in a 96 well flat plate, and final volume was made up to 150 microliter by adding DMEM medium, and DMEM is a basal medium for mammalian cell culture. So we had used that, and by adding different concentrations of endophytic extract, and then incubating same for 48 hours at 37 degrees Celsius in a humidity chamber containing CO2 incubation. Followed by that, we had added, after incubation period, we had added 20 microliter of MTT reagent, and after 4 hours of dark reaction, the supernatant was removed, and then without disturbing the formazine crystals in this tray, you can see the formation of formazine crystals, and without disturbing those, we had added 100 microliter of DMSO, which is used also as a negative control, and then OD was taken at 492 nanometer. Then in this tray, you can see the growing cells with a darker intense purple coloration, inhibiting the cells, when lighter color, purple color showing the cells which are inhibited by the endophytic extract. So here the percent viability, the percent inhibition, as well as IC50 value was calculated. Now results. Ethyl acetate was used as a solvent system, as I've said, and then among 4 cultures showed a very good response, that is belonging to Alternaria alternata, Pastropsis species, Cladosporium curvularia species showed a very good response when it comes to inhibiting the lung cancer cell line. However, in the Nigrospora species showed a lowest inhibitory percentage. Now here you can see this was the morphological change in the plates. You can see here very clearly that this is in the control, this is in the experimental plates. This is with respect to Cladosporium species extract. Now this is with respect to Alternaria alternata, wherein we had got a similar response. Now this is when tested against inhibitory activity of endophytic extract. Here you will see a lowest.
You have 2 more minutes.
Yes. Here you will notice lowest viable cancer cell percentage was observed in MEN89, that is with respect to Cladosporium species, having a very negligible cytotoxic effect on the normal growth of the L929 cell, which you can see in the B graph. Now, similarly, in other species also, we had noticed a similar phenomenon, and we can say here that endophytes are the better organisms to work with. And here we had also noticed that MEN85, as I've said, that Nigrospora showed a highest showed very less this one anti-cancer potential. But when it comes to survival of viable cells in the L929, it showed a more than 80% of the viable cells in the normal cell. So we can still work more on in these areas. And this was the GC chromatogram which we had done, and these are the compounds which was isolated and identified by using GCMS analysis, which have reported to have various properties, anti-cancer properties, antimicrobial properties. Conclusion: endophytic fungi are a novel source of natural origin microorganisms, and which deserve exploration. So we would still would try to work more in this area, so that we can get some more organisms which have anti-cancer properties. So thank you. Thank you.
Any questions? Any questions from the audience? As there are no questions, I would like to thank all the presenters. You have beautifully presented today for the award. I congratulate each and every person who presented in this session. Good luck. And I would like to thank the organizers, Dr. Avneet, and the organizing committee of the Department of Botany, University of Patiala. And I would like to thank Professor Anand, Secretary, MSI, for giving me the opportunity. Thank you, one and all.
1 minute, madam. Can I take 1 minute?
Sure, sir.
Yeah. This session originally planned with Chairperson Professor Krishnananda Acharya and Co-Chairperson Dr. Samir Damere. since both of their students are in this session, they opted out, and so Dr. S. Bhuvaneshwari and Dr. Sanmugia were opted as chairperson and co-chairperson, and Dr. Bhuvaneshwari has effectively chaired the session. I don't know whether Shanmugia has joined or not.
No, sir, I think
okay.
he was not.
Okay, you are the single judge, Madam Dr. Bhuvaneshwari.
Okay, sir, I'll do my best.
Another 1 hour time, kindly send to my mail one topper's name and marks for 100.
Okay, sir.
Another 1 hour so that I can write in the certificate.
Thank you, sir.
Thank you, Dr. Geetika, for an effective moderation. Thank you, Madam.
Thank you, Dr. Raman. Thank you, Madam Srinivasan, for nicely handling and managing this session, which is very knowledgeable, as well as the presenters. They have very comprehensively presented their work, which is very related to the present situations we need for the society, different biological ways to protect our earth as well as environment. Thank you so much, and thank you, presenters, and wish you good luck for the competitive result, which Madam Srinivasan will prepare and send to Professor Raman. Thank you so much, and thank you all.
Thank you, ma'am.
Thank you, ma'am. God bless you. Over to Dr. Avneet. Dr. Avneet.
Thank you very much, ma'am. So we are extremely thankful to Dr. Geetika for taking the pains for this long session. It's really a commendable job. And even more thankful to Dr. S. Bhuvaneshwari for the devotion which she has given to the evaluation part. It will really encourage our young scientists to do better and better in their careers. So we are extremely thankful to the chairperson, to the co-chairperson is in his absence for their cooperation. Thank you very much, ma'am.
Can we continue with if the chairpersons and the co-chairperson moderator are available?
I think, sir, we have Dr. Rodriguez is with us.
Okay.
Dr. Rodriguez?
Yeah, I'm here.
And we have Dr. Paneer Selvam.
Yes, sir. Yes, sir. Yes.
So, sir,
Good evening,
what do you
sir.
say? Shall we take some break or we should start immediately?
Okay, good. I think moderator Manish Kapoor is there.
Sir, he's having some break. Let me ask him, and then we can continue. Dr. Rodriguez will be able to continue, or shall we take some short break?
I'm able to. I can continue. No problem.
Okay, just a moment. I'll just ask Dr. Manish to join.
Yes.
Okay, good. Thank you, Dr. Pandit Selvam, for joining as a co-chairperson, and Professor Rodriguez, the new president of MSI.
Yes. Happy congratulations, sir.
Yeah.
Rodriguez. Since we are lagging behind by some 15 minutes or so, yes. Should we reduce the time or it should be 15 minutes only?
First check how many people are there.
Yes. There are totally about 10 oral presentations are there in this session.
Check whether all are there.
Dr. Rodriguez,
yes.
The presenter, Dr. Vandana Ghormade, she has just sent an email shared that she won't be able to present her talk.
Okay.
Yeah,
Dr. Vandana won't be able to present. We will have 9 presentations.
Then I think it should be okay. Yes.
Dr. Manish is there. Let me just.
I think if you wish, you can reduce if the chairpersons are chairperson and co-chairperson.
13 minutes.
15 minutes. Okay, it is finally it will be validatory only. No. There is a break. Okay.
Friends, we have with us Dr. Manish.
Good afternoon, sir.
Good afternoon, sir. Over to you, Dr. Manish.
Thank you, Dr. Avni. So we are there with the last session of our conference, the 11th session, which is for oral presentations for middle-aged faculties and scientists. And the chairpersons of this session are Dr. Bernard Rodriguez and Dr. Panir Selvam.
Yes, sir. Yes, sir.
I hope
Yes, sir.
both of you are there. Welcome, sir.
Yeah. Yes, sir. Thank you. Good afternoon, sir. Yes.
Good afternoon. Good afternoon, sir.
Good afternoon, sir.
Let me introduce our chairperson and the co-chairperson. Our chairperson is Dr. Rodriguez, who is a senior professor in the Department of Botany, University of Goa. He's also director of the Directorate of the Internship, Incubation and Industry Partnership. And he has worked in most of the university committees from Board of Studies to Executive Council members. He has 29 years of vast experience and 31 years of research experience. Completed more than 12 research projects funded by various funding agencies. He has attended many national, international conferences, workshops, seminars, delivered plenary lectures and field lectures. He has to his credit 145 research papers in national and international journals, 5 books, and has guided 12 PhD scholars. He's a recipient of several awards for best paper presentations and medals. Twice recipient of Young Scientist Award from Department of Science and Technology, New Delhi, and Excellence Award from Tanzanian Partnership Network Foundation. He was awarded Professor Binde Memorial Award by Indian Society of Mycologists and Plant Pathologists. He's the Best Teacher awardee by the state government of Goa, and awarded by Dr. Agnihotru Memorial Award by Mycological Society of India. He is also a fellow of Indian Society of Mycology and Plant Pathology, fellow of the International Scientist Unique Researchers Year Association. He is member of the editorial board of many journals. So with this, I welcome you, sir.
Thank you. Thank you.
Our co-chairperson, Dr. Panir Selvam. He is associate professor and head of the botany department of microbiology at Shri Pushpam College, Pundi, Tamil Nadu. He is a PhD from Bharati Darshan University. He has 36 years of experience, published more than 320 research papers and 12 books. His area of specialization is mycology and plant pathology. He has deposited more than 75 gene sequences in the gene bank. He has visited many countries, which include Singapore, Malaysia, Bangkok, and Nepal. He has several projects funded by UGC, DST, CSIR. He has 27 PhD scholars and 60 MPhil students guided. He has organized several seminars and conferences, awarded INSA Visiting Fellow and Academic Excellence Award, and held several positions in editorial board, chair, several training programs in the field of edible mushroom cultivation. Welcome you, sir.
Thank you, Professor. Thank you for your nice introduction. Thank you, Professor.
Dr. Manish, before start, 1 minute. Not 1 minute, half minute, I can take. Dr. Manish.
Sure, sir, sure.
I request the chairperson and co-chairperson to consider this as one of the award. It will be considered for 2022. One of the award for the middle-aged group, and kindly give one top person. You can evaluate for 100 marks. And you can take, I think you can give 12 minutes each, and 3 minutes questioning, like that we can do that. Okay. And since it is going to be considered for an award, kindly stick to the timing, please.
Sure.
Thank you, Professor. Thank you. Definitely, we will follow.
Yes.
So over to Dr. Rodriguez and Phylogenetic, sir.
Yeah, thank you for the nice introduction. A very good afternoon to one and all, respected senior mycologists, office bearers of MSI, convener of this conference and head of the Botany Department of Punjabi University, Patiala, Professor Manruchi Kaur, other members of the organizing committee, and my young student friends. I deem it a great privilege to chair this session 11, wherein there are 9 oral presentations from middle-aged mycologists. I also welcome the co-chair for this session, Professor A. Paneerselvam. Before we begin with the presentations, I would like to remind the presenters that as per the directions of the organizers, you have been, and we have a time limit of 15 minutes at your disposal. And then at the end of the 12 minutes, in 12 minutes, you have to finish your presentation, and the remaining 3 minutes will be for the question answers. So at the end of the 10th minute, we will ring a bell, or we'll remind you that you have completed 10 minutes, and then you'll be left with only 2 minutes to complete your presentation. See that you stick to the time, so that we have to finish this particular session by at least, before 4:30. So to begin with, I call upon C.K. Pradeep from TBGRI, Thiruvananthapuram, Kerala, to present his paper entitled The Fungal Dimension of Biodiversity Conservation. C.K. Pradeep, please.
Yes, sir. Am I audible?
Yes, very much.
Share your screen.
Yes, sir. Yes, sir. Is it visible, sir?
Yeah, you can go ahead, Dr. Pradeep. It's visible.
Yes, sir.
Yes, visible.
Good, respected chair and co-chair, distinguished mycologists, fellow mycologists, friends, very good afternoon to all. I'll be presenting on a topic on fungal dimension of biodiversity conservation. This is in general with related to my work that I was conducted in a particular district of Kerala. As we all know that biodiversity is the biological variation on Earth, and it become a very hot topic in science as well as politics after the Rio Convention on Biological Diversity. In all this, most of our attention has been directed towards the role of animals and plants on biodiversity conservation. We may neglect other groups of which is much more important or equally important to these groups, like fungi. We have already not much attention has been given to this group while we are discussing the conservation of in general. Coming to the So coming to the significance of fungal biodiversity, since fungi are the second most species rich organism after insects, and their current estimates indicates that we all know that it ranges from 3 to 5.1 million. But what we know, or what we know only about 7 to 10% of this estimation. And when we consider the key roles that
the slide is not moving, Pradeep.
Yes.
Yes, sir. This is second slide only, sir.
No, you are still in your first slide.
First slide only, yeah. Yes, yeah, no.
No, it is not.
Sir, can you? Yeah, now it's visible.
Sir, there is some issues here when you are sharing the screen.
It's fine now, sir. It's fine now.
Yes, yes. Go ahead.
Okay. Okay. So when we consider the key roles that these groups in ecosystem functioning, from decomposers to these mutualists and pathogens, their role is very important. And so the conservation, the diversity and conservation of this group is of equally important as with respect to other organisms. So are they worth to be considered in conservation? When we consider all these things, it is worth it to be considered, but whether they are being considered in that or whether they got the same respects with the other groups of organisms in conservation. If we look into the IUCN Global Red List of species that is accounted in that list, fungi, there are only very few fungal species are listed. And I will be discussing this conservation with respect to the study that I have conducted, and we'll be discussing on the diversity with respect to the terrestrial, lignicolous, symbiotic, and coprophilous habitats, and the conservation with respect to that habitat. Coming to the first, that is the terrestrial habitat. What we have observed is majority, almost 50% of the species or 50% of the groups that belongs to this saprophytic terrestrial habitat. And though they in this habitat, we can see that there is not much preference of soil is there, but in some cases, some species or some genera like Hygrosebia and Neosebia found to be prefer some special habitat or specific soil conditions. And in such cases, we have to think of how these species, that is those coming preferring this terrestrial habitat or the soil conditions, what are the strategies to be considered for their conservation. That is very important. In that terrestrial habitat, we can see that in our study that some genera like Hygrosebia and Agaricus are found to prefer those habitats where the living bamboo stands are there. So in such habitats, we can see that they are nutrient deficient when they are compared to other forest habitats. The problem is that what we have observed in our study is that usually the forest department in Kerala auction these bamboos regularly, and a lot of activities are accomplished with this cutting and removal of these bamboos, which is very detrimental to this group of mushrooms. And another threat to the terrestrial species are the littering by the tourists, whether if it is those areas are coming in the tourist places or forests that is of tourist importance, they will litter these areas, which is also very detrimental to these groups of mushrooms. And also the those areas where these highways, national highways, interstate highways, that also we have observed that these are also very these areas are also being littered by these tourists and other people passing through these roads. And the another group is the Lignicolous, that is wood-dwelling species, where in our study that majority are wood-inhabiting species are mainly composed of those genera, that is Pleurotus, Lentinus, etc. And these species, as you know, that many of them are edible. So locals and these tribals will collect these during the season. And in this case, even though these lignicolous species also shows some specificity, and the majority are not showing any specificity for any type of substrate. And these are exceptions to that. That is, these are some of the specific species that dwell in specific substrate, and that is Lentinus. These are all associated with the bamboos, and one that is Pepidotus is a palmarin that is restricted to the dead and decaying fronts of palms. So, except for these such groups, there is not such specificity can be observed. And coming to the conservation of this group, the major threat is I've already, many of them are edible, and they're also important that they constitute a secondary saprophytic group in the forest ecosystem, which is very important as it forms the play a vital role in the decomposition of litter. When we consider the tropical forests, the litter turnout is huge, and this it signifies its importance in the in such a forest. The major threat to this group is the indiscriminate collecting. That is, many of them are edible. So this indiscriminate collecting is a serious challenge to its conservation, and we have to check this by appropriate laws or other means. Another group is the coprophilous group, that is dung inhabiting fungi. Of this, as you know, that dung, we have different types of dung in areas of forest areas, but in all this, we can have many fungi, that is micro to macro fungi. But with respect to the macrofungi, it is since this dung is ephemeral, it cannot support this mushrooms or large fruit-bodied macrofungi.
What happened?
Sir, I think there is some network problem.
I think so. He has some network problem. Yes.
Maybe.
Pradeep?
No, I think he is left.
I think he
disconnected.
Yes.
Yeah.
This got disconnected. Yes.
Should we go to the next presenter?
Is he completed around 9 minutes.
Yes, yes.
Yeah.
3 or 4 minutes.
I started. Yes, yes, yes.
He's
yes.
back. He's back. He's back.
Pradeep, go ahead.
Mr. Pradeep?
Pradeep, are you there?
I think Dr. Pradeep joined, but his voice.
Mr. Pradeep?
Sir.
Yes, can you able to hear our voice?
Yes, yes, I can hear, sir.
Yes, we don't hear your voice.
No, now it's okay. I think you can continue.
Okay, sir. So conservation of this dung inhabiting fungi is depends on the availability of the dung in that area. That is actually directly proportional to the population of wild elephants in this
We have 2 more minutes.
area. Okay, sir.
Mr. Pradeep?
Yeah, sir. It's not, there is some issues.
Conclusions.
Dr. Pradeep, you can continue.
Sir, there is some issues with the internet connection. That's all. So
slide
another,
has changed.
Yeah, another group is the ectomycorrhizal group, and which is also a very particular group and having a high degree of adaptation to a set of ecological conditions. So coming to the conservation and survival of this group, we have to be very particular, because the survival of this group is particularly dependent on the tree partner, and any forest practices such as selection, felling, making, firebelt, or trench making, all affect seriously the existence or the survival of these species. So we have to have, and also some of them are also good edibles, and over harvesting of these species will challenge its conservation. Another group is the termite mushrooms. That's also most of them are edible, and the most serious threat to this group is also over harvesting. And what are the challenges in conservation aspects to this group? Mainly because of its we are not discussing due to lack of knowledge on this group. That is less diversity, knowledge on this diversity, and we are not able to discuss, or we cannot able to bring this group to the global conservation discussions. And in conservation is challenging aspects. We know other things are the its habitat. We don't know much about its habitat requirements, reproductive biology, how these grow, and etc. All these are very crucial while we are proposing this conservation. And this conservation recommendation is based on our study, and these are the major things, conservation points that I found to be I want to recommend with respect to my study. That is, that is the most important, create awareness among locals, tribal people, forest officials. That is also very important. They don't know anything about the conservation aspects of this group. They are only know about the above ground plants and higher organisms. So there should be awareness among, by any means, by either seminars, classes, or etc. And there is another important thing is we have to identify mycologically important areas and mark these areas to protect these groups, because we cannot see these groups in all time. So during this season, we have to make these areas where are the most important regions that we can mark these areas. So that also create awareness. And this also helpful for tourists and other people, and those are entering into the forest. So this sort of activities are, this sort of marking is very essential for their conservation. And we have to check the over exploitation. They don't know, the people don't know, either the locals or the tribals don't know the importance, and they will have indiscriminate, even they disturb the habitat. So they have to be also been, that has also been checked by appropriate means. And this wild elephant population is also have to be maintained in if we want to conserve these those coprophilous species. That's all. Thank you.
Yeah, thank you, Dr. Pradeep. Questions?
I have one question.
Yes.
I'm Professor Bhatt from Goa.
Yes, sir.
Pradeep, how about conserving them by culturing?
Yes, sir. That is there, sir.
because that I think that is the prime, because
yes,
there
sir,
is a possibility of your
yes.
habitat destruction, all
Sure,
those.
sir.
So,
Yeah. Like we have the generally we have the ex situ and in situ conservation. So in that we have to make that culture, sir.
no, I think your recommendation should have that culturing
Yes,
also,
yes, yes, sir.
isolation.
That is a
Okay.
common one, the ex situ and in situ that I just skipped that, sir.
It's okay. Fine. Thank you.
Yeah, thank you. I call upon the next presenter, Dr. Parasan Singh from Agarkar Research Institute, Pune. He'll be presenting on isolation and qualitative analysis of phosphate solubilizing fungi and eco-friendly approach.
Yeah,
Parasan, please.
Hello.
Yeah, I can hear you, Dr. Parasan.
Yeah, good afternoon, sir. Rodrigue, sir.
Good afternoon. Good afternoon. Please go ahead, sir.
Yeah, so respected chairperson and co-chairperson, Dr. Rodrigue, sir, Vrinda, sir, and Panir Selvam, sir. So with the help of a few slides, I would like to present on the topic isolation and qualitative analysis of phosphate solubilizing fungi and eco-friendly approach. So this is the topic what I have selected. And this objectives, this is given to the one of my student that dissertation work. This is the 4 months work only. So this objective is collection of samples from different regions of Maharashtra for the analysis of this phosphate solubilization purpose. Isolation and identification of fungi, whatever the things we have isolated and collected. So screening of isolated fungi for their phosphate solubilizing potential. And after this, qualitative characterization of potential phosphate solubilizing fungi. And after collection and isolations, their in vitro conservation and initiation of phosphate solubilizing fungi in our germ plant bank, NFCCI, that is National Fungal Culture Collection of India. So in general, this phosphorus is an essential micronutrient next to nitrogen, playing a key role in the growth and yield of crop plants. An adequate supply of phosphorus is the early stage of the plant growth is important for their development of primordia for the reproductive parts of the plants and fruiting. In soil, phosphorus is abundant in both organic and inorganic forms, but it is available is restricted because their precipitated forms cannot be absorbed by the plants. Means they are the inert. Soil contains organic phosphorus that can be used by plants only if it is mineralized. The phosphorus content in average soil is about 0.05, but only 0.1% of the total phosphorus is available to plant because of the poor solubility and it fixed in the soil. 75 to 90% of added phosphorus is precipitated by metal carbon complexes and soon becomes the fix in soil. And now day by day, excessive use of fertilizers and chemical pesticides also affect the soil health, and such environmental concerns have led to the search for sustainable way of phosphate nutrition to the crops. Therefore, in this concern, the microorganisms like fungi can play an important role as phosphate solubilizing agent to maintain the soil health eco-friendly. So the significance of phosphorus, we can see this diagram. So adequate supply of phosphorus in early state of plant growth is important for laying down the primordial development of plants and their fruiting, flowering. And the judicial low cost of soil fertility can be improved by this using phosphate solubilizing fungi, which are eco-friendly, and the use of as a biofertilizer is an economical and does not damage the ecosystems because of this phosphate solubilizing fungi are the eco-friendly, we can treat them. So during the course of study, we have collected different soil samples, this rhizosporic soil of tomato and wheat crop and other soil samples collected from the various places like Maharashtra and ARI and other places. So these are the list of given samples what we have collected, and we have given the sample code for the each and every soil samples. And this for the isolation of these fungi, what we have collected the samples for the phosphate solubilizing purpose. So for the isolation purpose, we have used this general purpose potato dextrose agar media, and this is the composition. And for the screening of qualitative analysis of phosphate solubilizing purpose, we have used Pykowski's media, and this is the composition of this media of this Pykowski agar media. And this is the model what we have isolated, the isolation and purification of fungi from the different samples. So we can see this is the mixed colony, and with the help of the direct and indirect isolation. So this is the indirect isolation of the fungi. We have done the serial dilution of this fungi in the distilled water, and different concentration of this solution has been spreaded on the plate. So we can see this is the mixed colony, and from the mixed colony we have separated this colony, and this is the pure colony of this Aspergillus. This is one of the example of this how we are going to isolate and purify this organism, and after purification we can identify this fungi this morphologically, as well as by using other tools like molecular. So this is the what we have isolated for the purpose of phosphate solubilizing fungi. So this is the Aspergillus niger, this architecture, this is the penicillium, this is the cladosporium, this is the complete vesicle of this Aspergillus niger. This is the one penicillium which looks like that vesicles-like structure, but it is a penicillium. This is the trichoderma, and this is the another traverticillative penicilli what we have isolated. So like this we have isolated 50 fungi, more than 50 fungi for the purpose. So now I will come to the main point, this qualitative in vitro screening of the phosphate. So this is the culture in vitro. So what we have done, we have the one set of the cultures in the single point inoculation as a control, and one set of the cultures we are putting for the single point inoculation on the Phylogenetic media, and we can see this clear zone which indicates that this fungus is giving this phosphate solubilizing zone around the colony. And the sum out of the means total we have isolated 50, more than 50 fungi. So this is the sum of the list of the isolated fungi for the purpose of such phosphate solubilizing purpose. So this is the Niger, idulens, Aspergillus stellatus, flavus, Aspergillus oryzae, Periconia atra, Percuricillium liliacinum, Aspergillus stellatus, Aureobasidium pullulans, which is the black yeast of this oil isolation, Cladosporium cladosporides, Penicillium citrinum, Penicillium sclerotiorum, which is producing sclerotia apart from the xenomorph that fungus, and this is the Penicillium purpurogenum. So like this we have, I'm not giving here this complete list of this fungus, but some parts I have given here in this slide. So this is the protocol for the screening of phosphate using this tricalcium phosphate by this Phylogenetic medium. So this is the schematic diagram of this media what we have used, and for the purpose of the phosphate solubilizations we have used this formula, this calculation of phosphate solubilizing fungi solubilization index. That solubilization index we have calculated like this colony diameter, whatever we have put in the single center on the plate, and plus we have used that clearing zone, whatever the clearing zone around the colony, and divided by the colony diameter, so it will give that solubilization index in terms of measurement and reduction. So this is what we have isolated during the 3-4 months of this period. So these are the pure fungal colony, and the front of this we can see that how the diversity looks like and how this culture macromorphology looks like. So before the putting in experiments, we are supposed to do this colony morphology, and almost all morphological and other analysis we have to do that. So this is the same things what we have isolated in continuation of this work of this 50 fungi. So this is the colony morphology what we have given. And of course, whatever fungi we have isolated, so we have put the screening for the phosphate solubilization. So we can look this colony, this is the penicillium, and this is the fungi which is giving this zone around this colony. So here we can see that clear zone around the colony. Here this fungus is also giving. Here we very apparently we can see this clearance of the zone of this around the colony, and some of them they are not giving this zone of colony. So we can think that the fungi which is not going that zone of that clearance, so they are not phosphate solubilizing in terms of this qualitative analysis. So these are the different fungi what we have isolated and what we have put in the Phylogenetic media for phosphate solubilization analysis.
So you have 2 minutes to conclude.
Yeah, sorry.
You have 2 minutes to conclude, sir.
Yeah. So this is the same things what we have given this. Out of 50 fungi, 38 fungi are giving this phosphate solubilization. All are giving this clear zone around this colony. So we can see by this. So this is the same things. Almost all this 38 fungi they are giving this phosphate solubilization zone around the colony. And this phosphate solubilization index we have put the triplicates of this. Average of this 3 colony, we have taken the diameter zone of this average of 3 colony. And solubilization index is, we can see this here, this more than 2.8 to 1.5, and other than this, something less than 1, even we have got this phosphate ability. So these are the fungi we have given this code, and the maximum fungi they are giving this Aspergillus niger and Penicillium, and even some Cladosporiums we got in results. So in summary, we can say that out of 50 fungal cultures, 38 isolates have given this phosphate solubilization potential on Pycovaskia jagar media. This suggests that these fungi have the organic phosphate solubilizing ability in the media, and this solubilization index in the fungal isolates range from what we have done in our experiment. So we have got the solubilization index that 1.02 to 2.18, what I have given in this table already. So among the 38 fungi, 22 isolates have shown the very good phosphate solubilizing ability, that is their solubilization index is more than 1.5. And mostly whatever the fungi they have given their phosphate solubilization around the colony, that zone of clearance, so maximum they are the genus of this Aspergillus, Penicillium, Cladosporium, and some others. So I can say that among the screened fungal isolates, Aspergillus penicillium was found most efficient with a solubilization index of 2.8. This suggests that these potential fungi can be useful as an eco-friendly biofertilizer in agriculture to improve the soil health and crop productivity, because the fungi are having the immense potential, so that we can use this as a phosphate solubilizing eco-friendly agent. So in conclusion, based on the observation and results of this project study, we finally conclude that isolated fungal cultures have promising phosphate solubilization potential. Therefore, they can be used as eco-friendly, safe biofertilizer to increase the soluble phosphate content in the soil, which ultimately improve the soil health and crop yields.
Yeah.
I thought in the whatever fungi we have isolated, so that's like this we are conserving the fungi among the tube, and apart from this Aspergilli and Penicilli, the best source is the we are maintaining on the cereal grains, and apart from the cereal grains we are maintaining the cultures on the liquid nitrogen, and apart from this we are lyophilizing this culture also for the future study. So thanks.
Thank you, Dr. Singh. Any questions from the audience? Any questions? If there are no questions, thank you, Dr. Singh, for a nice presentation. And next presenter would be Dr. T.S. Murli from Manipal Academy of Higher Education, Manipal, and he'll be talking on epigenetic modifications to improve metabolite production in endophyte cultures. Dr. Murli?
Yes, sir. Can you hear me, sir?
Yes, yes.
Yes, yes.
Is the screen visible, sir?
Yes, it's visible. You can start. Continue.
I'll put it in the slide share. Yeah. Good afternoon to all the mycologists of India. Today I'm going to present on the topic epigenetic modification to improve metabolite production in using some endophytic cultures. So the major theme is fungal physiology, biotechnology, and applications. Coming to why we did this, we all know that plant and fungi, in fact, plants are considered to be the major organisms from which most of the secondary metabolites or new molecular entities are used against bacteria, as well as for many industrial and pharmaceutical applications. But there are several issues associated with using plants as the sources of new molecular entities. One of the major issues is the growth rate itself, and also the exploitation, overexploitation can lead to extinction. So that is where fungi and bacteria are considered to be better, because they can be grown much faster and in using very efficiently in a small space. And they can be allowed to produce the metabolites for a longer period of time. So endophytes, one such group of organisms which are seen symbiotically with the host plants, are also known to produce lots of these secondary metabolites. And that is where this importance of these endophytes are realized. So for example, if you see, The knowledge of traditional medicine is known since very ancient times. In India, we have this Ayurveda system where several medicinal plants have been used as drugs, and several important molecules from these plants have been used to treat different diseases. However, even though they are important treasure houses of phytochemicals, the overexploitation, as I told, can lead to extinction of rare species, especially if the natural habitat is very small. That is where one of the possible alternative is to look for endophytes, which can produce, but again, not all endophytes can produce this. Still, studies are going on why certain endophytes are able to produce these plant-derived metabolites, but some endophytes can produce these host-derived secondary metabolites, and if we can identify those, that can be used as an alternative for producing these metabolites. But endophytes, they also have their own drawback. So when you keep culturing these endophytes, they usually lead to attenuation of the metabolite production in exenic culture systems. What I mean by attenuation is that levels of metabolites come down drastically when you keep culturing them. So we know that to keep the metabolite production, we have to keep culturing the fungus. And when we do this, usually this is what happens, attenuation of the metabolite production happens. So how to overcome this? So one of the hypotheses was that it is due to methylation, and probably if we use epigenetic modifiers, we can overcome that attenuation and allow the continuous expression of these metabolite clusters. So there are many reasons why we are looking at epigenetic modification. So before going on to that, I would like to say what are the factors which are responsible for attenuation. Again, these are all hypotheses. We still don't know what exactly is the reason. In many cases, it is the absence of the optimum environment. So when the plant and the endophyte are present together, probably they get the optimum environment and they are able to produce the metabolites. But once you culture them and you grow them in a nutritive rich condition, they are not able to produce the metabolite. Or there might be the absence of some precursors from the host because this is exenic culture. Or it might be that several endophytes within the host come together and they are able to elicit this particular metabolite production. And in the absence of these cultures, other endophytes, the endophytes are not able to produce. It might be also due to selection pressure, or it might be due to some cross-activation signals. The host is absolutely essential for the endophyte to produce the metabolite, or it can be also due to the silencing of the acquired biosynthetic gene clusters. So this is something that we looked at because all the other parameters are very difficult to control, while the silencing aspect, the methylation itself, can be probably overcome by using certain treatments. So what are the strategies? So there are several strategies which we can use for using endophytes or producing metabolites at industrial level. One, we can take the gene and heterologously express it in some E. coli or PKA or some other systems and able to produce these metabolites. But usually the metabolite clusters are not single genes, they are clusters, and therefore it's very difficult to clone a huge gene fragment. So that is a problem with these secondary metabolites. Co-cultivation with other endophytes, yes, but it is a hit or miss approach. Use of genetic and molecular biology tools, yes, it is still picking up. We still have to do a lot more. Addition of precursors in tissue cultures, again, it leads to a lot of time, and again, it doesn't increase the yield too much. Whole genome sequencing and identification of gene clusters. This is one of the steps which will help us in identifying the gene clusters, but the optimal use is by using epigenetic molecules. So what are these epigenetic modifiers? So I will come to that. Before that, just to give a brief introduction of what we are looking at, we were looking at Gloriosa superba, which is the host plant, which we all know produces colchicine. So both colchicum and Gloriosa superba are known to produce this colchicine, which is, these two plants are used in treatment against gout and the Mediterranean fever. And the reason why they are not used that much is because of the toxicity of the metabolites. But recently, FDA has given approval for treatment with these colchicine, and now it is also available in the trade name called. So colchicine, as I have explained, is a very important metabolite, and that is where we were looking at the production of colchicine from fungal culture. So what are the major questions that we asked? whether what is the importance of attenuation in endophyte producing important metabolite, and whether methylation plays any role in the reduction of the metabolite synthesis, and if so, how we can overcome this, and what exactly is the reason behind that production of these metabolites by the fungus. So what we did is that we isolated some fungal endophytes from the host plant Gloriosa superba and screened them for production of colchicine. We found one particular endophytic culture which was able to produce this metabolite. So then we picked up this isolate and we did the screening and characterized it both molecularly as well as for functional ability to work like colchicine. And then we studied the attenuation and reactivation using the epigenetic modifiers. And the final approach we have done genome. So I'll explain all this. So first part, we isolated the fungus and then we were able to identify by sequencing different genes we have sequenced, not just ITS. So we identified it as diapothecasia. We also did morphological identification, alpha and beta conidia we were able to see. And based on this, we identified as diapothecasia. Then we went on to do TLC and then identified that this fungus was able to produce colchicine. We went on to do HPLC and showed that the crude extract had a little bit of colchicine production. And then we purified it, and this purified product, when we did for LCMS, we could see that it had the same signal as the pure colchicine. So on the left-hand side, you have on the right-hand side, sorry, you have the LCMS-MS profile of colchicine, while this is the fungal extract, which shows the similar peaks. And we could say that the fungal extract had colchicine. And then we went ahead and did NMR spectrum of this fungal extract, and we could see the presence of colchicine even with the NMR spectrum. The next thing what we wanted to do was a functional characterization of the colchicine metabolite. So we took the microtubule polymerization assay and did this assay, and we showed that the colchicine worked very similar to Taxol. Even though the Taxol and colchicine are both microtubule inhibitors, their functioning is slightly different. Taxol works by inhibiting depolymerization, while colchicine inhibits polymerization, but both of them lead to the prevention of the cell to progress to the next stage in cell division. So we showed that our fungal extract, also the blue line here, did the same thing as colchicine, and therefore indicating that the fungal extract had colchicine. The next part was to study the attenuation. So here you can see we subcultured it repeatedly up to 10 generations, and we found that the yield reduced drastically when we studied the production of colchicine. And after this, we treated it with azacitidine. There are different ways by which epigenetic modifiers can work. The epigenetic actually means that other than the changes in the sequence, other ways by which it can inhibit or it can affect the organism, and that is where the treatment with demethylating agents like 5-azacitidine comes into picture. And when we treated it with 5-azacitidine, we showed that again with increasing concentration, we could get lots and lots of the metabolites. Actually, it is still a work in progress because we haven't attained the level of production that we were looking at. But interestingly, we could show that the levels of azacitidine treatment, the levels of azacitidine increase the production of colchicine as well.
Dr. Murli, you have 2 minutes more.
Yes, yes. So we went ahead and did a global methylation analysis. There we have treated the fungal cultures, the genomic DNA, to see whether actually it is due to methylation. HSP1, HPA2 and MSP1 are two enzymes which are methylation sensitive and methylation resistant enzymes. And this experiment showed us that very clearly that it is only the methylation which was responsible for the attenuation, because the cultures when treated with azacitidine showed less amount of methylation. And we also did the global methylation analysis for different cultures. When treated with the azacitidine, the levels of the global methylation came down, indicating again that that is the reason behind the increased levels of colchicine production. And again, when we studied the yield in these cultures, again, the yield of colchicine was much higher. So finally, we did the whole genome sequencing. Again, this is work in progress. What we have now shown is that I will skip this slide, but what we have shown is that there are several enzymes involved in the colchicine pathway, and we could identify several enzymes which are involved in this pathway to be present in the genome of the fungal culture. And this clearly indicates that there is some sort of horizontal gene transfer from the plant to the fungus, or the other way around. We still don't know how it works, but we have now constructed the colchicine pathway, which might be working in fungus, because we could identify certain of these intermediates in our LCMS analysis. So this is something that we have built. So to summarize the whole thing, what we have shown is that the plants and fungi can produce certain novel biomolecules or host-derived molecules, and due to symbiotic evolution, both of them could produce these biosynthetic pathways. And by treating them with these demethylating agents, we can allow the continuous production of these secondary metabolites even in the attenuated culture. So this is the summary of the whole study. And we published this recently in Journal of Biotechnology, and these are the references. And I would like to thank my guide, Dr. T.S. Suryanarayanan, the organizers and the MSI for giving me an opportunity to present, my director and ISER Mohali, Dr. Kavita Dorai, who was a collaborator in this work, and all my students, and Mahe and the CSAR for funding this work. Thank you, and I'll be happy to answer any questions.
Yeah, it's open for questions. Well, if there are no questions, then we'll move on to the next presentation. But before that, let me thank Dr. Murli for a very nice presentation. Thank you.
Thank you, sir.
We move on to the next presentation by Samanjana Katua from Krishnagar Government College, Nadia, West Bengal. So we'll be talking on immune boosting effect of traditionally reprised wild edible mushrooms.
Good afternoon, sir. Am I
Good
my
afternoon.
slides?
Yeah.
Okay, thank you. Thank you, sir.
Continue.
Good afternoon, respected chair and co-chair, dignitaries, my colleagues, dear students, ladies and gentlemen. Today, I'm going to tell a story about two wild edible mushrooms from West Bengal. The recent pandemic situation let us understand one thing very clearly, that if our immune system remains strong, it could protect our health from a number of diseases where there is no medication. Besides, immune boosting becomes very essential for those who are suffering from cancer, AIDS, malnutrition, autoimmune diseases, or elder people. Research in this area is very new, and there is very less amount of research has been done, although researchers have shown that polysaccharides from mushrooms, especially beta-glucan, has ability to boost our immunity. The reason behind this is that it can activate the immune system components, for example, macrophages, who act like soldiers in a country. Recently, only few mushroom polysaccharides are in clinical state as well as in industrial application. So we wanted to know the immune boosting effect of wild edible mushrooms from West Bengal, being a phytogeographically unique state extending from Himalayas to Bay of Bengal. There is another important zone in West Bengal that is called lateritic area, which supports forest of Shorea robusta, facilitating natural growth of a number of ectomycorrhizal specimens. Besides, it is the home of a number of tribal people who reside in the lateritic area, forest area, and spend their whole lifetime in that area. That is why they are dependent on the forest products for livelihood. Fortuitously, during our recent expedition, we were able to collect two morphologically unique specimens from the lateritic area of West Bengal with the help of local tribal people. One specimen with red-colored pileas is colloquially known as Murgi Chatu. Murgi means hen and Chatu means mushroom. As the color resembles comb of hen, that is why it is colloquially known as Murgi Chatu. And the other mushroom with orangish yellow colored pillias is colloquially known as jhal chatu because of taste. It has bitter taste. That is why local people call it as jhal chatu. Local people also let us know that the mushrooms they are traditionally consuming these mushrooms as health promoting food that can protect their health from seasonal hazards. Although they remain abundant from city markets because the city people doesn't know about the culture as well as the edibility of this mushroom, so they remain neglected. This information motivated us to carry thorough investigation on these mushrooms, in which identification was the first step. After thorough characterization based on morphological characterization, anatomical features, DNA barcoding, and phylogenetic position, the Murgi chatu appeared as a novel mushroom, and we named it as Rusula alato-reticula and published in a journal. On the other hand, Jhal chatu appeared as Rusula senesis based on these characterizations, but our work for the first time reported this mushroom from West Bengal, as well as reported the edibility of this mushroom as a tribal food for the first time. And all these informations are published in a previous article. Then we wanted to characterize these mushrooms chemically as well as therapeutically. For that, a conventional hot water process was followed to isolate crude polysaccharide from these mushrooms. At first, we wanted to know the molecular composition of these fractions. For that, spectrophotometry was performed. In short, it can be said that both the fractions were composed mainly of beta-glucan. To verify this output, then we performed FTIR, HPTLC, and GCMS. The chromatographic profiling also showed that presence of glucose in dominant amount along with other monomers like galactose, mannose, etc. So it suggested that both the fractions were composed of beta-glucan mainly. Then we performed Congo red assay, and the result showed that the carbohydrate backbone was organized in triple helical conformation. So in short, it can be said that the hot water extracted polysaccharides from R. alato-reticula and R. senesis were composed mainly of beta-glucan, where the carbohydrate backbone was organized in triple helical conformation, which is known as the most stable form. To determine the bioactivity, we purchased and cultured murine macrophage raw 264.7 cell in our laboratory. At first, we wanted to know whether the polysaccharide fractions has any effect on the macrophage cell or not. For that, WST assay was performed. Result showed that after 24-hour incubation, the macrophage cell number was increased in presence of polysaccharide fractions, and the effect was more prominent when the incubation was extended for 48 hours. So this result, or this WST result, showed for the first time that the polysaccharide fractions might have immune stimulatory activity. To verify the output, we then performed neutral red assay. It is to determine the phagocytic activity of macrophage. As we know, one of the most prominent activity of macrophage is engulfment or phagocytosis. The result showed that the phagocytic activity of macrophage in exposure of the polysaccharides increased in a time-dependent manner significantly. We verified the output using yeast as a food for the macrophage, and the result showed that in negative control, the macrophages are engulfing less amount of yeast, but when they are exposed to the polysaccharide, the engulfment is increasing. Then we verified or observed the morphology of macrophage cells under inverted microscope. In case of negative control, when they are unstimulated condition, the cells are smaller in size with less number of production of pseudopodia from the surface. While when they were incubated, macrophages were incubated with the polysaccharides, the cells become larger in shape with high number of production of pseudopods or lamellipodia or philopodia from the surface. It signifies that the macrophage is in stimulated condition or they are ready for engulfment, as this philopodia and lamellipodia helps macrophages to engulf. We know that when the macrophages engulf a pathogen, it produces nitric oxide and reactive oxygen species as a weapon to kill the pathogen. So production of these molecules signify that the macrophage is in stimulated condition. Our results showed that when the polysaccharides were when the macrophages were incubated with polysaccharides, production of NO and ROS synthesis increased in a significant way. So all these methods, all these results showed that the polysaccharides have immune stimulatory activity, although the mechanism of action was not still clear. For that, we incubated the macrophage cells with the polysaccharides for 24 hours. Then total RNA was isolated, cDNA was performed, and RT-PCR was done. Gel pictures showed that the treatment increased mRNA synthesis of TLR2, TLR4, NF-kappa B, COX2, INOS, TNF-alpha, I-kappa B alpha, and IFN gamma in a significant way. And to our surprise, we found that the effect of the polysaccharides was even better than the positive control LPS or lipopolysaccharides. And the immune stimulatory activity or the expression of these genes was found in both the fractions, that is R allotereticular and R senesis. So in short, it can be said that it is known that beta-glucan act as ligand for TLR2 and TLR4 receptors. As these polysaccharides has beta-glucan in significant amount, so might be they are acting as ligand for TLR2 and TLR4. After binding to the receptor, it can activate the downstream signaling pathway, resulting in augmentation of ROS production. When ROS is in excess amount, it activates NF-kappa B. NF-kappa B is a transcription factor. So when it is getting activated, it enters into the nucleus, binds to the promoter region of a number of genes, resulting in production of pro-inflammatory cytokines and chemokines. All these mediators are known not only to activate macrophage, but also the T lymphocyte, or in other words, adaptive immune system. And the pathway has been published in our previous article. So our research introduced a mushroom, that is R. allotereticulum, as a new to science. Besides, it reported a mushroom, that is R. senesis, as a first report from West Bengal, as well as first report as a tribal food. Both the mushrooms were composed of beta-glucan in significant amount that can be isolated easily by hot water process. Both the polysaccharide fractions have strong immune stimulatory activity mediated through TLR NF-kappa B pathway. So at present, we are working on the isolation of active component by chromatography and validation of the immune stimulatory activity at in vivo level. So the journey is continue to shed light on wild edible mushrooms and ethnically prized mushroom of West Bengal for exploitation in food and pharmaceutical arenas for human welfare, in which a strong collaboration between R&D and industrial enterprise is highly required. Thank you for listening.
And now the presentation is open for discussion. Some questions from the audience?
Excellent talk, Sumanjana.
Thank you, sir.
Very good. Nice.
Any questions?
Sumanjana, congratulations for really a fantastic talk. Thank you.
Thank you, sir. Thank you.
Yeah, it's a wonderful presentation you did, Sumanjana.
Thank you, sir.
Now we'll move to the next presentation by Ranjit Singh from Dev College, Batinda, Punjab. We're talking on assessment of elements in some gynoderma species and their potential contribution to dietary intakes. Dr. Ranjit Singh.
Hello, Dr. Paneet Selvam. Good afternoon.
Yes, sir. Good afternoon, Professor.
Dr. Acharya, good afternoon.
Good afternoon.
Dr. Krishnendu Acharya, can we
Yeah,
have...
sir. Good afternoon, sir.
Cannot have your video. It's great to see you, sir.
I am here from the morning.
Yes, sir. But I was looking for a chance to talk to you and luckily got that. Ranjit, continue.
Yeah.
Good afternoon to all the distinguished gathering. I am indeed honored to present a paper on
assessment of elements in some Ganoderma species and their potential contribution to dietary intakes. Ganoderma is a wood-degrading polyporoid mushroom and causes different types of rots in both conifers and hardwoods through the breakdown of lignin, cellulose, and other structural components of wood. Ganoderma has been perceived as a natural adjuvant for its miracle cures in traditional system of medicine for thousands of years. Various ancient scripts recorded herbal history of mushroom regarding its efficacious effects. The mushroom is an affluent source of active metabolites, imparting multifarious pharmacological effects. Various Ganoderma waste products such as toothpaste, soaps, and beverages, food products such as tea, coffee, pills, wine, beer, etc. are available in the market with significant market value. Now, the analysis of nutrients in natural products is highly relevant for consumers' demand, not only from a nutritional perspective, but to evaluate quality and toxicity from their consumption. Monitoring of macronutrient and element levels in mushrooms provides basic information in terms of safety, regulation, and nutrition. Adequate dietary intake of essential elements is important for human health and wellness. Further, there is a need to standardize the permissible levels of elements in natural products to evaluate the potential health risks. With all these attributes, the purpose of the study was to determine the level of elements in 4 Ganoderma species: Ganoderma applanatum, Ganoderma brownii, Ganoderma lucidum, and Ganoderma philippi, collected from Uttarakhand, India by wavelength dispersive X-ray fluorescence spectrometry. Statement of novelty. This study gives a comprehensive evaluation of elements and their contribution to dietary intakes of elements with a view to provide information on the nutritional quality and safety aspects of the mushrooms. Until now, most available data was limited only to the quantitative analysis of the selected elements in Ganoderma lucidum and Ganoderma applanatum. Quality indices were reported for the first time. The fruiting bodies of the mushroom samples were collected during the monsoon months from different forest sites of Uttarakhand, India, and identified on the basis of morphological and microscopic characters. Voucher specimens of the mushrooms were deposited at the herbarium of Department of Botany, Punjabi University, Patiala. And this table shows the herbarium number and sampling sites and hosts of examined mushrooms. The concentrations of different elements were determined by the wavelength dispersive X-ray fluorescence spectrometry. These are the instrumental conditions. WDXR spectrum acquired of elements of Ganoderma applanatum. This is the WDXR spectrum of elements detected in Ganoderma brownii, and WDXR spectrum of elements detected in Ganoderma lucidum and in Ganoderma philippi. This table shows the concentrations of different elements quantified in different Ganoderma species. It is evident from this table that calcium and potassium were detected in predominant amounts in the studied mushroom species. And the results were determined in triplicates, and student test significant was applied to test the mean significant differences among different elements in mushrooms. Then the different elements, concentrations of elements were plotted on logarithmic scale. to show the clear-cut distinction of the amount of elements in the studied mushrooms. The level of elements depicted in ranges that is shown in red lines, along with the names of the species marked by their corresponding values. Such a way of data presentation gives clarity regarding which mushroom is beneficial and provide direct information on the potential risk to the health of consumers due to the consumption of these mushrooms. The most heterogeneous distribution was observed for zirconium. Among all analyzed mushrooms, the lowest maximum amount was observed in fruiting bodies of Ganoderma applanatum. That provided the lowest, extreme lowest values, and whereas the highest values of elements was observed in Ganoderma lucidum. The next analysis consists of Pearson's linear correlation analysis that was used to establish the strength of the relationship between analyzed element contents. Significant correlation coefficients, that is equal or greater than 0.514 at 0.05 probability level, was established. Red marks show the very high significant correlation at R equal or greater than 0.9. Additionally, the Friedman rank sum test was performed to indicate differences between the studied species of mushrooms concerning levels of essential macro elements, trace elements, that is essential as well as toxic elements, and all the elements jointly, followed by post hoc Nemini test. Friedman rank test reached a lack of significant differences among the studied Ganoderma species regarding the amount of all 19 elements jointly in particular mushroom species, because p-value is greater than 0.05. Concerning the amount of 13 trace elements, that includes both the essential as well as the toxic elements jointly in the basidiocarps of the mushrooms, no significant differences have been observed between them. Similarly, a comparison of the analyzed mushroom species with reference to the amount of all 6 macro elements, there is also no significant differences on the basis of jointly all the macro elements. The highest level of all 19 and 13 trace elements has been confirmed in Ganoderma browni, while Ganoderma lucidum revealed the highest content of 6 macro elements jointly. Then the dietary intakes for essential elements were calculated in terms of estimated daily intake and recommended daily intake and nutritional quality index. This table showed the average estimated dietary intakes by a normal 60 kg adult person in milligram per day of essential metals from mushroom consumption and their contribution to recommended dietary intake. The most important source of calcium is Ganoderma philippi, which alone contributes 96.3% of the total RDI. And the percentage realization of copper met between 58.2 to 95.8% of the daily requirement of male and females. The species that contributed most to the intake of iron, potassium, magnesium, and phosphorus is Ganoderma lucidum. That contributes 97.1% and 78.6% for iron and 10.5% for phosphorus and 11.5 and 12.6% for men and women. For sodium and potassium, the studied mushrooms represent low percentage values of RDI. However, in comparison to potassium, the amount of sodium is significantly lower in examined mushrooms, which is appraised to be favorable from the nutritional perspective, as consumption of diet rich in sodium, that is with a high risk of Na to K ratio, leads to health issues. Nutritional quality index. Nutrition quality index is the amount of nutrients that is supplied from 100-gram powdered sample as a function of the RDI in relation to the
another 2 minutes more. 2 more minutes.
Energy supplied. And a value exceeding 1 indicates a positive contribution of an essential nutrient. All the values NQI values are greater than 1 except for sodium and potassium. It means that the studied mushrooms are not good sources of sodium and potassium. Now the dietary intakes of toxic elements. Now, the toxic metals present in functional foods may exhibit harmful effects on the health of consumers. The safe levels of intakes can be determined in terms of tolerable intake, either daily, that is tolerable daily intake, or weekly, that is tolerable weekly intake. The tolerable intakes of heavy metals as provisional tolerable weekly intake are laid by the Food and Agriculture Organization, World Organization, that is FAO. Now, two that is toxic elements were detected in mushroom species, but they are lower than the permissible limit. For aluminium, provisional tolerable weekly intake, that is PTWI, is 1 milligram per day. At high levels, aluminium have serious effects on the nervous system.
Dr. Ranjit, conclude in another minute's time. You have 1 minute to conclude.
Conclusions. Detailed information on proximate and elemental composition is crucial for estimating dietary intakes, safety of food consumed, for sharing relevant information regarding nutrients to consumers, which may be a basis for future monitoring studies. The studied mushrooms contain appreciable amounts of essential elements for maintaining good health. The concentration of different elements varied widely among studied mushroom samples. Mushrooms can serve as a dietary source of calcium, copper, iron, magnesium, manganese, zinc, and therefore makes remarkable contribution to dietary intakes for humans. Potassium contributions are inadequate in comparison to the reference intake values. Thus, supplementation of potassium is recommended before formulation of Ganoderma powder in various dietary supplements. There is a need to screen out more species for higher uptake of potassium and sodium in order to decrease the deficiency of these essential elements in humans. Consumptions of these mushrooms does not represent toxicological health risk for humans, as mean exposure estimates of the elements such as aluminium and lead were found to be below the toxicological reference values. Thank you.
Yeah, thank you. We'll have maybe one question from the audience.
Dr. Ranjit, don't you think that elemental composition of these mushrooms will vary based upon the type of soil composition where they are growing?
Yes, sir. Because mushrooms are bioaccumulators, and they are very efficient in accumulation of that is different elements from the soil.
Maybe having only 2 heavy metals in them, it may be more. It depends upon the type of soil where they are growing, and also the various elements which are you have documented.
Sir.
Very depending.
But the only one, that is Ganoderma lucidum, is connected with the roots of the tree. Other 3 species, they are connected with the stem. That is their substrate is stem of the host tree.
Okay.
Yes, sir.
So otherwise, making generalized recommendations based upon this, it may be a bit risky. That is what I intend to say. Yes. Anyway, thank you. Nice presentation.
Thank you so much.
Yeah. Thank you, Ranjit. Now we move on to the next presentation by Nazir Ahmed Malik from Central University of Jammu and Kashmir. He will be talking on characterization and identification of some novel taxa and new records of laminate fungi from Himalayan belt of India. Dr. Nazir.
Am I audible?
Yeah. Please, you can see your presentation.
So, very good afternoon to all, and thank you very much for giving me an honor to present a paper on the entitled this characterization and identification of some new species and new records of laminate fungi from the Himalayan Delta of India. So the mushrooms, if we talk about their history goes back to the ancient Romans, who considered these fungi as a divine food. And they were believing that these fungi, these mushrooms it is due to the result of lightning thrown down by the planet Jupiter. So if we talk about its definition, mushrooms, these are large and complex spore-bearing fleshy fungi that are typically grown above the ground in soil or on various other substrates. So the agaric is a common term, that is a common term for the gilled mushrooms. These gilled mushrooms are basically characterized by fleshy, stiptate, centric, eccentric, or we can say annulate or axonulate coprophones with pilus, that is the lamellate. And they are usually consisting universal, some are partial veils, which are with or without vulva at base of the stipe. Spores may be smooth or ornamented. If we talk about their habits, they may grow either solitary or scattered, or they grow in groups, gregarious or in cespitous clusters. So far as the habitats are concerned, they may be folicolous, lignicolous, hemicolous, coprophilous, dermatophilous, etc. and etc. So these recently, Frank and his other co-workers in 2010, they discovered a species, Cethrilla aquatica. This species is growing in the water, and another habitat, that is aquatic habitat, is also possible for these mushrooms. So the fungal forays were undertaken to the various localities, or we can see the various dense forests of South Kashmir, which is a part of Himalayan belt. And all the information, basic information, such as the collection deeds and other ecological aspects, they were noted down, and habitat photographs, spore prints were taken, and after identification and after their identification, these specimens were submitted in the herbarium of Department of Botany, Punjabi University, Patiala. Both the macroscopic as well as microscopic characters were noted down, and some new species that classically we identified the species, and some species were proposed in new science, and further they were confirmed with the molecular taxonomy. So this is the area which have been surveyed, and these are localities, dense forests of this South Kashmir, which is very difficult to go there, and because of certain threats. And so far as my work is concerned, I have collected 86 species 86 population belong to 32 genera and total 78 species. 17 families belong to 5 orders, and out of which 7 species were proposed as new science and were further conformed with their molecular taxonomy, and some ethnobotanical data have been collected. Sociobiology have been done, and among which 4 species were selected for their biochemical analysis. So these species were proposed as new science and are communicated for their publication. Lentinus transitus. This species, it has been proposed as a new species because due to the mixing characters of both subgenus of Lentinus as well as subgenus of Panus. These are the both subgenera of genus Lentinus. So the mixing characters were found in this species. This species were characterized by having Giliocystidia and having the hyphalpags. Hyphalpags are the main characters of the subgenus Lentinus, and the cystidial hairs, which are the main characters of genus Panus. So there are some mixing characters, and this species have been proposed as new science and further confirmed with the molecular taxonomy. So similarly, Zerula magnispora. This species, there is only one genus, there is only one species in this Zerula genus in which the large, very large deciduous spores are present, that is Zerula megaspore. But in our present collection, we have a large deciduous spores, very large deciduous spores, and the shape of Chilocystidia, both the large deciduous spores and shape of the Chilocystidia makes it different from the other 2 species that are in comparison, that is in Zerula megaspore and Zerula astris. It is further compounded with the molecular taxonomy. So similarly, we have proposed another species, that Lactyrus volcani. This species is characterized by having the young carpophores that are bluish green in color, and after maturity it changes its total color into red, and the concentric rings are present on the pilus, and in the center the pilus is depressed. Here you see the concentric rings. These scales are present in the concentric rings. So here the color of the young carpophores, they oozes out the greenish bluish this color oozes out a latex, and while as at maturity its latex is reddish in color, but in a young carpophore it is just only the greenish blue latex is out from the same specimen. So further this species was confirmed that it has ectomycorrhizal association with the Pinus volcana. This is the structure here shows it. So similarly, it is further confirmed with the molecular analysis. So similarly, we have another species, that is Clericinda indica. This species is characterized and have been proposed as new to science because of certain main characters. The doliform pilus cuticle is always present in the every species of genus Clericinda, but here the doliform pilus cuticle is totally absent. And if you see the annular rings, these annular rings, it consists of both cellular cells as well as the hyphae. The hyphae has more breadth as well as the cellular, they have more breadth in comparison to the other if we talk about this Clericinda records. So similarly, it is confirmed with the molecular analysis. Further, we have another species, Lentinus cashmirinus. It has been published in this Kawaka Journal. This species, it has a pseudorhiza type style. So this is its main character, and further, this species consists the Chilocystidia, which are just metalloid. So this species falls under the subgenus Panus and section Phylogenetic, and it is compared with the allied species, that is Lentinus adherens and Kaufmanni. So there are certain new reports from India. These are new reports to India. 34 new reports were recorded from India, and this is from North India. These are new records, and some other records from Jammu and Kashmir, Kashmir Valley, and South Kashmir. Further, the 4 species were selected for their this nutritional and nutraceutical analysis, among which the 2 species are new science, that is Lentinus transitus, which is in communication in this general Sidovia, and the same species which has been published in the Cloaca, that is Lentinus cashmerinus. Both 2 species are new science, and these we selected these 4 species for their nutritional analysis. So if we talk about among these 4 species, the best species having the protein content, that is the Phylogenetic cornucopia. This Phylogenetic cornucopia has good content of proteins, carbohydrates, crude fat, and crude fiber as compared to the other 3 species. Similarly, we have nutraceutical composition of these elements, these 4 species. The best these steroids, maximum steroids were reported in Phylogenetic cornucopia as compared to the other, whereas in Lentinus transitus, maximum amount of beta carotene and lycopene were found. So similarly, we have done the macro element analysis of these 4 species, that is this Phylogenetic cornucopia, Conatus, Cashmerinus, and Transitus. So these are 4 species, among which the Phylogenetic cornucopus has maximum amount of calcium, followed by sodium. Similarly, the Conatus, this Conatus species has we found maximum amount of phosphorus and sulfur. Similarly, the Cashmerinus, minimum amount of calcium is found as compared to the Phylogenetic cornucopus, and potassium, magnesium, sodium, these were found in minimum.
Dr. Nazir, you have 2 more minutes.
Yes, sir. So similarly, the microelements were reported in these 2 species. So far as this distribution, ecology, and association, seasonal availability, and economic ability of this species, maximum collections were done during summer, followed by spring, autumn, and no collection were reported during winter. Similarly, month-wise distribution, maximum collections were done during July, then August and May. Similarly, so far as this habitat is concerned, the maximum species were reported as ectomycorrhizal, followed by Lignicolous, followed by Folicolous, Lignicolous, terrestrial, and Coprophilus. Similarly, we have reported the 3 main hosts, that is Pinus volcanus, Citrus dildara, and Abies pando, in which these fungi had their ectomycorrhizal association. So far as this edibility is concerned, maximum 30 species, 30% species were found they are edible, and while as 14% were found as a poisonous, and medicinal 9%, and unknown species were 42%. So these are some summary of these medicinal species and some poisonous species, and they were confirmed for the literature. So thank you very much.
Thank you. It's open for questions. The topic is open for discussion. Maybe we can take 1 question, 1 or 2 questions.
Nazir, you should check the nomenclature of Lactarius new species, what you have written, Valikiani, something like that. Should check it as per the
Actually,
code.
okay, sir. Okay, sir.
Yeah, thank you. Thank you, Dr.
Thank you, sir. Thank you very much.
Move to the next presentation by Manoj Kumar A, Government College for Women, Thiruvananthapuram, Kerala. We will be talking on morphological and molecular perspectives on systematics of genus Crepidotus in Kerala state.
Sir, he is not presenting, sir.
Not presenting. Okay. So then we'll move to next presentation is by Sapna Sharma from DAV College, Jalandhar. Sapna is there?
Yes, sir. Yes, sir.
Yeah. So we're talking on the potential of arbuscular mycorrhizal fungi for the cultivation of important medicinal herb, Mycorrhiza corea.
Is it visible, sir? Is it visible, sir?
Beg your pardon?
Not visible.
Your presentation is not visible.
You have started sharing your screen. There is
Yes,
some.
sir, I have started.
Sir, I think there is some internet issues.
Go ahead.
Something is
Sir,
coming.
again, I am sharing.
Okay. In that case, we'll move to the next presentation, and then you can. Yours would be. We'll take it next after the presentation. My Mridu is present. Ah, yeah, it's come. Yeah, we can see it now.
Is it visible now, sir?
Yeah,
Yes,
yeah.
it's visible. You can start. Yeah.
You can go ahead.
Okay, good evening to all. Today I'm going to present my presentation on the potential of the arbuscular mycorrhizal fungi for the cultivation of important medicinal herb, that is Mycorrhiza puroa. So, as all of you know that medicinal plants are the potential renewable resources in our country.
Dr. Sapna, your voice is not audible. Kindly check if it is unmuted.
It is audible.
Yeah, it's okay.
Is it audible?
Yes.
Okay.
Please continue,
Please.
please.
I'm sorry.
Okay, sir. So, due to the demand of this valuable resource, the increasing demand with their survival in natural condition is under the threat. Due to the unsustainable exploitation of resource that has led to the extinction of useful plants and that result in the extinction of many medicinal plants. Hence, there is a critical need to standardize some agro techniques for the conservation of these important medicinal plants. And we have also tried in this investigation one such category of the biofertilizer, that is endomycorrhizae, and these are also called as arbuscular mycorrhizal fungi. So, mycorrhiza term that was first coined by the scientist A.B. Frank in 1885, and it is a combination of two words, that is myx means fungus and rhiza means roots. Means this is a symbiotic relationship between the fungi and roots of the higher plants. So, mycorrhizal symbiosis that plays an important role in transformation, mobilization, solubilization that form that limited nutrient pool in the soil. And this mutual symbiotic relationship is a three-way interaction between the host plant, fungal partner, and the soil factors. Though approximately 90% of the plants that form this symbiotic association. One of the unique features of the association is it is obligatory in nature because it requires a particular living host for its survival. So my mycorrhizal fungi, they are specialized in absorbing nutrients from the soil. They found under all the climates and in all the ecosystems. These mycorrhizal fungi, they enable the plant to survive, grow, and reproduce more effectively. These fungi, they are the supplier of nutrients to the plants and in return get sugars, amino acids, and vitamins from the plants. One more unique feature of these fungi is lack of host fitting, means one fungi can colonize so many plants. So these are the cellular structure which are formed during the symbiosis. When the spore that comes in contact with the roots of the plants, then they form the mycelium, and then this extensive mycelium is formed in the, this is intracellular, means between the cell, and the structure formed their arbuscule. Arbuscule is the major site for the nutrient exchange between the fungi and the roots of the plant. So here I have listed some of the benefits that these fungi improve the fertility of the soil, uptake of phosphorus, uptake of water, nutrient mobilization and cycling, soil stabilization, also protects against the environmental stresses and also against the root diseases. So when the symbiotic phase is there, then three main phases are there. First is pre-symbiotic phase, when the plant and fungi, they come in contact with each other and some signals are there. And response of plant to these fungi signal is there and the colonization that will be formed. Aprisorium development will be there. Then the penetration of the M fungi is there. Then finally arbuscule development is there inside the cells. And these arbuscule, these are the major sites for the nutrient transfer. I have worked almost 85 medicinal plants of Himachal Pradesh, and I have isolated different strains of M fungi from these medicinal plants. I have listed some of them here. These are the some microscopic pictures of the spores of the mycorrhizal fungi, which I have isolated from these medicinal plants. These belongs to different genera of the arbuscular mycorrhizal fungi. So this is the colonization. Here you can see the extensive mycelium. Different types of vesicles are there. Then these vesicles, they are intracellular. And this is the photograph in which you can see the spore is infecting the root cell and forming the extensive mycelium. So now coming to the selected plant that I am presenting, Picrorhiza kuroa. So commonly it is known as atish, and it is traditionally expected by the local people for the traditional medicines. So it is a distinguished medicinal herb of Himalayas. It is a high hill plant and belongs to the family Scrophulariaceae. Roots and the rhizome, they are medicinally important parts, and they can be used as powder, decoction, infusion, and alcoholic extract, and that is used against the different diseases like jaundice and joint pain, other inflammations. So according to Red Data Book data, it is listed in the endangered medicinal plant list because it is overexploited by the traditional people from the wild conditions. And when we try to raise the seedlings of this plant in the nursery condition, then high mortality rate is there. So for the conservation of this valuable resource, this important medicinal herb, we have tried here arbuscular mycorrhizal fungi as biofertilizer for their survival in the nursery stage. So with the two objectives, that first we have assessed the biodiversity of M. fungi associated with the Picrorhiza kuroa. Then we screen the effect of efficient and dominant indigenous M. fungi on the growth and physiological parameters of Picrorhiza kuroa. We have isolated the species from the rhizosphere of this plant, and again inoculated these indigenous species to the this plant's roots. So these are the locations from where we have collected sites. These are different types of these are the sites where from where we have collected the first the soil sample, and this is one of the picture we have tried the farmers field the trial of our bioinoculants. So material methods, we have collected the soil samples, and these samples were analyzed for isolation of M spares, mycorrhizal quantification, identification of M fungi, then colonization in the roots of the plant, and again mass production of indigenous dominant M. fungi by funnel technique. And then these mass produced M. fungi, they are further inoculated in the seedlings of the Picrorhiza kuroa. So these are the initial soil properties, as I have collaborated with the soil science laboratory, Punjab Agricultural University, Ludhiana, for my study. So these are the soil properties and the various methods by which we have analyzed the different components of the soil analysis. So this is the table which is showing the association of the arbuscular mycorrhizal fungi in the form of presence of mycelium, arbuscules, vesicles, percent root colonization, and number of species which are associated with the roots of the Picrorhiza kuroa from in the soil sample collected from these different sites for the study. So these are the two, after isolation and after identification, these two species, one is Acolospora laevis and second one is Funliformis mossi. These were dominant species, and these are some pictures of the colonization with the Picrorhiza kuroa. So for further study, we have selected these two dominant indigenous species for further experiment. So these two species then further mass multiplied by using different hosts like barley, onion, and maize. This is the Acolospora laevis mass production, and this is the Funliformis mossi mass production setup. So then final bioinoculation is done with the roots of the Picrorhiza kuroa and the plants of the seedlings of the Picrorhiza kuroa, and these are the treatments which we have tried with the Funliformis mossi. In addition to that, we have also tried vermicompost and farmyard manure at the recommended dose, that is 10 ton per hectare. So from after 120 days of inoculation, we have taken data, and according to that observations, we have taken in consideration number of leaves per plant, root length, fresh weight, dry weight, root and shoot, then percent mycorrhizal root colonization, spore number per 10 gram of the soil, and type of colonization in the form of presence of mycelium, arbuscules, and vesicles. So when we observed it carefully, all the bioinoculations, they increase the biomass, and they also help in the survival of the seedling. Mortality rate, that is also reduced. Then some physiological parameters, they were also studied, like pH, EC, OC, available nitrogen, phosphorus, potassium, and percent phosphorus content in the root and the shoot. So with these recommended doses of the vermicompost and farmyard manure, we have also tried that cultivation of this valuable resource under the organic conditions. We have not applied any other fertilizer. We have only in the farmers field, we have tried only these treatments, and with the when we recorded the results, then further we have just like enzyme activities were also screened. I'm not presenting here the data. So all these parameters, they were found to be increased with the help of these bioinoculants over the control plants. So we came to the conclusion by this bioinoculation experiment that these fungi strains, they alone and in consortium treatment, they improve the growth and yield along with the promoting nutrient availability of nitrogen, phosphorus and potassium in the rhizosphere of Phycrorhiza courroa. So we can select the best plant fungus combination for the inoculation of these seedlings. Then further we have, as we have mass produced Colospora levis and Glomus mosi in our conditions in our laboratory, then we have also supplied this culture to the forest nurseries in the Himachal Pradesh, which are just raising the seedlings of this valuable plant Phycrorhiza courroa. Then they inoculated the mass culture in the just in normal seedling bed without any other treatment. They just inoculated the Funliformis mosi and the Colospora levis. Then we have taken data from these nurseries. Then they just reported us that the mortality rate that is reduced highly reduced mortality rate जो है वो इस fertilizer से बहुत कम हुआ है. So our future prospects, they are just like we just want to assess the alkaloids present in this means effect of these fungi inoculation on the alkaloids of these valuable medicinal plants. So with this, I sincerely thanks to University Grants Commission New Delhi for the financial assistance in the form of postdoctoral fellowship and Punjab Agriculture University Ludhiana for the necessary laboratory facilities. My sincere thanks are due to DAV College Jalandhar who provide me opportunity to present my paper here, and I am here also thanking all the delegates, all the dignitaries and Punjabi University Patiala for providing me the platform for presenting the work. Thank you, sir.
Yeah, that's the presentation is open for discussion. Any questions? I have a question, Dr. Swapna.
Yes, sir.
Did you prepare any trap cultures for the identification before you attempted to identify the main fungal species?
Pardon, sir.
Did you prepare trap cultures? Did you prepare any trap cultures?
Yes, sir. We have prepared our own laboratory, Colospora levis, Funliformis mosi, and Glomus intraradicis, macrocarpum, 4 species we have multiplied.
Okay. Which was the most dominant species in this?
Sir, 2 species only, Funliformis mosi and Colospora levis.
Okay, the same species you have taken as multiplied and taken as inoculants. The
Yes,
same.
sir.
Okay. Okay.
Okay. Thank you, sir. Thank you
so much. Yeah. Okay.
Honorable Chairperson, can I take a minute, sir?
Yes, sir. Please.
Sir.
We have with us the revered teacher from Botany Department, the former President of Mycological Society of India 2005-06, the great man of field of pyrinomycetes, Professor J.S. Dargan with us. He has joined all the way from England. He is currently in England with his son. We welcome you, sir.
Welcome, sir.
Sir,
Good to have you, sir, with us. Really lovely to see you.
Good evening, friends. My best wishes to you all.
Yes.
See you, sir.
Theek-thak, sir. All well.
Hello, sir. Everything is fine. Thank you very much.
Sir, we
are grateful you joined. It is a blessing.
Sir, we wish to listen to you.
Thank you, Dr. Tri. Thank you. So nice of you. I was trying yesterday also, but unfortunately I could not get the link.
Thank you, sir. Thank you for joining, and it is really heartening.
Welcome. You very happy to see you all.
It's very encouraging for us, sir.
Thank you.
Professor Dargan Saab, namaskar.
Great. Namaskar, sir. How are you?
Fine, sir. Fine, and very nice seeing you.
We met at Bombay conference also.
Yes, we met in Bombay conference, and in Bangalore also we met. Yes.
Bangalore we met. Yeah.
Sir, Dr. Lakhanpal is also there.
Okay. Hello, sir. Dr. Lakhanpal.
Hello, sir. Welcome. Most
Thank
welcome.
You, sir. So nice of you. Very nice to see you. Very nice to see you all and join you.
Very nice, sir. Yes.
Thank you, sir. Carry on. It's going wonderful.
Thank you, sir.
Everything is so coordinated.
Thank you, sir.
My best once again. Hello, Geetika. How are you?
Fine, sir.
Good. it seems that I'm sitting here it seems I'm sitting here with you there.
Good morning, sir.
Hello.
Good morning, sir. Good morning. Sir,
बुरे
हो गए थे जी. Sir, you are looking very smart.
Good, Abhi, how are you? I especially took a time because the time is a 5 hour, I think, behind.
Yes.
So I think this time I could join you. Thank you.
अच्छा लग गया सब बहुत अच्छा
Sir, the event
Very
is
nice, sir.
now complete in real sense with your gracious presence, sir. Thank you very much.
Thank you, so nice of you. Please carry on.
Chairman, sir, please carry on.
Thank you. Yeah.
Okay, sir. Thank you.
Thank you, sir. We'll move to the last presentation by Mridu from Dayal Singh College, Karnal, Haryana. We'll be talking on a study on fruiting phenology, distribution, ecology, and economic utility of wild laminate mushrooms from Haryana. Dr. Mridu?
Yes, sir. Good afternoon, all of you. Am I audible to you, sir?
Yes. Yeah. Your presentation also can be seen.
Should I start the presentation now?
Please, yes, go ahead.
Make it full screen.
Yes, sir.
It's okay.
Okay, sir, fine. So today I am going to present a study on fruiting phenology, distribution, ecology, and economic utility of wild laminate mushrooms from Haryana, which I have undertaken under the kind supervision of Professor N.S. Attri and Professor Manruchi Kaur during my PhD research work. As we all know, despite the important contribution of fungi to environment, little is known about the factors that influence fungal phenology. Therefore, in order to further our understanding on how fungal fruiting pattern of lamellate mushrooms change along a gradient, we investigated the phenology pattern of some about 51 species of lamellate mushrooms from 3 districts of Haryana, that are Ambala, Yamunanagar, and Panchkula. We all know lamellate mushrooms derive their nutrition from complex organic matter, and for their sustenance, they occupy a great range of substrates such as soil, leaf litter, living or decaying wood, dung. They can be found in mycorrhizal association with higher plants or in association with termitaria. Every agaric also has its own habit of growing either solitary, scattered in groups, or cespitous clusters, or in fairy rings. These are some of the photographs of the mushrooms which I have collected. Like Podexis pistillaris was found growing on the soil, solitary habit, and Lentinus crossulus was found growing on a dead log in cespitous manner. Volvacia, it was found growing on paddy straw, as it's commonly known as straw mushroom or paddy straw mushroom. And Termitomyces microcarpus was found growing gregariously on a termitaria. Leucopaxillus paradoxus I found growing in the form of fairy rings. Agarics, like other fungi, require high humidity and low temperature for their growth. And Haryana, being in northern part of India, receive maximum rainfall during monsoon season, that is in the months of July to September. Therefore, we planned excursion during monsoon season, although some trips were also taken in the pre-monsoon and post-monsoon season, but hardly any workable specimen could be found. And collection of wild mushroom besides this, collection of wild mushroom for personal consumption is a common activity all around the world, including Haryana, as we all know mushrooms are rich in proteins, carbohydrates, dietary fiber, mineral elements, and these are low in fat content. Besides this, mushrooms are important sources of many bioactive compounds such as polysaccharides, terpenes, phenol, etc. So the mushrooms which I found in the field were, some of those were edible, inedible, poisonous, hallucinogenic, or medicinal. The area which I surveyed in Haryana particularly was Shivaliks of Haryana, which covered three districts: Panchkula, Yamunanagar, and Ambala. 45% of total area of district Panchkula is covered by the Shivalik Hills, 9% of Yamunanagar, and that of 0.2% of total area of district Ambala covers the Shivalik Hills. And the soil of this region is made mainly alluvial and silty loam or other type of soil. The climate varies. All variation can be found here in this region. Summers will from mid-March to last week of June, monsoon from July to September, and post-monsoon season September to November, while winters here in the late November to the first week of March. Normally, the annual rainfall is 1057 to 1107 mm, and especially in the months of monsoon, the annual rainfall is 80 to 86%, and relative humidity is 85%. The specific areas which I have covered during my survey are highlighted in the maps. These are the views of some of the localities which I have visited. Like this is the village Adohi from Ambala, Kalesar National Park area, Yamunanagar, Morni Hills, which is the only hill station in this area, that is in Panchkula, and the last photograph is from village Rador of district Yamunanagar. As I have mentioned, we have undertaken exploration towards to these districts during the monsoon, mainly the monsoon period of years 2013 to 18, and majority of the collection has been done during this period. And during the field survey, first of all, we spotted the gill mushrooms, and multiple photographs were captured from various angles in their natural habitat with a good quality camera. The ecological details with respect to the forest types, soil type, putative association of mushrooms with the nearby vegetation, their habit, their habitat, ecology, distribution, and morphology of individual mushroom was noted down. All the details were noted down in the field key, and the mycological information was also noted down in a special questionnaire which was designed by Paul 2005 and 17. All these specimens were collected, and after studying their morphology and anatomical details and need-based molecular details, all the specimens were
Dr. Mridu?
identified by comparing with the
Dr. Mridu?
literature available.
Yeah, I think you have kept.
This is the questionnaire which were designed for the for noting down the sociobiological data. Like the local people of the area of investigation were inquired about their activity of collecting mushrooms and eating the wild mushrooms. Like do they eat mushrooms or not, and who in the family collects the mushroom, in which form they consume the mushroom, either they make recipes or and also if they preserve the mushrooms for the off-season use, and is there any myth or belief or stories about the mushrooms among the people. So all these information, all these questions were acquired from the local people and noted down. So during this study, I collected 76 specimens which belong to 51 species and 24 genera, spread over 15 families and 3 orders. This is the table showing the list of taxa which were identified along with their date of collection, locality, and altitude of the place from which these were collected, the habit in which they were found growing, and if these were found in association with termitaria or other plant, it was also noted down, and the edibility status and medicinal status was also noted down. The edibility status was asked from the people, as well as the information was taken from the literature. On the both of the basis, we have constructed this table with respect to each specimen. So this is the list of 51 species with all the details, and on the basis of these details, we analyze that the maximum of the mushrooms were found during the month of August, that is during mid of the monsoon period. 62% of the collected fruit bodies were collected during the month of August, followed by the month of July and September. And among these, we noted that the species of termitomyces could be collected during whole of the monsoon period, while some of the species were found only at the beginning of the monsoon, while some of those could be found during end of the period, while the species of termitomyces could be collected in whole of the monsoon period. This is a graph showing frequency of occurrence of various taxonomic categories, and the family Agaricaceae carried the maximum number of genus and species. This is the percent genus-wise frequency of occurrence in different families. The family Agaricaceae lead in this analysis also, and in percent-wise species distribution, the number of species were maximum belonging to the family Agaricaceae. If we talk about the distribution of agarics in various habitats, I found maximum of the fruit bodies growing terrestrial, that is 47%, and followed by termitophilous and lignicolous species that were 13% each, and follicolous species were about 10%, followed by 6% of the coprophilous, 8% ectomycorrhizal species, and 1% of the species were found growing on the wheat straw. And most of the species were growing in solitary or scattered habits. 25% of the species were solitary, and 25% of the species were growing scattered in the nature, and followed by the cespitous and gregarious cespitous and cespitous species were 18%, and those of growing in groups were 15%. Gregariously growing species were 14%, and fairy rings, in the form of fairy rings, I found 3% of the collection.
Dr. Mridu, you have 2 more minutes to conclude.
Okay, sir. Okay, sir. I also categorize the whole data on the basis of edibility and medicinal utility, and I found more than 50% of the collection was edible, including 21% of the species only edible, and while 31% of those edible were also having the medicinal utility. From all the study, I can conclude that in the present study, 51 species, including a good number of edible and medicinal ones, growing in the wide range of habits and habitats, were documented from 3 districts of Haryana for the first time. It is the first ever study which I have undertaken, and Agaricus was found as the most dominating genus. The presently examined texts have been categorized according to their edibility status, from which about 51% of these species were edible, including 31% with medicinal utility, and rest were inedible, poisonous, hallucinogenic, or some of the species have been unreported for their status of edibility. And a majority of the species were found growing in terrestrial habitat. Amanita, species of Amanita and Drusilla were found in growing in putative mycorrhizal association with Shoria robusta, and Calocybe gambosa was found forming putative ecium association with Delbergia ciso. So at the end, I would like to say the phenology pattern or fruiting pattern is important study. As we all know, distribution and other aspects of the fungi are being, the studies are being undertaken widely, but somehow we are lacking in the records of the phenology or distribution pattern, which is important. Spatio-temporal classification or distribution according to the climatic and phytogeographical gradient also plays important role in our environment. So there is a need to undertake these studies more in the case of agarics. Thank you very much.
Okay, thank you. Any questions from the audience? So if there are no questions, we come to the end of this session. And before I give my concluding remarks, I would request the co-chair, Professor Paneer Selvam, to give his comments on this session. Professor Paneer Selvam, please.
Very good afternoon to all. I would like to thank the speakers of the 11th session. I mean the last session of the 3 days conference. All the lectures were excellent, and my best wishes to all. I thank the chairperson, Professor Rodriguez, Professor of Botany, Goa University, organizers of this program, Dr. Avneet Pal Singh, Dr. Mundrich Kaur, Professor Inna Satri, Professor Manish Kapoor, MSI Secretary Professor N. Raman, and all the eminent mycologists. Thank you for giving me the opportunity. Thank you, Professor.
Dr. Manish, please.
Thank you, Dr. Rodriguez and Dr. Paneer Selvam for conducting the session very smoothly and evaluating the mid-aged faculty members and scientists for the award. And I thank the organizers, Professor Manruchi, Dr. Avneet Pal, Dr. Atri, for giving me this chance to be here to moderate this session.
Sir, I am a guest like.
No, sir.
No, sir. You are the person behind.
Sir, we don't consider you a guest. Okay. Thank you. Thank you, Dr. Avneet. Over to you.
Thank you very much, sir. We are blessed to have such wonderful. Yes, sir, please. Dr. Raman, kindly unmute yourself.
Right, thank you. I thank the moderator Manish Kapoor and also the chairperson and co-chairperson for efficiently conducting the session in time. Thank you very much.
Thank you.
So extremely thankful.
Thank you, sir. Thank you,
Dr. Manish and all the delegates and presenters. So we have we are now left with our valedictory session, which is originally scheduled at 4:30. Because of the two presenters who were not able to present their work, we have finished it 10 minutes earlier. And secondly, the efficient management of the chair and the co-chair that has made it to be finished in time. Big thanks to Dr. Rodriguez and Dr. Paneer Selvam.
Thank you.
What we can do, we can have a short, crisp break of 10 minutes.
Yes,
and we will start with our valedictory session at exactly at 4:30 p.m. So 10 minutes break for refreshing yourselves, for resting your back and making a bit relaxed. And we all be together at 4:30.
Okay.
Good evening, Dr. Yashpal ji.
Sir, good afternoon, good evening, everyone.
Thank you, sir.
Good afternoon. Appreciating your patience.
Rupam Kapoor, Madam, Professor Rupam Kapoor, President.
Yes, Professor Rupam, ma'am, good evening.
Namaskar, sir. Namaskar. Been a wonderful 3 days.
And
Dr. Yashpal, we have Dr. Dargan with us from England.
Oh, that's really good. Yeah, I could read and I could see also. And good evening, sir, from India.
Good evening, sir.
How are you,
Dr. Yashpal?
Sir, I'm all good, sir.
Great to see you.
Sir, we have been a great admirer of you, sir, and it's been inspiring seeing you, sir.
Thank you so much. Hello?
Sir?
Yeah, sir.
Yashpal, sir, he's the youngest and smartest psychologist from Patiala.
Don't say this thing.
Sir, the best thing which I have always been maintaining and telling, sir, I believe all these mycologists, I mean, our senior mycologists and contemporary and our young budding mycologists, sir, they all have those properties of humor and also humility in them. And I think that is the best definition, sir, we have in mycologists.
My
Life is
smiling
sir,
also.
actually life is what this is.
Yes.
One should
Yes, sir.
enjoy that every moment of the life like anything. Of course.
Absolutely, sir. Absolutely. And
Professionally,
great efforts, sir. Great efforts, sir. ਤੁਸੀਂ ਜਿਹੜੇ ਪੌਦੇ ਲਾਏ ਹੋਏ ਨੇ sir, Professor Manu Ruchi and Professor Dr. Avneet and all, they have been, sir, wonderful people and they are doing wonderful job.
ਸਾਰੇ ਸਾਡੇ ਜੈਮਸ ਹੈ, ਜੈਮਸ ਜਿਹੜੇ ਅਸੀਂ ਉੱਥੇ ਰੱਖੇ ਹੋਏ ਹੈ.
Absolutely, sir. Absolutely. That's really
we are
admirable,
very
sir.
happy. And
Sir, we wish you a
especially
very good health. We wish you a very good health and just keep blessing all of us, sir.
Thank you, sir. So nice of you. Life is like that. It will go. Perhaps we don't know what is going in the next for the offing.
All is good, sir. All is good and all would be good, sir.
Thank you, sir. Thank you, sir. Thank you.
Thank you. Thank you, everyone.
और मनुची जी की कर रहे हो? मैं ਹੁਣ सोच रहा भी मैं अपने आप चाह ਇੱਥੇ ਪੀ ਲਾਂ। मैं क्या टी ब्रेक होगी, मैं ਹੁਣ ਟੀ ਆਪਣੀ ਤੁਹਾਡੇ ਨਾਲ ਜੁਆਇਨ ਕਰ ਲੈਣਾ। ਹਾਂ ਇੱਥੇ ਤਾਂ ਭਾਈ ਆਪ ਬਣਾਣੀ ਪੈਣੀ ਹੈ।
ਜੀ ਜੀ ਅਸੀਂ ਡਾਕਟਰ ਸਾਹਿਬ ਅਸੀਂ ਵੀ ਕੱਲੇ ਬੈਠੇ ਆ ਕੱਲੇ ਬੈਠੇ।
ਹਾਂ ਨਹੀਂ ਚਲੋ ਠੀਕ ਹੈ, ਵਧੀਆ, ਬਹੁਤ ਅੱਛਾ ਲੱਗ ਰਿਹਾ ਤੇ ਇਟ ਇਜ਼ ਵੰਡਰਫੁਲ। ਮੈਨੂੰ ਮੌਕਾ ਮਿਲ ਗਿਆ ਤੁਹਾਨੂੰ ਜੁਆਇਨ ਕਰਨ ਦਾ, ਨਾਲੇ ਸਾਰਿਆਂ ਨੂੰ ਤੁਹਾਨੂੰ ਦੇਖਣ ਦਾ ਮੌਕਾ ਮਿਲ ਗਿਆ, ਸਭ ਤੋਂ ਵੱਡੀ ਗੱਲ ਇਹ ਹੈ।
Let me share an incident. When Dr. Dargan was to present his presidential address at Bangalore, the conference was hosted by Professor J. Savitha. It was, I think, January last 2006, and my marriage was scheduled on 19th of February 2006. Few days before, I was, we all were Dr. Dargan, Mrs. Dargan, whom we miss a lot, Dr. Gurpal, Mrs. Gurpal. We were traveling in Karnataka Express all the way from Delhi to Bengaluru. And I still cherish that moment. And the signal which was given to my in-laws is he's such a serious guy that even before few days earlier his marriage, he's on to some conference. And it was all because of Dr. Dargan's guidance. And he was my PhD co-supervisor.
And Avneet, what about your wedding dress which you bought?
Sorry, ma'am?
The coat pant and the wedding dress which you bought?
Yeah, when we were back from Bangalore to Delhi, I could make my shopping of my wedding day suit from Delhi on way back from Bangalore. And the kind of guidance,
that suits reminds again, Avneet, that suit will remain you reminding mycology as well.
Absolutely, sir. Absolutely. It is related with MSI more precisely, sir.
Yeah, that's wonderful. So nice of you all. It was really wonderful to see you all. And I still see that I am still the same person with you there, though the time has left me a little beyond that, but still I still exist there, I suppose, every time. Thank you so much.
Sir, we have been cultured by you people. How can you ever think of in this manner? You're always there with us. You are the guiding force behind
always
all
tell
this.
everyone,
We are able to deliver at this
meets
moment
me,
in our life.
Whosoever meets me, Manruchi, I always tell them that this is we have the only place, touch wood, every faculty member, whenever he goes back to his department, he ever feels so great and part of the department, never any bit of sourness or anything one feels there. So that is the great thing which we all inculcate, but that is what we inherited from all of us. I think this is the legacy which will continue. You people are carrying it so nicely. Thank you. Thank you. Dr. Atri, where is Dr. Atri?
Sat Shri Akal, sir.
Are you online? Sat Shri Akal. ਤੁਸੀਂ ਸੋਲਨ ਬੈਠੇ ਹੋ ਕਿ ਪਟਿਆਲੇ ਆਏ ਹੋਏ ਹੋ?
ਪਟਿਆਲੇ ਮੈਨੂੰ ਕੱਲੇ ਕਮਰੇ 'ਚ ਬਿਠਾਇਆ ਜੀ ਇਹਨਾਂ ਨੇ, ਨਾ ਅੱਗੇ ਨਾ ਪਿੱਛੇ ਕੋਈ ਨਾਲ।
Good.
ਸਰ ਡਰਦਾ ਵੈਸੇ ਹੀ ਨਹੀਂ ਕੋਈ ਕਮਰੇ 'ਚ ਜਾਂਦਾ ਜੀ।
ਕਿਉਂ ਡਰ? ਐਸੀ ਕੋਈ ਗੱਲ ਨਹੀਂ। ਮੈਨੂੰ ਇੱਥੋਂ ਆਣਾ ਔਖਾ ਹੋਇਆ ਪਿਆ ਸਾਰਾ ਕੁਆਰੰਟਾਈਨ ਕੀਤਾ ਪਿਆ
Yeah, true.
I was scheduled to come on 19th,
Yeah.
but anyhow, now I am coming on 6th, but still there are certain question marks by the way traveling. We have applied to the home department still, but they express me how they leave me because it's the lockdown chal raha na bada. You
Take
can't
care,
move
sir.
out of your
Okay,
house.
just travel safe if you have to. Otherwise, stay where you are. Because with this age and everything, you have to be very careful.
If they will allow me to stay, only then I can stay. Otherwise, my 6 months period is almost over now.
Okay.
Okay. Doesn't matter. It's good. I have joined you. Thank you.
Waiting for you to come back, sir.
Take care. I will join you again after, I think it is at 4 o'clock or 4:30.
That's 4:30, sir. It's almost 4:30. Another 2-3 minutes to go. 4 minutes to go.
That's a well-dig tree. ਇਸ ਤੋਂ ਬਾਅਦ ਤੁਸੀਂ
ਹਲੇ ਜਾਓ ਨਾ ਸਾਡੇ ਨਾਲ ਵੀ ਗੱਲ ਕਰ ਲਓ
ਮੈਂ ਕਿਤੇ ਨਹੀਂ ਰਹਿੰਦਾ ਜੀ ਹਾਂਜੀ ਲਖਨਪਾਲ ਜੀ ਕੀ ਹਾਲ ਚਾਲ
ਮੈਂ ਕਿਹਾ ਹਲੇ ਜਾਓ ਨਾ ਰਹਿਣ ਦਿਓ ਤੁਸੀਂ ਗੱਲ ਕਰੋ
ਮੈਂ ਜਾਂਦਾ ਨਹੀਂ ਕਿਤੇ ਵੀ ਮੈਂ ਇੱਥੇ ਬੈਠਾ ਮੈਂ ਅੱਜ ਪੂਰਾ ਇਸੇ ਕਰਕੇ ਬੈਠਾ ਤੁਹਾਡੇ ਕੋਲ ਹਾਂ ਨੋ ਪ੍ਰੋਬਲਮ ਹੋਰ ਸਰ ਕਿਦਾਂ ਸਿਹਤ ਕਿਦਾਂ ਚੱਲ ਰਹੀ ਹੈ
ਠੀਕ ਹੈ ਜੀ ਠੀਕ ਹੈ
ਗੁੱਡ
ਗੁਰਪਾਲ ਨੇ ਦੱਸਿਆ ਸੀ ਕਿ ਫਿਰ ਉਹ ਲੋਕਡਾਊਨ ਹੋ ਗਿਆ
ਉਹ ਲੋਕਡਾਊਨ ਕਰਕੇ ਨਾ ਫਿਰ ਮੇਰੇ ਸਾਰਾ ਪ੍ਰੋਗਰਾਮ ਖਰਾਬ ਹੋ ਰਿਹਾ ਕੱਲ ਬੇਟੇ ਨੇ ਫਿਰ ਹੋਮ ਡਿਪਾਰਟਮੈਂਟ ਨੂੰ ਲਿਖਿਆ ਵੀ ਟੂ ਐਕਸਟੈਂਡ ਮਾਈ ਸਟੇ ਤੇ ਆਈ ਡੋਂਟ ਥਿੰਕ ਪਤਾ ਨਹੀਂ ਉਹਨੂੰ ਜਵਾਬ ਕੀ ਦਿੰਦੇ ਆ ਵਟਐਵਰ ਬਟ ਸਟਿਲ ਇਫ ਦੇ ਨੋਟ ਅਲਾਉ ਮੀ ਆਈ ਵਿਲ ਬੀ ਕਮਿੰਗ ਬੈਕ ਤੇ ਕੋਈ ਨਹੀਂ ਉੱਥੇ ਥੋੜਾ ਜਿਹਾ ਹੈਜ਼ਲਸ ਹੈ ਦੇਰ ਆਰ ਸਰਟਨ ਹੈਜ਼ਲਸ ਐਟ 에ਅਰਪੋਰਟ ਨਾਓ ਦੇ ਫਸਟ ਵੀ ਹੈਵ ਟੂ ਗੈਟ tested here, then we have to get tested at the airport, then at home 3 times they will test, then the things will they will release. And उन्होंने 6 घंटे उसे एयरपोर्ट से बिठाना जिन्ना जे टेस्ट दी रिपोर्ट नहीं आंदी
अच्छा
that is difficult little
हाँ वो सेकंड स्ट्रेन आ गया ना इस करके प्रॉब्लम हो
ਉਹ ਸਟ੍ਰੇਨ ਕਰਕੇ ਨਾ ਜਿਹੜੇ ਯੂਕੇ ਤੋਂ ਪੈਸੇਂਜਰਸ ਹੈ ਨਾ ਕੋਈ ਵੀ ਦੇ ਆਰ ਨਾਟ ਅਲਾਉਡ ਟੂ ਲੀਵ ਆਊਟਸਾਈਡ ਦਾ 에ਅਰਪੋਰਟ ਉਹਨਾਂ ਨੇ ਉੱਥੇ ਲਾਉਂਜ ਬਣਾਤਾ ਦੇ ਵਿਲ ਆਸਕ ਯੂ ਟੂ ਸਿਟ देयर ਉੱਥੇ ਟੈਸਟ ਕਰਨਗੇ ਰਿਪੋਰਟ ਆਏਗੀ ਅਗਰ ਨੈਗੇਟਿਵ ਰਿਪੋਰਟ ਆਉਂਦੀ ਹੈ ਦੈਨ ਦੇ ਵਿਲ ਰਿਲੀਜ਼ ਯੂ ਅਦਰ ਜੇ ਪੋਜ਼ਿਟਿਵ ਆਉਂਦੀ ਹੈ ਤੁਹਾਨੂੰ ਉੱਥੇ ਕੁਆਰੰਟਾਈਨ ਚ ਲੈ ਜਾਣਗੇ ਕਿਤੇ ਦੈਟ ਇਜ਼ ਬੰਦਾ ਡਰਦਾ ਤਾਂ ਰਹਿੰਦਾ ਹੀ ਹੈ ਨਾ
ਹਾਂ ਬਿਲਕੁਲ ਡਰ ਰਹਿੰਦਾ ਹੈ हाँ कह नहीं सकते ना नजर तो आंदा नहीं
नाले की पता जी नाले अगे है ना यू आर ट्रैवलिंग अलोन मुश्किल हो जाती चलो
हाँ है जी
ठीक है सर और मर्दुला की हाल चाल मैडम दा
ठीक है
वो आए होंगे नाल के नहीं
your call was put on mute
नहीं शिमला ही है जी कोई नहीं है
शिमले तो ही बोल रहे हो
शिमले तो अलग अलग ही नहीं जी हर कोई अपनी जगह तो ही बोल रहा
हाँ सारी जगह तो ही बोल रहे मैं देख रहा थी तो मैं कहा जेडे क्लोज है शायद वो तो आ गया हुन बाकी तो सारी जगह से बोल रहे
नहीं जी वो तो हर कोई मेरे ख्याल अपने अपने कमरे से बोल रहा है पटियाला भी ये 2-3 बंदे नहीं करते मनरुचि अवनीश मुन्नी ये सारे ने बाकी
अपने अपने कंप्यूटਰਾਂ ਤੇ ਬੈਠੇ ਸਾਰੇ
ਡਾਕਟਰ ਪਟਿਆਲਾ ਗਏ ਨੇ ਠੀਕ ਪਟਿਆਲਾ ਪਹੁੰਚ
ਗਏ ਨੇਚਰਲੀ ਚਲੋ ਠੀਕ ਹੈ ਵਧੀਆ
we would have loved to host this in an offline mode
ਮਜ਼ਾ ਆਪਣਾ ਹੀ ਹੈ hello Dr. Raman hello Dr. Raman how are you
yeah good madam good sir namaste sir
good how is Chennai
good sir and the Punjabi University Botany Department they made a breakthrough in the history of MSI.
Good
conference excellently they organized under the leadership of Professor Munrochi Kar.
Yeah really sir you have already seen our culture developing here. They are very hard working people and I think everybody's participation, you people coordinate so much. It is all your coordination, because you are steering MSI, and it was a long time. It was in Chennai.
Yes,
so
sir.
that roots are there. I am still remember. I was a founder member of the society when it was founded. I was there during that time also, and big people, Alexopoulos, and all those people were there.
Yes, sir.
Yeah, that was nice. Still legacy is going on.
Secretary for past 10 years, sir.
Yeah.
10 years I am continuing as secretary.
Yeah, I know.
I am doing good for the society.
You are as young as you were 10 years back, as young, as agile, as energetic.
There's no, this is something, I think the fungi gives some sort of potential energy. That is why.
Professor Lakanpal's Ganoderma, Rishi mushrooms are
even the names of all these fungi, I think they are giving off enough energy to be like that.
Yes,
but
sir.
I don't find change in everyone, not only you or Dr. Atri. I have taught them as a student, but I still find them the same way.
Yes, sir.
Yeah.
Thank you, sir.
Atri did a very good exercise.
Yeah.
As the editor-in-chief.
Yeah, that is very good.
And botany department, Punjabi University, in total.
Yeah.
Individual cannot do anything, sir. It is the department makes the work, basically. Department,
it is the best wishes of every corner. That is why things are doing so nicely there.
Everything.
Now, is the vice president, sir.
Yes, sir.
He has been elected recently as vice president.
Who won? Whose name you are telling?
Munraj, Professor Munraj.
Good.
Recently, he has elected as vice president.
Sir, we have with us Professor M. Sharma, a very senior teacher. Dr. Dargan is there from all the way from UK.
Namaskar, sir.
Heartiest congratulations on your publications, first of all. You are not audible. ਤੁਹਾਡੀ ਆਵਾਜ਼ ਨਹੀਂ ਆ ਰਹੀ।
ਸ਼ਰਮਾ ਜੀ ਨੂੰ ਅਨਮਿਊਟ ਕਰੋ।
Sir, please. Ma'am, ਇੱਧਰ ਜੀ।
Actually, sir is sitting with us in the computer lab only upstairs.
Good.
He's just reached up.
Yeah.
Ma'am is also there.
Good. Where is ma'am?
There she is, sir. How sweet.
Anji. Namaskar ji, namaskar.
Camera is there. That's how it.
ਮੈਂ UK ਚ ਇੰਗਲੈਂਡ ਤੋਂ ਬੋਲ ਰਿਹਾ ਤੇ ਬਹੁਤ ਵਧੀਆ ਲੱਗਿਆ ਜੀ ਤੁਹਾਨੂੰ ਬਹੁਤ ਵਧਾਈਆਂ ਹਾਰਟੀਐਸਟ ਕੰਗ੍ਰੈਚੁਲੇਸ਼ਨਸ ਬਹੁਤ ਅੱਛਾ ਸ਼ਰਮਾ ਜੀ ਦਾ lifelong ਕੰਮ ਸੀ
congratulations ਲੈਣ ਵਾਸਤੇ ਹੀ ਬੈਠੀ ਸੀ ਅਸਲ ਚ
we really appreciate, ma'am has a big contribution in the publication, beautiful one which has come out, which will be released today. She has a big contribution to it. Hats off to you, ma'am. Thank you.
So no doubt about it.
See, जो कुछ भी सी सबने कुछ ना कुछ अपना participation, encouragement, moral support बहुत सी. So he could do it.
नहीं पर देखो जी, you have proved true the हर success ਦੇ ਪਿੱਛੇ lady ਦਾ ਹੱਥ ਹੁੰਦਾ ਹੈ.
अच्छा डॉक्टर, continue with your.
Okay जी.
Okay, thank you.
So with the arrival of our chief guest of the day, we can start with the valedictory session of this 3-day conference. हाँ जी डॉक्टर
नमस्कार सर नमस्कार बहुत
Thank you so much for your sweet words.
Namaskar.
So first of all, a hearty welcome to the honorable chief guest, Professor Mangaleshwar Sharma.
Namaskar, sir.
Who's a senior plant taxonomist, immense contribution. We will go through his contributions.
Raman namaskar kar. Raman sir namaskar kar rahe Sharma ji nu.
Ji sir.
Yeah, namaskar. Welcome to the validatory function, sir.
Thank you so much. Thank you so much.
Yes,
sir. We have Mrs. Sharma also with us.
Good evening, sir.
To chair this session, we have with us Honorable President of the Mycological Society of India, Professor Rupam Kapoor from University of Delhi.
Namaskar, sir. Namaskar, Professor Sharma. We welcome you to this final valedictory session of the conference.
So we have with us the Honorable Secretary of MSI, Professor N. Raman, Center for Advanced Studies in Botany, University of Madras, Chennai. We have so many senior mycologists with us from all over the country, and I heartily welcome you all to this valedictory session. Now, to start with the formal proceedings, may I request the Honorable Convener of the conference, Professor Manruchi Kaurji, to have the formal welcome address. Unmute kar lo.
Unmute karo, unmute. Ask Madam Anruchi to unmute.
It is my very pleasant duty to welcome you all to the valedictory function of this virtual conference. First of all, I would welcome my revered teacher, Professor M. Sharma ji, who is the chief guest for today. And being a student, I know how immaculately he works with his angiosperm taxonomy. I welcome you, sir, to the valedictory function and for gracing the occasion. Our Vice Chancellor could not make it to the valedictory function, but she has sent her best wishes. I welcome a very graceful personality in the form of the President MSI, Professor Rupam Kapoorji. I welcome a very energetic Secretary of MSI, Professor N. Raman, all the office bearers and the executive members of the Mycological Society of India. A very warm welcome to all the delegates who have made this conference a real big success. As far as I am concerned, I think we have done it to the best of our capacity. I welcome Professor DJ Bhagyaraj, Dr. Atri, Professor C. Manorachari ji, Professor T. N. Lakhanpal, Dr. Rodriguez, Professor DJ Bhatt, and a special welcome to Professor Ru, Dr. Sabade, Dr. Langer, and Dr. L.M. Sureshji for gracing the occasion and making it a success. We really are indebted to them rather for giving us such good lectures and a good exposure to the young budding mycologists who have been attending this conference with full zest and zeal. And I welcome my entire team, the organizing committee, Professor Khattar, Professor T.P. Singh, Dr. Manish, Dr. Geetika, Dr. Arneet Garewal, Dr. Avneet Pal Singh, the organizing secretary. I welcome the press and media. I welcome you all from the core of my heart to this valedictory function. I would like to acknowledge all the delegates and my supporting team who have worked really hard to make this conference a truly successful one. Welcome one and all. Thank you.
Thank you very much, ma'am. Thank you very much for the formal welcome. May I request Professor D.P. Singh ji to kindly introduce the honorable audience, our great chief guest, our great teacher, a great researcher, a great taxonomist indeed, to kindly introduce Professor M. Sharma to the guests and delegates. Dr. D.P. Singh.
Thank you, Dr. Amnit Palji, for giving me a chance again to introduce my revered teacher, Professor M. Sharma. It gives me an immense pleasure to introduce Professor M. Sharma on the auspicious occasion of book release during National Conference on Biodiversity and Biotechnology of Panjai and 47th Annual Meeting of MSI. Along with this book, Professor M. Sharma, a professor of botany, Punjab University, Patiala, was born on June 6, 1940 in Rawalpindi, now in Pakistan, did his MSc in 1964 from Punjab University, Chandigarh. The inspiration provided by teachers during this study and their love for nature and plants brought him more and more in contact with floristic studies, an interest which became the professional hallmark of his subsequent career. After passing MSc, Professor Sharma served for a brief period at Government College, Bathinda, then at DAV College, Chandigarh, and then at Punjab Agriculture University, Ludhiana. Later, he joined Punjab University, Patiala as a curator of Museum and Herbarium, Department of Botany, Punjab University, Patiala. To enrich herbarium and museum of our department, Professor Sharma sir has undertaken extensive plant collection trips to eastern and western Himalayas, whole of Punjab state and Chandigarh, Mount Abu, Panchmadi Hills, etc. While doing all these collections, along with normal teaching assignment, he obtained his PhD in 1981 under the supervision of Professor SSB. He was promoted to reader in 1982 and became professor in 1990. He always accompanied students to various hill stations for plant collection. During excursions, sir had been a very keen field observer and provided guidance and help to both the postgraduate students and research scholars in the identification of their plant material. Besides teaching assignment, Professor Sharma held many official positions in the university, such as head department of botany, officiating head department of human biology, dean, faculty of life sciences, member of academic council and undergraduate and postgraduate board of studies, Punjab University, Patiala, and member of syndicate and senate, Punjab University, Patiala. Besides this, Professor Sharma sir become a fellow of many prestigious institutions such as Indian Association for Angiosperm Taxonomy, Indian Botanical Society, Linnean Society of London, Indian Association of Plant Taxonomy, Bombay Natural History Society, life member of Punjab Academy of Sciences, and member of various boards such as Biodiversity Conservation Committee of Punjab, State Level Biodiversity Strategy and Action Plan Committee, Consultant Higher Plant Biodiversity, Punjab State Council for Science and Technology, Chandigarh. Besides this, Professor Sharma has received many scientific awards. Prominent awards include Professor Y.D. Tyagi Gold Medal by Indian Association of Angiosperm Taxonomy, Calicut, awarded in 2011. H.S. Santpau Medal by Association of Plant Taxonomy, Dehradun. For over 49 years, Professor Sharma has been studying the flora of Chandigarh, Punjab state, and Himachal Pradesh. He has reported about a dozen new introductions in the Indian flora. Besides a couple of new taxa, his primary contribution have been focused on three districts: flora, of Kullu, Sirmaur, and Chamba of Himachal Pradesh, and flora of Patiala. Besides Punjab plants checklist, flora of semi-arid Punjab, India, and flora of Ropur district are his other noteworthy contribution. Professor Sharma has published 9 books and 150 research papers. Today, along with this, as a chief guest of this valedictory functions, we are also going to release the 3-volume book entitled Vascular Flora of Punjab and Chandigarh. At the end, I congratulated to my teacher and man of taxonomy, Professor Sharma, for presiding of this valedictory function, as well as for getting his 3-volume of Vascular Flora of Punjab and Chandigarh released at this occasion. Thank you. Thank you very much, sir. Thank you. Over to Dr. Avneet Palji.
Thank you very much, sir, for a wonderful introduction. So it is an absolute delight. We are fortunate enough to have a man of taxonomy with us, though the field is angiosperms, but it is absolute treat to learn taxonomy from teachers like Dr. M. Sharma. When I entered for my MSc in Department of Botany 2000, the same year Professor Sharma retired, so we missed that chance, but he is always there to guide us. He guides us how to practice taxonomy. So he is a source for the taxonomy. He's an inspirational source for all the taxonomists. Thank you very much, sir. Now, before proceeding further, let me share with the audience the summary of this conference. After this, we will formally release the flora, which is published by Professor M. Sharma. Now, just to familiarize the audience, as we all know, this 3-day virtual national conference on biodiversity and biotechnology of fungi, along with 47th annual meeting of Mycological Society of India, it was organized at Department of Botany, Punjabi University, Patiala, from 22nd of February to 24th of February. There were 4 themes: fungal diversity, systematics, evolution and conservation, fungal physiology, biotechnology and applications, mycorrhiza and endophytes, medical mycology, plant diseases and management. So the brochure was circulated and there was very encouraging response from the mycologists, not only from the country, but even from abroad. The conference was virtually inaugurated on 22nd of February by Professor G.S. Batra, Honorable Dean Research, Punjabi University, Patiala. Professor Rupam Kapoor, President, Mycological Society of India from University of Delhi, delivered her presidential address. Professor D.J. Bhatt from University of Goa was conferred with the Lifetime Achievement Award, and we were fortunate enough to be a part, to be company to such a great address in the form of Lifetime Achievement Award lecture. Besides this, 6 memorial award lectures, they were presented, they were delivered by Professor Sarita Nazarat, Professor Naveen Kango, Professor Rupam Kapoor, Dr. V. Sarma, Dr. Kharwar. And we were fortunate to have 7 fellows who have been conferred with MSI Fellow for the year 2020, including the Doyen of Mycology, Professor C. Manorachari, Professor Sanjeeva Naika, Professor Pradeep Verma, Dr. Mahavir Prasad, Professor Raj Kumar Salar, Professor Manduchi Kaur, and Professor D.V. Hande. So we congratulate the achievers and the fellow awardees. Large number of scientists from different universities, research institutes from all over the world, I must use the word world, they participated in the online deliberations. Some of the prominent delegates to name, they are Professor David Hibbert from Clark University, Professor Ewald Langer from Castle University, Dr. Roo Wendergrift from University of Oregon, Professor Rex B. Sadaba from University of Philippines, Dr. L.M. Suresh from International Maize and Wheat Improvement Center, Kenya. Besides these, we had with us Professor N. Raman from Chennai, the senior mycologist, Professor T.N. Lakhanpal from Shimla, Dr. D.J. Bhagyaraj from Bengaluru, Professor Manorachari from Osmania University, Professor B.F. Rodrigues from Goa, Professor Krishnendu Acharya from Kolkata, Professor R.N. Kharwar from Varanasi, Professor Yashpal Sharma from Jammu, Dr. Sunil Deshmukh from Mumbai, Professor M. Sudhakar Reddy from Thapar University, Patiala, and of course, the list goes on. And we were fortunate enough to be joined by the members of Botany Department, Punjabi University, Patiala, in the form of Professor M. Sharma, Professor J.S. Dargan, Professor N.S. Atri, Professor Gurpal Singh Dhingra, who are with us, and even the online proceedings, they were joined in by the teachers of Botany Department, Professor R.C. Gupta, Professor V.K. Singhal, Professor Amarjeet Singh Ji. They were also with us either through YouTube link or through the Zoom link. Now, it is a matter of honor that besides the presidential award, presidential lecture, memorial awards, and fellow awards, the conference also witnessed 11 lead lectures. And in addition to this, we had 35 oral presentations and 42 poster presentations. They were also made. So this is what we did in the form of 11 sessions spread over these 3 days. And the deliberations, they were made through the platform Zoom and YouTube links. And finally, we have come to this concluding part, the valedictory session. So that is all what we had during these 3 days of virtual national conference. Now, may I request the honorable convener, the co-convener, to accompany Professor M. Sharma ji for the release of this book.
Mrs. Sharma also.
Yeah, sorry, I'm extremely sorry. May I invite Mrs. Sharma also to kindly join in, without whose help this would not have been possible. I'm extremely
Yes. Last note.
Ma'am Sharma, please. हाँ जी थोड़ा जा पीछे वो तो सर ऊपर सर वो चल वो वखरा चलेगा ये वखरा मैम प्लीज जो खोलो So this in the form of 3 volumes. The title is, Dr. Manish, kindly play it. Yeah, that's what we have. It is entitled Vascular Flora of Punjab and Chandigarh in the form of 3 volumes.
Yes, congratulations.
Thank you.
Congratulations, Professor Sharma.
Congratulations, sir.
Congratulations, sir.
Congratulations to Professor M. Sharma, sir. Sir, we were lucky to have this copy.
Thanks a lot.
Sir, congratulations, and I'm thankful to Sharma Saab for having compiled this wonderful, book series.
Dr. Manish, kindly mute your laptop. Dr. Manish, kindly.
So we received these copies yesterday itself, and we are lucky that you are inaugurating, and it's really a wonderful compilation, sir. And it would go a long way in helping our students who are working in the field of ethnobotany and taxonomy. Thank you so much.
May I request?
Thank you so much.
May I request Professor M. Sharma to kindly share his feelings with us. Dr. M. Sharma.
First of all, I am fortunate to be amongst you, esteemed colleagues. It is indeed a pleasure that I am becoming a part of this conference, maybe indirectly. So thank you so much for inviting me here. Now, so far as the books are concerned, I'll be very brief in telling what they really are.
Please.
Indeed, it took me about 57 years. And probably the beginning was not 57 years. It started about 2 years earlier when I was the BSc student. My teacher, Professor Jagdish Singh Ji, I gave him the credit. He inculcated the spirit of plant taxonomy. He himself was a student of Dr. R.R. Stewart. Dr. R.R. Stewart is very well known internationally. Now, of course, he is not there. And he was such a big taxonomist that his annotated catalog of vascular plants of Punjab, Pakistan, it made the starting point for writing the flora of Pakistan. So such a big scientist, and then my teacher, Professor Jagdish Singh Ji. Why I am specifically referring his name is, see, he was one of the rare teachers, and you will appreciate why I have used the word rare. He was head of the department here. I was the student in BSc, Ludhiana, Government College, Ludhiana. So what he did was, all the plants which we were supposed to do in practical, he grew them in the botanical garden. But more importantly, what is most important is that before he will take a family description of the family within in the classroom, all of us were supposed to go to the botanical garden along with him. He will show the characters there. They were in our mind. Then we'll rush back to the class, and then he will tell us the character. The result was that because we had already seen the characters, so we will never forget those characters. So that is the person from where we started. And then, of course, MSc, I was given the dissertation on flora of Chandigarh and its neighborhood. Grateful to my teacher, Professor O.P. Sharma. Then I did my PhD here, right in Punjab University, Patiala. My heartfelt thanks to Professor S.S. Beer, my guide. And beside these persons, out of the 36 years of career, 32 years I have spent in this Punjab University, Patiala. So I owe these books to my teachers, to my colleagues, to my students. All of them, they have contributed. And one special mention regarding the post-retirement phase. That is very important because that is a phase where you have to face certain challenges. And in that, I must most humbly admit that some of the teachers who are the head of the departments here, Dr. Atri is there, Dr. Dargan, and Dr. Khattar, Mandruti, and some other teachers also who are not here. They helped me a lot. It is only due to their cooperation and other things that I have come to this day. And as I told you, my students, my colleagues, all of them, they are responsible. Now, such jobs, they are not done by one person. You need lot of collaboration and cooperation, moral support, even maybe the material support from the various persons. So I will end this with some small communication which I received from Dr. M. Sanjappa. Most of my colleagues, they must be knowing Dr. M. Sanjappa is a retired Director of Botanical Survey of India. Yes. Just this morning, I received a small message, and I am passing on this as a tribute to all my colleagues, all my students, and above all, my family, without whom it was not possible. We generally don't appreciate the efforts which our spouses, they put into bringing us to a particular level. You know, as you can see, this is not a work of 9 to 5 p.m. And the whole of my family, my wife in particular, she took the entire responsibility to give me lots and lots of time and managing whatever little bit resources we could provide to the families. So my hats off to my family along with the others. This is a crucial contribution by her and my children. Now I was talking to you about this, just one more minute. I received a sweet message from Dr. Sanjappa. And as a tribute, I am passing on to all of you, the persons who helped me in bringing out this book. It will hardly take 1 minute. Please listen to it. And this is a tribute to the persons who helped me to bring about this. Dear Professor Sharma Saab, greetings from Bengaluru. At the outset, congratulations on bringing out this mammoth publication. It is so nice to see you and Madam at all IAAT meetings. We have been attending them without break since the last 13, 14 years. And that is where the credit to Madam goes further. You are a wonderful example for all taxonomists to follow. Dedication, perseverance, and hard work always pay. You are a quiet personification of all this. With warm regards to you and Madam M. Sanjappa, PhD, FNA, FASC, FNASC, former Director, BSI, CSIR, Emeritus Scientist, INSA Senior Scientist, and presently he is at Mahatma Gandhi Botanical Garden working as the partner. So thank you, Dr. Sanjappa, thank you, my colleagues, and every other person. It is a tribute not to M. Sharma, but to all of you. Grateful, indeed grateful to all of you for all this help. Thank you so much, and thank you.
शर्मा जी मेरे भी थैंक्स कर दियो
डॉक्टर लखनपाल
जी जी
लेफ्ट हैंड कॉर्नर लेफ्ट कॉर्नर
लेफ्ट
शर्मा जी
To be honest,
बहुत बहुत मैं कह मेरे भी थैंक्स कर दो
meet me here. So I was going to bang you for it, but then I think you are on this virtual, so sorry, can't do anything. But thank you so much for your sweet words.
Sharma, ਦੋਸਤਾਂ ਨੇ ਥੈਂਕਸ ਦੀ ਲੋੜ ਨਹੀਂ ਹੁੰਦੀ ਦੋਸਤ ਤਾਂ ਦਿਲ ਚ ਰਹਿੰਦੇ ਆ ਹਮੇਸ਼ਾ ਨਾਲ ਰਹਿੰਦੇ ਆ
Dr. Lakhanpal and we were together rather at Punjab University, Chandigarh during 1963-64. And then since then we are together and but I can just say from the core of my heart, the inspiration, the help I got from him and his wife also. She is also very encouraging. She will always encourage. So thank you, Tejna. Thank you so much for being here. Thank you.
Thank you so much.
सवारी याद आ रही साइकिल दी साइकिल दी सवारी याद आ रही ਤੁਹਾਡੇ ਨਾਲ
we could infer the kind of relation which Dr. Sharma, Dr. Lakhanpal, Dr. Dargan must be sharing.
Yes, yes.
From the kind of we are really blessed to have you all with us, sir.
ਸ਼ਰਮਾ ਜੀ ਨੂੰ ਸ਼ਰਮਾ ਜੀ ਦੀ ਸਾਈਕਲ ਦੀ ਬੈਕ ਸੀਟ ਤੇ ਬੈਠ ਕੇ ਜਾਂਦਾ ਸੀ ਕਲੈਕਸ਼ਨ ਤੇ ਇਹਨਾਂ ਨਾਲ
Great,
So
sir.
sweet. Great.
So
great.
once again, we congratulate Professor M. Sharma, the entire family, and indeed the botany family for this magnanimous contribution, for this mammoth contribution to the field of taxonomy.
I again say my department and my family, they are responsible for it. Once again, thanks to all.
Thank you very much, sir. Now I would like to hand over the mic to Honorable Secretary MSI Professor N. Raman for further proceedings. Professor Raman, please.
Yes. Now the fellowship announcement. Before I announce the fellowship, I thank profusely the selection committee, Professor DJ Bhagiraj, Professor Satyanarayana, Dr. S.K. Deshmukh. They made a very good scrutiny, and they have selected 7 members as fellow, and that has been approved by the office bearers and finally by the executive committee as well as the general body meeting. So the first is the Professor C. Manograchari. I would make a record here that at the youngest age, he has sent his nominations obeying the rules of the MSI. So Professor Manogarachari, Professor C. Manogarachari obtained his PhD degree in 1974 from Usmania University and BSc in 1999 from Mysore University. He has been engaged in research and teaching of mycology and plant pathology for well over 5 decades. He did his postdoctoral work with the late Professor Webster, UK, Professor T.D. Goose, USA, and Professor Kiesel, Germany. He was visiting professor of CABI, UK, Wales University, UK, and Texas University, USA. He served Usmania University in different capacities, including Dean, Director, Coordinator for staff, Emeritus Professor in Faculty of Science. He has also served as Vice Chancellor of Oriental University, Indore, Madhya Pradesh. He made outstanding contributions on biodiversity, taxonomy, conservation, and biotechnology of different groups of fungi, plant growth promoting rhizobacteria, myxomyces, and lichens. He has discovered 20 new genera and 82 new species of fungi. He has guided 50 students for PhD, published 40 research papers in international and national journals, besides being editor or author of 30 books, including that of Zicker Publishers USA. His monographs on soil fungi around habitats, and Trichoderma from India are widely referred by researchers. Professor Manogarachari is the recipient of the prestigious awards Dr. J.C. Bose Award by UGC, New Delhi 2001, Dr. Birbal Sahni Medal in 2002, Dr. E.K. Janaki Ammal National Award by Ministry of Environment 2006, and Lifetime Achievement Award by the Indian Botanical Society 2016. Professor Manogarachari is also bestowed with several outstanding awards such as the Best Teacher Award by Government of Andhra Pradesh 1995, Andhra Pradesh Outstanding Scientist Award, Government of AP 2001, Dr. Mundukar Lecture Award by the Indian Phytopathological Society, Dr. N. Prasad Memorial Lecture Award, Dr. Saligaram Sinha Memorial Lecture Award, Nasi 2006, Platinum Jubilee Lecture Award, ISC 2006, Dr. V. Agnihotri Endowment Lecture Award, MSI 2009, etc. He is served as President of Indian Phytopathological Society, Indian Botanical Society, Sectional President of Botany, Indian Science Congress, and Mycological Society of India. He is an elected fellow of National Academy of Sciences, India, National Academy of Agricultural Sciences, Indian Botanical Society, Mycological Society of India, and Indian Phytopathological Society, inspiring teacher, an outstanding researcher, and educationalist. Professor Manogarachari is respected both in India and abroad for his scholarship and dedication to science. The Mycological Society of India is very glad to give the fellow to Professor Manogarachari, and I request the chief guest, Professor Sharma, and also Rupam Kapoor, the President of Mycological Society of India, to join with me to virtually hand over the fellow to Professor Manogarachari. Professor Manogarachari is there?
No. But it is indeed a privilege of all the other members of Mycological Society. Now that Professor Manogarachari is also in their group, they should enjoy the privilege of being the fellow of the society.
Congratulations.
It has.
You are the award is the fellowship is in your Gmail inbox. It will be sent to your by post. The next fellow is Dr. Sanjeeva Nayak. Dr. Sanjeeva Nayak did his MSc from Mangalore University and PhD from Awadh University. He joined CSIR MBRI Lucknow as scientist B in 2001 and currently working as senior principal scientist and head of the lichenology laboratory. He is also professor at the Academy of Science, Scientific Innovation and Research, Ghaziabad. His major area of research is systematic mathematics of lichens. During his 20 years of dedicated service in lichenology, he has discovered 26 taxa as new to science, reported 96 taxa as a new distributional record to India, and proposed one new combination. He has authored over 170 research papers, 22 chapters, and 3 books. He has traveled extensively all over the country for collection of lichens and enriched the herbarium of LWG with thousands of specimens. He has participated in 22nd and 28th Indian Antarctica expedition. He was a part of 29 research projects, of which he was principal investigator in 8 projects. So far, he has supervised 4 PhD students, 3 postdoctoral fellows, and 4 MSc dissertation. He is a fellow of International Society of Environmental Botanists, secretary of Indian Lichenological Society, and life member of 11 other academic associations. Dr. Nayak is deeply involved in popularization of lichenology in India, for which he has organized 2 national conferences and 18 workshops. He has trained over 60 research scholars from various parts of India and provided technical assistance to 20 organizations for lichen study. The Mycological Society of India is very glad to confer fellow to Dr. Sanjeeva Nayak. Congratulations, sir.
Congratulations, Dr. Nayak.
The next fellowship awardee is Professor Pradeep Verma. Professor Pradeep Verma completed his PhD at Sardar Patel University, Gujarat in 2002. Presently, he is a professor at the Department of Microbiology, the Central University of Rajasthan. He has also served as head dean, School of Life Sciences, Central University of Rajasthan. He has been working extensively on the analysis of fungal diversity from various ecological areas and their bioprospecting for different industrial and environmental applications. He has also been working on developing greener approaches for valorization of lignocellulosic biomass, bioremediation of toxic dyes and wastewater, etc. He has been bestowed with several prestigious fellowships and awards such as JSPS Fellow, UNESCO Fellow, and Swedish Society, Sweden, German Science Foundation. He is life member of several research organizations such as Mycological Society of India, Biotechnology Research Society of India, Association of Microbiologists, Indian JSPS Alumni Association, and Society of Biological Chemists. He is also a guest editor and editorial board member of several journals. The Mycological Society is very glad to honor him with the Fellow of Mycological Society of India. Congratulations, sir. Your fellowship will be in the is in your inbox. This will be sent to you by post.
Professor Verma.
The next fellowship awardee is Dr. Mahavir P. Sharma. Dr. Mahavir P. Sharma did his MSc in nematology from Rajasthan Agricultural University, and while working at Cherry, New Delhi, he pursued doctorate degree and awarded PhD in microbiology from Jivaji University, Gwalior. Presently, he is working in ARS as principal scientist, agricultural microbiology at Indian Institute of Soybean Research, Indore. Dr. Sharma has received gold medal award during his master's program and awarded many best paper and post presentation awards during the conferences. He has also been awarded with prestigious DST Crest Postdoctoral Award 2013 by Department of Biotechnology, Government of India, to work in USA on signature fatty acid biomarkers. He has also been awarded many international grants by DST, Government of India, visited Perth, Lisbon, and Oregon for presenting papers, and during July 2015, he was a visitor to Research Institute of Organic Agriculture, Frick, Swiss Federal Institute of Technology, Zurich, Switzerland, for developing linkages for mycorrhizal research and soil nutrients aspect. He has been implemented 5 major research projects funded by DST, DBT, ICR, and also the Agri International companies. He has deposited 15 microbial gene sequences to NCBI and has been supervised 12 MSc and 2 PhD theses. He served as a member of several committees, project of task force committees, etc. The MSI is very glad to honor him with a fellow of Mycological Society of India. Congratulations, sir.
Thank you, sir. Thank you very much, sir.
Congratulations, Dr. Sharma.
Thank you, sir.
The next fellowship awardee is Professor Rajkumar Salar. Professor Rajkumar Salar did his MSc, MPhil, PhD from Kurukshetra University, Kurukshetra. Presently, he is Dean of Faculty of Life Sciences and Chairperson, Department of Biotechnology at Chaudhary Devi Lal University, Sirsa, Haryana. He has worked on various aspects of thermophytic fungi and other microorganisms for the practical application in industrial microbiology, mushroom composting, and biodegradation of xenobiotic compounds. He has identified Alternaria alternata as causal organism of leaf spot disease of Icania somnifera. He has published 4 books from Springer and CRC Press and over 100 research papers in journals of national and international repute. He has also implemented 2 major research projects from EGC and DST and supervised 10 PhD and 14 MPhil and more than 30 MSc theses. Dr. Salar received postdoctoral fellowship from the Ministry of Education, India, in 2010. He was also awarded by Government of Japan to attend international for cultural collection at Tsukuba, Japan. One of his research on stress influenced the increase of phenology content and the radical scavenging capacity of Rhodotorula glutinus, was recognized as the best paper by Springer and was awarded with a medal and citation along with cash prize sponsored by King Abdullah City for Science and Technology, Saudi Arabia. He visited several countries including Norway, Japan, Hungary, and Australia for academic pursuits. The Mycological Society of India is very glad to honor him with a fellow of Mycological Society of India, and it will be sent to you by post. Professor Rajkumar Salar, congratulations.
Congratulations, Professor Rajkumar.
Thank you. Thank you very much. Thank you, everyone.
Congratulations, sir. Congratulations.
Thank you, sir. Thank you.
Then the next fellowship award is none other than the convener of the conference, Professor Munruchikar. Professor Munruchikar did her MSc with gold medal, MPhil and PhD from Punjabi University, Patiala. Presently, she is the professor and head of the department, Punjabi University, Patiala. Along with this, she is the director, UGC, HRTC, and former director, Directorate of International Students, Punjabi University, Patiala. Furthermore, since last 3 decades, she has been actively contributing in the field of mycology through her immense, remarkable, and dedicated work in the field of diversity and systematics of lamellate mushrooms. Her interest also lies on correlating the approach for molecular taxonomy. She has published 70 research papers in various international and national journals of repute. She has also coordinated major research projects from DBT, DSA of UGC and PIST of DSC, etc. He has supervised 8 PhD students, 5 MPhil students, and 8 MSc dissertation students. He is a recipient of Dr. S.K. Som Memorial Award by Mycological Society of India. He has been organizing secretary of the National Conferences held in her department. She is the life member of Botanical Society of India, Mycological Society of India, the Biotech Research Society India, Indian Botanical Association, life member of Punjab Academy of Sciences, and the Indian Science Congress Associations. She has been attending and delivering lectures in various international and national conferences. The Mycological Society is so glad to honor her with the fellowship 2020. Congratulations, madam.
Thank you so much, sir. Thank you.
Congratulations, madam.
Thank you, thank you, ma'am. Thank you.
Congratulations, ma'am.
Thank you, sir.
Thank you.
Thank you, sir. Thank you. It means a lot. Thank you.
Only deserve this.
Thank you.
1 minute, sir. And the last candidate. The next candidate is Professor Dilip B. Ande. Professor Dilip B. Ande did his MSc Botany and PhD from St. Godge Baba Amravati University, Amravati. Presently, he is working as a professor at Department of Botany, Sri Shivaji Science College, Amravati. He has been working on taxonomy of lower fungi. His line of research is fungal secondary metabolite synthesis and analysis. He is also working on delivery of endophytic fungi of medicinal plants of Malkat forest. He worked on biology of fungal diseases of Malkat forest plants. He has worked out the diversity of folicolous fungi and microfungi associated with litter plants from Malkat forest. He also served as a research associate of HRDG sponsored CSIR research project. He has implemented research projects funded by UGC. He has supervised 3 PhD and 2 MPhil students, and 5 PhD students are also working under his supervision. He has presented 60 research papers in national and international conferences. He has organized 2 national, 2 state level conferences, workshops, seminars. He chaired sessions for international conferences, Second World Diversity Congress 2011 at Malaysia. He has contributed chapters in books published by national and international publishers. He has delivered more than 20 guest lectures in various colleges. He is the life member of 12 state, national, and international associations. He has been nominated by Honorable Vice Chancellor as District Coordinator, Amravati District for Intercollegiate Research Festival. The Mycological Society of India is very glad to honor him with the Fellow of Mycological Society of India. Congratulations, Dr. that is
Congratulations, Dr. Devi.
Richly deserved.
Thank you, sir. Thank you so much.
Congratulations, Dr. Hande.
it will be sent to your.
Thank you so much, sir.
The soft copy is already in your.
Thank you, sir.
Congratulations.
The next is the.
Thank you, sir.
Dr. M.J. Tirumalachar Young Scientist Award, the young budding mycologist of India. The best oral presentation for Dr. M.J. Tirumalachar Young Scientist Award. The first prize goes to Uzma Altaf from University of Jammu. Congratulations,
Thank you very much.
Usma.
Congratulations to the guide.
Congratulations, Dr.
Thank you. Thank you so much.
Congratulations, Dr. Sharma.
Award
It's excellent.
prize also.
Thank you all for blessings. Thank you very much.
Good to see that smile and that proud feeling on your face, sir.
Thrill, and I'm very happy to see all senior sirs, and their blessings are immensely being poured on us. So thank you, everyone. It's a great occasion.
Congratulations, Yashpal.
Thank you. But sir, thank you so much. Thank you, Atri sir, and thank you, Lakhanpal sir, Raman sir, everyone, sir. Thanks for blessings. Thank you.
The second prize, it carries only the certificate of Dr. M.J. Tirumalachar Young Scientist Award. It goes to Sudeshna Nandi, University of Calcutta. Congratulations.
Congratulations. An excellent presentation. Congratulations.
So, the certificate.
Congratulations, Dr. Acharya.
Yeah, thank you, ma'am. Thank you.
Acharya.
Thank you, Dr. Acharya.
Congratulations, Dr. Krishnendu.
He is very, he has opted out of that chairmanship.
Yes. Thank you.
The student.
Yes.
Next, the best presentation under Dr. M.J. Tirumalachar Young Scientist Award. The first prize goes to the first prize with 500 rupees cash prize also. Komal Verma. I don't know. I will get the postal address from Dr. Avni Paul.
Double congratulations.
Komal Verma from.
Can't believe it.
Komal Verma.
So good.
Coming to you for a party and relaxing out in Jammu after this conference, sir.
Thank you. Thank you very much, everyone.
Sir, congratulations.
This is unbelievable, incredible. I just can't.
In Jammu University. Yeah. Oh, okay. The examiners have not given me the university. And the next second prize for best poster presentation for Dr. M.J. Tirumalachar Young Scientist Award. Second prize goes to Disha Dasgupta. Disha Dasgupta, congratulations.
Congratulations.
Disha Dasgupta. I will get the information from Dr. Avni Paul Singh. Congratulations for all the young scientists. Scientists. Thank you very much. I have finished the award announcements.
Thank you very much, Dr. Raman. And
You did a great job, Professor Raman.
Yes, sir. Yes, sir.
Big congratulations to Professor Yashpal Sharma for making a double treat.
Oh, double.
To all the achievers.
I'm simply honored and blessed, and thank you very much for thank you everyone. I mean for us it's a learning experience. Thank you so much.
I thank
so much.
chairpersons and co-chairpersons for selecting the award for Tirumalachar Young Scientist Award.
Okay, let's proceed to the next part.
Yes, sir.
Can we have the feedback brief session from the delegates, among delegates? Dr. Bhagyaraj is there, Dr. Bhatt is there. The feedback for this event, sir. The experience.
Yeah, I think it was wonderful. The whole thing was organized so well, and only I missed a part of it because I had to give another talk to a webinar in BHU yesterday. That is how I missed yesterday morning. And otherwise, everything went on very well. Hearty congratulations to the organizers. Very hearty congratulations.
Thank you very much, sir. Dr. Bhatt, please.
Yeah, is truly remarkable proceedings that we conducted last 3 days. I'm more than delighted with the exercise what Professor Avneet and his team and Madam Manruchi Kaur and a whole lot of people from Patiala. You did a great job. And it's not small thing to do this, such an orderly way. Everything went on timely. I literally sat all the 3 days and watched the proceedings. Wonderful papers, both young and middle-aged people, the team. And it's such a remarkable thing. And we are no way small from the people elsewhere who are doing mycology. So each and every paper, probably a few of them requires a little more attention, but in general, I see we have taken from morphology to molecular taxonomy, morphomolecular, whatever said, and such remarkable presentations. And it's such a treat for us to sit at home and watch these proceedings. So once again, I thank Professor Avneet and his team for everything that you have done. And a special thanks to Professor Atri Saab, because I know how much of efforts people have taken for this exercise. And
thank you very much, sir.
And it is a good fortune for me and my wife to have this award taken, Lifetime Achievement Award taken in this conference. So once again, thank you very much.
Thank you very much, sir.
Best wishes to all of you.
Yes, Dr. Shashi Rekha, a moment, please.
Yeah.
Then I'll hand over to you. Dr. Bhatt has been generous enough. Actually, this is the teamwork of the entire team of botany department. We have with us the senior most professor, Professor J.I.S. Khattar, who is always leading from the front, followed by Professor Manruchi Kaur. She is being at the forefront for this conference. We have with us Professor D.P. Singh, everybody. Then we have with us Dr. Manish Kapoor, Dr. Geetika Sarhandi, Dr. Arneet Garewal. So all of them, they were contributing double than the capacity for the last few days, because for us also it was a new experience. Managing the things online, we were totally new to this platform, but with the cooperation, with the kind of responsibility which is shared by all, this event could have been possible. Thank you very much. Dr. Shashi Rekha, please.
Yeah, first of all, I would like to congratulate the entire team of Patiala. Because I'm not naming everyone, I may miss someone, so I'm telling the entire team, including including the teachers, the students, the research scholars, everyone. I'm sure all of them are involved in making of this entire conference, and it was a wonderful experience. Though I missed the first day, I was involved in another conference, national conference, so I missed the first day. But the second 2 days I was there completely throughout the day. And I enjoyed every minute of it, every second of it, and wonderfully organized. I only missed the hospitality. As I just put it in the chat box, I thought I may not be able to speak, but then I put it in the chat box. I really missed the hospitality of Patiala. Punjabi hospitality I miss. That is underlined. I really enjoyed last time when I came. Obviously this time I was able to attend every lecture, which otherwise would have been missed during the physical conference due to parallel sessions. we do miss certain lectures. This time we didn't miss those. I mean, we could attend each and every of the sessions. You know, that was one advantage of this online. But obviously, 3 days it continuously. Even then, maybe there would have been more presentations. But whatever said and done, it was a wonderfully organized conference, and congratulations all again. And
thank you, ma'am.
I enjoyed it. Thank
We
you.
have with us Dr. Lakhan Pal. Sir.
I have no words to thank you and no words to congratulate you. All words fail here. It was better than the real. Though it is termed as virtual, but it was better than real. And the way it was organized, systematic, and how you proceeded, all of you, you deserve kudos. Lot more congratulations to all of you who have worked like a team, and it doesn't appear to be a virtual conference. It seems to be real and even better than the real. Heartiest congratulations to all of you, and may God bless the department and keep you all together.
Thank you very much.
Thank you, sir. Thank you.
That's all.
And I'm happy to share here that we were live streaming on the YouTube, and all the lectures, they are there for all times to come on the Botany channel.
Yeah,
So
that's
anyone
one great
can access to
advantage.
them anytime.
That is an advantage of this.
That's
Yeah.
हाँ नहीं नहीं उसका बात कर तुझे करना कुछ शेयर लखन पाल सर ने कुछ बोला ना Okay जी any the honorable editor in chief.
Well, I have been saying and observing from the day one, and it's been really a moment of great satisfaction that under so much of challenges, the way you have conducted, I have written this in the comment box also, that is, it's been organized in a most professional way. And the best part is that I think the kind of people who were glued to the proceedings and listening to each other, blessing each other, giving all kind of feedback and how to proceed ahead. That was the best part. And as far as the organization part is concerned, Meet sir, now I need not tell anything more about Patiala, wherein people like Atri Saab and today we met Sharma Saab and Jargan sir and Khattar Saab, so generous, full of humility, full of humanity, I would say. We can't ask for anything more than this. And we have seen a lot of good things happening this year, in spite of the fact that we had a very challenging time. But I think we all mycologists, we all scientific fraternity has come out very vigorously and taken this challenge head on, and the results are before all of us. Now, I sincerely wish to thank Dr. Raman for making all his presence throughout the proceedings and the hard work which he has put in, all the office bearers. And I'm particularly thankful to Professor Rupam Kapoor, whose presidential address was so inspiring. And after that, she presented a very good memorial lecture, K.R. Natarajan memorial lecture. And my all good wishes to the new incumbent president, Dr. Rodriguez. I know his coolness, I know his calmness and the kind of work he has been doing, but particularly the presence of Professor Lakhan Pal, Professor Bhatt, then Professor Atri, and all those senior mycologists for simply a great honor for all of us and blessings for all of us. Now, in the end, अवनीत जी, आप तो सूत्रधार बने रहे और जिस प्रकार से आपने पूरी इस प्रोसीडिंग्स को एक उसमें सूत्र में बांधकर प्रेजेंट किया है, I think that convergence of energy was phenomenal. So for this, you need to be applauded by everyone. You need to be applauded, and of course, Manruchi madam, जिस वेले नाल बैठे होते फिर energy इस तरह भी आंदी because she's always, saying, go ahead, do it. So that's the wonderful. And of course, the team of Punjabi University Patiala, I don't want to name all of them, but we know them personally, and I have seen the positivity in all of them. And this is how, things, they take very meaningful shape. So congratulations, and my earnest request to everyone that Kavaka is your journal, the journal which has been nurtured by the senior mycologist with a big dream. And we have been from the day one, Dr. Raman has been, requesting and and exhorting upon all the young mycologists that kindly contribute to this journal so that we make our India proud. Rather than going for some other platforms, I think we first need to nurture our own garden. We need to have flowers blooming in our own garden. And once we do that, I think that fragrance would be going all across the globe. So we can together do that. There have been wonderful award lectures, then presentations and other things. And I am sure, pretty sure, that all of you would be sending us very good quality articles to this journal. And I assure you that together with the kind of guidance I'm receiving from the earlier editor-in-chief, Professor Atri, and the secretary and the treasurers and all office bearers, including president and all that, I think we'll be able to get on to have some good scopus indexing, as well as we'll try our best that journal somehow get into some kind of impact factor list as well. So that's the next challenge, but that challenge would only be fulfilled once we get good quality articles from all of us. So thank you very much for making me part of this very mega event, annual event. And I'm very happy that all of us were on the same page with respect to moving ahead, taking mycology ahead. And for this, I think we always would be seeking blessings of our senior mycologists, accomplished mycologists. So thank you very much, and congratulations to Team Patiala. So big round of applause for Team Patiala.
Thank you, sir. Thank you so much.
Thank you. Thank you.
The kadis are more important, sir. When they come together, then they make a beautiful garland, and then they are right
Yes.
on the same page. So each and every kadi, that is equally important, and we have been lucky to have the contributors with us. Thank you very much for all the
Yes.
appreciation. And now may I request the Honorable President, who has been with us all the 3 days throughout without any break, and we salute to her dedication.
Before the president addresses, I have a suggestion to make.
Yes, sir.
You kindly seek recommendations from everybody, online recommendations, which should be sent to different ministries. Okay.
Sir, I was coming to that, sir. I was coming to that.
And then compile them by circulation.
Yes, yes, good.
Yes, sir. We will share the rough draft with all the members of Mycological Society of India. We will share it in the form of soft copy, and whatever recommendations will be coming back, suggestions, they will be incorporated into the final draft, and then we will be sharing it with all the agencies to whom we can share, and we can have the progress in mycology.
Yes,
yes,
it is required.
Very much, sir. So as Dr. Atri has rightly highlighted, we will make a draft, and it will be shared with all the members of MSI through an email, and then you can extend your suggestions, and they will be incorporated and will be shared. So
Sure.
may I request the Honorable President for the remarks for this session.
Good evening. Our chief guest, Professor Mangleshwar Sharma. Mrs. Sharma, first of all, I would like to congratulate you for the release of your volume. It's indeed an immense work of 57 years that you have compiled. That really needs to be applauded. Congratulations, sir.
Grateful.
The smile on Dr. Avneet Pal's face shows about the success of this event. It's an east to west smile which is writ on his face that largely tells about this event. Good evening, Professor Lakhan Pal, Professor Bhatt, Professor Raman, Professor Bhagyaraj, our President, Professor Rodrigues, Professor Kharwar, Professor Atri, Professor Deshmukh, Professor Acharya, Professor Dargan, Professor Kelai Selvam, the names, Professor Hande, whatever the names I'm reading, they are here with us till the valedictory function again says about the success of the event. I want to thank you all for being with us during this 3-day conference. We have reached the end of the final day of the conference, and with this, we shall be drawing curtains on such a lovely and stimulating discourse with the promise to reunite again in the next meet this year, end in the November. The conference aims to provide a common platform for deliberation on thematic areas of fungal diversity and biotechnology. I think it is fair to conclude that this event was a great success. So many people have contributed in so many ways to turn this event into a smoothly running meeting with many very interesting presentations and posters and a very good atmosphere for discussions and networking. It has been a great learning and a rewarding experience to be an audience to eminent scientists such as Professor Bhatt, Professor Manorachari, Professor Bhagyaraj, Professor Lakhan Pal, and Professor Atri, who have devoted years of dedicated research to the scientific fraternity. Although we did not get chance to physically interact to exchange innovative research ideas, seek collaboration, and inquire about the welfare of our near and dear ones over hot tea or lunch, we also missed enjoying the vibrant, colorful cultural evening that we have at Patiala. Nevertheless, online meeting has extended the advantage of listening to some eminent mycologists from abroad. To name some, Professor David Hebbett from Clark University, Professor Edward Langer, University of Kassel, Dr. Rue from University of Oregon, Dr. Rex Sedapa, University of Philippines, Professor Suresh from Kenya University, and I wish to thank them for presenting their talks in the late evenings or night hours, depriving their sleeps. I could see many of our members being benefited from these talks. Generally, any meet with similar proportion of foreign participation is considered an international meet. I wish to thank organizers for arranging these talks. I would like to thank all the speakers, chair and co-chairpersons of different sessions for sharing broader perspectives in their respective fields. And most importantly, on behalf of MSI and on my behalf, I congratulate our young dynamic research scholars and young scientists who will be holding the baton of mycology in future years. Each one of you needs to be felicitated for the wonderful work that you are doing, irrespective whether you have been awarded or not. We cannot give up trying, doing our best, creating our best, and being our best. Keep working hard. I feel honored and equally exhilarated to have been entrusted as a president, even though for a truncated period of the society that envisions promoting development of mycology in India in all respects. With this being said, I wish to congratulate the organizing committee of this conference, whose enthusiasm and support have allowed all of us to virtually gather in this groundbreaking exchange of views and perspectives. I would be failing in duty if I won't mention my sincere gratitude to all office bearers of MSI 2020, especially Professor Raman, for his enthusiastic work and coordination among the members of MSI, Professor Reddy, Professor Atri, for their cooperation and efforts. I request all virtually gathered here to please unmute your mic for a few minutes and extend a loud round of applause for the team of organizers, Professor Atri, Professor Manruchi Kaur, Dr. Avneet Pal Singh, and for their untiring efforts, and their colleagues Professor Khattar, Professor Gitika Sirhanti, Dr. Manish Kapoor, Professor D.P. Singh, Dr. Arneet Grewal, and Professor Gurpal Singh Dhingra. Thank you, all of you. It was really very wonderful attending these 3 days. It was a treat for any mycologist, and what more complimenting it could be that Dr. Arneet Grewal, she said she's tempted to move across the fence. Well, you are most welcome and join as a mycologist here, Dr. Grewal.
Ma'am, the entire team is there.
Congratulations to all of you for the wonderful work. I really wish
Congratulations, everyone.
I sincerely wish that MSI meetings will continue to be constructive forum for deliberations on how progress can be made in the field of fungal biology, taxonomy, physiology, as well as applied aspects. I hope we will be physically gathering in the next conference of MSI scheduled in Meghalaya under the able stewardship of Professor Rodrigues as president and Professor Manruchi Kaur as vice president, and it will be a grand success and will present the budding scientists with wider horizons.
Yes,
I believe that knowledge sharing is an important part of what we do, and a forum like this imparts us with ample opportunities to work towards building connections with people having similar interests, trade ideas, and ruminate on newer aspects of science that can be explored. At this juncture, I would like to thank all the past presidents who have laid the successful foundation and have guided us in this direction for the development of our society to the highest peak, and also congratulate the new members who have come on board. Welcome to all of you. Hereafter, Professor Rodrigues will shoulder the august responsibility, and I'm sure he will perform even better. I quote what he mentioned in yesterday's mail: "We will come and go, but our society will remain forever. Let us leave a footprint that will immensely satisfy us that we have contributed in a small way to the cause." A small sentence, but speaking loudly about his intentions to work as president. Congratulations, sir. I welcome you and I hand over the baton to you for 2021. May you have a very successful and fruitful tenure. Thank you very much.
Thank you very much, ma'am, for your
excellent
words. It's absolutely inspiring to all the generations, be it the younger ones, be it the colleague ones. So it's absolute delight to have you as president of Mycological Society of India. Thank you very much, ma'am.
Thank you.
It's been a
Now,
privilege. Thank you, Bhatt.
now may I request the chief guest of the day, Professor M. Sharma ji, for his remarks. Professor M. Sharma.
Well, thank you so much. Before I start reading it, let me express my gratitude to the organizing committee. Not only they released the book, but they have given me an opportunity to be with you and share some of my views about this. Dear Dr. Manruchi Kaur, convener of the conference, Dr. G.I.S. Khattar, co-convener, Dr. Avneet Pal Singh, organizing secretary of 3 days virtual national conference on biodiversity and biotechnology of fungi, and 47th annual meeting of Mycological Society of India under the able leadership of Dr. Rupam Kapoor, President, Dr. N. Raman, Secretary, MSI Officer, Bearer of MSI and delegates. It is my pleasure to address such a dynamic gathering from far and wide. The conference was inaugurated on the 21st of February, and the organizers have successfully conducted 61 lectures and presentations, which include Lifetime Achievement Award Lecture, Memorial Lectures, Young Scientist Award Lectures, Mid-Aged Scientist Award Lectures, and lead lectures along with 40 poster presentations spread over 11 sessions during the last 3 days. Professor D.J. Bhatt, senior mycologist from Goa University, was conferred with a Lifetime Achievement Award by the Mycological Society of India. Delegates around the globe have been a part of this revered gathering. Students from the field of mycology attended the conference, which include Professor Rupam Kapoor, Professor N. Raman, Professor C. Manorachari, Professor D.J. Bhatt, Professor N.S. Attri, Dr. Bhagiraj, Professor Naveen Kangu, Professor T.N. Lakhanpal, Professor R.N. Kharwad, Professor Sarita Nadrat, Professor M.S. Reddy, Professor Satyanarayanan, Dr. Sunil Deshmukh, Professor G.S. Dhingra, Professor Geeta Sundari. A special mention must here be made for the renowned mycologists who joined from out of India. Professor David Hilbert from Clark University, Professor Edward Menger from Kassel University, Germany, Professor Rex Sadava from Philippines, Professor Rao Wedgift from University of Oregon, USA, and Dr. L.M. Suresh from CMMYI, Kenya. And I feel happy to see that the Department of Botany has kept with the tradition of hard work and meticulous planning, which has helped them not only to keep pace but also excel in the field of modern scientific studies. I congratulate the faculty, students, technical staff of the department for successfully completing the deliberations. The young scientists have a lot to take away from such gatherings, as these offer an insight to the prospects and application of fungi for the welfare of society. And once again, thank you all the delegates. Thank you very much.
Thank you very much, sir. It's an honor to have you with us. Now we are moving to the concluding part of this conference. And for that, may I request Professor J.I.S. Khattar. The complete name is Professor Jasveer Inder Singh Khattar. And may I invite him for the formal vote of thanks. Professor Khattar, please.
Thank you very much, Dr. Ravneet. Good evening, everybody.
Good evening, sir.
Good evening, sir. So we are at the concluding part of a 3-day national conference. It was I will say, very new experience for all of us, and with your cooperation, we are quite satisfied that we are successful in this event. This has been possible due to cooperation of all of you, and for that, all of you deserve appreciation and thanks. So it is my pleasant duty to propose vote of thanks. So first of all, I on my behalf and behalf of the organizing committee, we should thank our Vice Chancellor, Madam Ravneet Kaur, for her continuous support and guidance for organizing this conference. Besides this, we have received very logistic support from other officials of the university, including Dean Academic Affairs, Registrar, then Finance Officer. So we are quite thankful to all of them. Besides this, we are now presently sitting in the lab of Computer Center. So it is our personal and special thanks to Dr. Kamaljeet Singh, Director Computer Center, and his team, especially Mr. Ashu, System Analyst, and staff of UGC, HRD, HRDC, and all staff members of this HRDC. Then special thanks to Mycological Society of India, especially President Madam Rupam Kapoor, Secretary Professor Raman, and office bearers and executive members for choosing this place as of that is the place, place for venue for what we can say for this conference. So thank you very much, all the office bearers. This conference has not been successful without the active participation of delegates from all over India, beside this other parts of the world, especially I wish to thank Professor Vandergift, Professor Swade, Professor Manorachari, Professor Langer, Professor Suresh, Professor Lakhanpal, Professor Bhaggaraj, Professor Bhatt, Professor M.S. Reddy, Professor Attri, Professor Gopal, Professor Dargan, and Professor Jashpal Sharma. So senior mycologists who have always guided us in organizing this conference, and your presence in this conference has motivated not only us but all other participants. At this moment, I would like to congratulate newly elected President, Professor August, and Vice President, Madam Ruchi. Besides this, I wish to congratulate Professor M. Sharma for giving this 3-volume Flora, and which was released today. I also wish to congratulate award lecture winners and MSc fellow awardees, which have been announced by the secretary this evening. I'm extremely thankful to my colleagues, Professor D.P. Singh, Professor Manish Kapoor, Dr. Geetka Sarandi, Dr. Arneet Karewal, non-teaching staff of the department, research scholars, and students, because to organize this type of event, it requires a lot of planning, and different duties are assigned. So all of them have contributed immensely. So our special thanks to all members of the botany department. So it is a great pleasure that we have been successful in organizing this event, but I'm sorry to say that we could not provide you the hospitality. You really, so it was our wish that if you have been here, then we have enjoyed more. And hopefully next time, whenever any event is organized at Patiala, we will be happy to shower our love on all of you. So
we will avoid. We will avoid. We will avoid and come during those times.
Sir, I am enjoying on behalf of MSI. I am enjoying here all hospitality.
So it is a. I will say. Yes, Professor Dargan, please.
Yes,
So,
please. I'm listening. Thank you very much.
Okay. So, okay, it is as it is said that we should go as the time is there. So, due to COVID-19, we have to organize this type of conference, but it is a, I will say, not only a new experience, but it's a learning experience. We have learned many things by this mode. So hopefully next time we will meet in person in organizing this conference. So thank you very much, each and everyone. And we are departing at this moment to meet next time in the next 48th meet of the MSI. So thank you very much, each and everyone. And I thank Professor Avneet for giving me this opportunity. This is very pleasant duty to thank, to propose vote of thanks. Thank you very much.
Thank you very much, sir. It's an honor.
To cast their shadows before. So you've been addressed professor because
Professor.
you're soon going to be professor.
Yes, very true.
Ma'am, we are already waiting for 2 professors to be formally announced. Dr. Manish and Dr. Geetika, they are due for their professorship. Dr. Manish has faced the interview. Dr. Geetika has applied, and soon we will share the good news of new 2 professors with us very soon with all of the members.
Yes.
Thank you very much for your
Congratulations in anticipation.
Thank you, sir. Thank you.
Yes.
So we will have the national anthem now, and after that we will sign off.
Dr. Raman. Dr. Raman has to thank.
Yes, sir. Yes, sir.
Dr. Raman.
Another part.
Request Dr. Raman.
Yes, sir.
Yes. Yeah. You request. you make the announcement. Hello? Hello?
Yes, sir.
There is on agenda is there. So okay, I will start. I will start.
Yes, yes. Start.
I will start, no problem.
He is muted. Actually, he is muted. So he is not.
How many?
He is muted.
Yes, sir. Yes, sir. Yes, sir.
You ask Dr. Rahman to.
Sorry.
Thanks on behalf of MSI.
Yeah, I forgot that. May I request the Honorable Secretary MSI to propose vote of thanks on behalf of MSI? I'm sorry for that.
Yes. So, dear members and all the faculty members of Botany Department, Punjabi University, Patiala, 2019 is a challenging year for the human survival. So that year, the Patiala Punjabi University was allotted the MSI 47th annual meeting, and the MSI and the organizers, they joined hand together, just like a mycorrhizal fungi, beneficial mycorrhizal fungi, and worked day and night for the conference. So even though some request was there to postpone, cancel like that, no, the organizers stood strongly to conduct the meeting. They successfully conducted the meeting. The conference and the annual meeting is an excellent and decent meeting. An excellent and decent meeting conducted by the Punjabi Botany Department, Punjabi University, Patiala. So on behalf of the MSI, I thank the Vice Chancellor of the Punjabi University, Patiala, and the Department of Botany Department, and Professor Atri, even though he is our past president, he worked for the department, for the conference effectively. Professor Munruchikar, the convener, the Qatar, Professor Qatar, the co-convener, Dr. Avneet Pal Singh, the young and dynamic, I can say just like a bee. So bee always work hard. So work together. The CBS symbol of motto is CBS always say together, work together, or together we can work. So the MSI and the organizers together worked, and Avneet Pal Singh, with the help of the convener and co-convener, he worked hard. And on behalf of the MSI, I thank the moderators, Dr. Arneet, Dr. Geetika, Dr. Manish Kapoor, Professor DP Singh, the whole team of research scholars, non-teaching staff, and students, Professor Dargan, Professor Dingra, and all the MSI members and the speakers, everybody, and Madam Munruchikar, the convener, please. Convener is there?
Yes, sir. There, sir.
Madam, the Punjab is with gold.
Thank you, sir.
Golden temple at Punjab.
Thank you, sir.
We could not give the gold, but we will give you the golden plated, gold like plated.
Thank you so much, sir. Thank you.
Appreciation certificate to Professor Munruchikar on 24th February 2021 by Mycological Society of India in appreciation for organizing the 87th annual meeting of Mycological Society of India on February 22nd to 24th, 2021 at Punjabi University, Patiala.
Thank you so much, sir. Really indebted. My whole team is really indebted, sir. Thank you so much.
The copy of this one is already in your inbox.
Thank
and
you so much.
you by post.
Thank you so much.
For seeing the young and energetic organizing secretary is there.
He's there.
Amanit.
Yes, sir.
Amanit. Dr.
Yes, sir.
Amanit. Yes.
Yes, we deserve.
On behalf of MSI, I am very glad to hand over this virtually to Dr. Amanit Pal Singh, the certificate of appreciation to Dr. Amanit Pal Singh on the February 2021, 24th February 2021, Mycological Society of India, in appreciation for organizing the 47th annual meeting of Mycological Society of India at Punjabi University, Patiala. So we always work together and make the success a Himalayan success. It is already in our inbox.
Thank you very much, sir. I dedicate this honor to the team Botany Patiala. It's absolutely because of the entire team. So all 7 people have been reserved, and without whose contribution, this would not have been possible. Each one of
Next meeting, 48th annual conference of Mycological Society of India at Meghalaya, we will meet together. Thank you very much.
Thank you very much, sir.
Thank you, sir. Thank you. Thank you, each one of you, sir.
Now we'll stand in respect of the national anthem, please.
Congratulations.
Congratulations.
Yes. Yes.
Thank you very much, one and all.
Thank you.
Thank you. Meghalaya.
You all.
All of us should meet in Meghalaya.
Greetings.
Thank you. Namaskar, ma'am.
Thank you all. Thank you very much.
Thank you, sir. Thank you, Bhagiraj, sir.
Thank you. Indira to baat nikalangi.
Congratulations, man Ruchi.